There's plenty of room up front, so please, folks in the way back, if you wish to hear and see, we can even get some more chairs or bring those chairs up. Before we actually get started with our first speaker, just a few notes. The moderators will flash you with warning signals. It's a sophisticated electronic system. So this means that you have 5 minutes left of your 9 minutes. This means that you have 2 minutes of your 9 minutes left. This is, you're done with your 9 minutes, and so if you continue talking, you will eat into your 3 minutes of question time, okay? Also, for visitors, great to have you here, very welcome. You may have already found the bathrooms, but if not, you go out the walkway, past reception, hang a hard right, and the bathrooms will be on the left side. And there's even some secret ones outside that are rarely occupied. And if they're occupied, I'm in there. And there's the part.
Se hace como para atrás ahorita porque ahí solo se le ve la mitad
La mitad de pero no está transmitiendo. Me dijo, alguien quería saber si está como grabando eso.
¿Sí? Sí. O sea, va a quedar en el
Welcome everyone to the 31st annual Fall UCEAP Simposyum. My name is Frank Joyce. We've been working with students on their independent projects, in some cases starting about a year ago, as some of you were inquiring about what you could do and what lab equipment is here. This is the result of 2 weeks of field work and thus far about a week and a half of analysis and writing and so forth. And we have 6 sessions and we'll be taking breaks. Sorry for we're a little behind, folks on YouTube. Remember that we'll give you a signal with respect to how much time you have left, and afterwards the moderator will help with questions and so forth. Generally, we like to let guests and students ask questions first, and staff, if there's time left, will ask questions, okay? So our first speaker today is Sasha Padilla, and she will be talking about microbes of Fish-urchin.
My name is Sasha Padilla, and this is my presentation on characterizing the integument microbiota of Costa Rica's marine organisms in Costa Rica's tide pools. So these are the organisms I studied in my project. I studied the sea cucumber, which is in the genus Holothuria, the brittle star, which is the species Ophiocoma aethiops, and then sea hares, which are in the genus Phytola brifera. Brittle stars and sea cucumbers are echinoderms, and so they have a calcium carbonate endoskeleton that's embedded in a thin layer of epidermis, whereas sea hares are gastropods and they're part of the mollusk phylum. They also have a shell, so they're soft-bodied, and they can defend themselves by secreting a mucus opaline substance, which is neurotoxic to their predators. And so for some more background information, many marine animals have a diverse skin microbiome that are dominated by phylums such as proteobacteria and cyanobacteria, and just to name a few. Most research on skin microbiomes has been done on humans, and very little has been done on marine animals and let alone invertebrates. Tide pools are unique because of their unique abiotic stressors. So the salinity and oxygen content fluctuates throughout the day. As the tide goes out, the water left in the pools evaporates and leaves behind salt. And so that creates a harsh environment that's hard for microorganisms and just normal organisms to live in. And during my project, I learned that tide pools are home to a lot of phytoplankton, which are marine microscopic algae. The thick they fix nitrogen and consume carbon dioxide. So they're very valuable for the marine environment. And they can be classified into diatoms, dinoflagellates, coccolithophores, cyanobacteria, and green algae. And there's been very little research done on what microorganisms exist in the tide pools and how they interact with the animals there. And so learning about this could be very telling about the health of the tide pool organisms in the face of climate change as sea levels rise and the water warms and becomes more acidic. So this could be very valuable information. So this leads me to my central question: what microbes are present on the integument of sea cucumbers, sea hares, and black spiny brittle stars, and how do the microbial communities differ between these organisms? And at the beginning of my project, I hypothesized that the integument of these creatures hosts a plethora of bacteria, archaea, and other microorganisms. Each creature hosts a distinctive microbiota on their surfaces. And so for my methods, every day, pretty much between November 18th and 29th, I went to the tide pools at low tide and I collected specimens to bring back between 3 and 7 per time, depending on how much time I had to analyze them. And so I brought them back to Casa Verde, and I kept them in an aquarium that Leia helped me make with an aerator. And when I was ready to analyze them, I swabbed the top of them and then transferred it to a slide. And then I examined that slide with a microscope at either 100 or 40, 100, 400, or 1,000 power. And I took pictures of the microorganisms I saw, and kept that in a file. And then when I got back to Monteverde, I looked at them on my iPad and I circled which microorganisms were there. And I was able to classify 799 microorganisms into 21 morphospecies. And so this is one group of morphospecies that I did, so diatoms. And so I identified 14 morphospecies. And then I identified 4 non-classifiable ones. I just wasn't able to put those in a distinct group. And then I found 2 cyanobacterial morphospecies and 1 coccolithophore, and then 1 bacteria, but it moved really fast, so I couldn't take a picture of it. It was just a small round bacterium. And then I also found some protozoa, which could have been predators of marine microorganisms, or parasites, or just like herbivores over there. And so here are my results. I compiled them into a table, and I found that the sea cucumbers harbored 443 microorganisms on their surfaces, which is the most amount, and then sea hares harbored 253 microorganisms, and then brittle stars had the least amount, which is at 103 microorganisms. And in total, I had 10 sea cucumbers, 8 black brittle stars, and 4 sea hares. And then by percentage, in this little graph thing, diatoms made up the biggest portion of the microorganisms, followed by non-classified, and then the other groups were smaller percentages. And also, and this is not shown on the slide, but diatoms had the highest species richness and alpha diversity value out of all of the morphospecies groups. And then I also calculated some measures of alpha and beta diversity. So for beta diversity, I used the Gray-Curtis dissimilarity values, and 0 being the most similar, and 1 being the least similar. So I compared the microbiomes of one species to all the other ones, and I found that sea cucumbers had skin microbiomes that were most similar to tide pool water, whereas brittle stars had skin microbiomes that were least similar to tide pool water. And then using measures of alpha diversity, I also saw that sea cucumbers, brittle stars, and sea hares all had high alpha diversity, which was over 0.8. And then, sea cucumbers, sea hares, and tide pool water all had alpha diversity over 0.8. And brittle stars had alpha diversity that was much lower at about 0.2. And so this kind of led me to this conclusion, which is that sea cucumbers have a highly diverse skin microbiome that is similar to their environment. And I thought this might be because they're kind of small and like they don't eat very much. So that might give microbes like a bigger chance or a greater chance to colonize their surface. Whereas brittle stars had a skin microbiome that was highly distinct from their environment, the tide pool water, and also the other microorganisms. And I kind of thought it might be possible that their hard exterior is unsuitable for colonization by microbes. This might be an evolutionary tactic. They may evolve their exterior to resist predation, but also to resist colonization by microorganisms and also like biofilm formation. And the drastically lower amount of microbiota on brittle stars may also indicate the possibility of antimicrobial properties. And then something that I thought was interesting was that I didn't really find any bacteria in any of the organisms. So I kind of thought this could be maybe that the diatoms competed for space with bacteria, and the bacteria weren't able to outcompete them. It's also possible that the animals derive more from coexisting with diatoms than they do with bacteria, so they might benefit from the nutrient cycling and other processes that diatoms do in the tadpoles. It's also possible that cyanobacteria are producing toxic compounds that are killing off the bacteria, which is a researched phenomenon. And so all this could be reasons why there was no bacteria on my samples. And so my conclusion is that brittle stars have highly distinct integument that may indicate the presence of antimicrobial properties or anti-colonization mechanisms. And then further research would be useful in further characterizing the microbiome and studying how microbiota changes under different environmental conditions. And then information on the microbes present in tadpoles is invaluable to learning how to maintain their health and the health of the creatures that live there. So thank you to the Monteverde Institute. Leo, he helped me so much. It would probably be a much different project without him. Frank and Naomi for being my advisors, and then Marisa, Claire, and Bryant for keeping company at the tadpoles, and then my host family for giving me food and shelter and being really nice.
Lots of time for questions.
What were some of the challenges you experienced, like in identifying the micro species, like morphologically? Because like in the pictures, it looked pretty difficult.
It was really hard. It was actually like the worst day ever. It was so hard. I pretty much just like I was on my iPad all day. I kept like circling things, and then I did a lot of research as to like what diatoms look like, or like what like dinoflagellates look like. And I kind of made like my best, most educated guess and like tried to match them to a certain category, but it was really hard to like one, see them, and then two, like determine if it was like one thing over another. So yeah, it was very challenging. It was probably like the most challenging part.
Yes.
Why did you choose the focus of your slides, like the different steps?
Like the
scale that you were studying each slide at. You said you were looking at it with 40 times
yeah.
and double evidence.
Well, pretty much I kind of just decided to go in order from like lowest power to highest power, so that like in highest power you can see things that you can't see in lowest power. And like with the microscope I was using, highest power was kind of, or I think it was 10 power, was the most like crisp view I could see, whereas highest power, like you can see smaller things that you can't see at higher powers, but it's a lot blurrier, so it's harder to like pick out which are microorganisms and which are just like dirt. So I kind of just did all of them for different levels of like focus. Yeah. Yes.
What exactly is a diatom, and do we as humans have them as well?
No. So a diatom is like algae. It's just super, super small, and they're basically just plants. Like they photosynthesize, and they have chlorophyll, and they're just kind of like marine plants. Basically, they're just really small, and they lie on the surface of the water. And humans don't have them because they're plants. Yeah.
Thank you, Sasha. Our next speaker is Wendy Yang from UC Santa Barbara, and she will be talking about antibacterial properties of fungi in Monteverde.
So we're going to get a little deep for a moment. Raise your hand if you can think of someone who is in medical danger every time you step out of the house. All right. And would anyone like to share who? You don't have to. Well,
My grandma.
thank you. Well, it's scary. My stepdad, he was intubated for a month due to COVID, and he faces a lot of complications from it still. And I understand that sinking feeling every time he goes to the ER. Well, I'm studying the percentage of fungi in the Monteverde cloud forest with antibacterial properties specifically against Staphylococcus aureus and Escherichia coli, the top 2 pathogenic bacteria that have multi-drug resistance. And to all the nail biters and people that eat spoiled food, you're probably going to be fine. But with this rise in antibiotic resistance, you could face more severe symptoms and longer healing times. And not all resistances are created the same. The World Health Organization lists antimicrobial resistance as the top 3 critical human health problems. The top 6 bacteria with multi-drug resistance cause 72.1% of all deaths, or let me say that again, of total deaths in 2019 due to antimicrobial resistance. And this is due in part because we're using the same antimicrobial or antibiotic classes over and over, just making small little tweaks to each one with each new wave of resistance. But it doesn't have to be this way. Fungi, they're natural antagonists of bacteria, and there are so many of them. There's an estimated range of 1.5 to 12 million species, but only 150,000 of them are described. That means that 90 to 99% of them are undescribed. And in a 2006 study, it was found that 30 to 80% of all of fungal strains had antimicrobial properties. That's a huge source of novel antibiotics. So what I did was find 21 fungi fruiting bodies along trails of the Estación Biológica or along Ruta Nacional Terciaria 620 between the Estación Biológica and Monteverde Institute. And just to clarify, the fruiting body is this part. It's what we call mushrooms, and it's where the spores are contained. And fungi have this entire system of mycelium that's like a thread-like structure that isn't involved in sexual reproduction. So I dried the fungi and then I grounded them up and added them to distilled water. Some of them I couldn't ground up just because they were too pliable, so I just submerged them in distilled water. And for others that were very easily groundable, I made a little homogeneous solution with them. And then I added 2 6mm filter discs into each extract. And for the positive control, I used Trozidex, which is an antibiotic eye drop that contains tobramycin. For the negative controls, I used just distilled water or nothing at all. I put 1 treatment disc per bacterial lawn, and then I incubated all the dishes at 37 degrees Celsius for at least 24 hours. And 37 degrees Celsius for all us US people is body temperature, which is perfect for pathogenic bacteria. And just before I get into the results, let me make sure that we're all on the same page. This is my control plate, and this is the positive control, negative with distilled water, and negative with nothing. This dark ring around here is an area where no bacteria grows, because this entire plate has bacteria except these 2 points. And I'm going to refer to this area or this distance as the zone of inhibition, or ZOI. Fungal treatments that have more antimicrobial activity have larger ZOIs, whereas these 2, they're negative controls, they have nothing. So on to the actual results. I had to omit 3 fungi. I really like them, but whatever. I had to omit 3 fungi because there was a lot of contamination around in this batch of agar dishes. These white specks, they are not characteristic of either E. coli or S. aureus bacterial colonies. So I just had to omit all the data to make sure it's not skewed. And this is a summarization of the percentage of fungi with antibacterial properties against E. coli and S. aureus. The y-axis is, the x-axis is the degree of antibacterial effects, and the y-axis is the percentage of total fungi per bacterial group. Let me break it down. So this group represents the fungal treatments that had any antibacterial activity, any ZOI. 6 out of 17 were effective against E. coli, and 7 out of 18 were effective against S. aureus. But most of them were under 1 millimeter. The ZOI was under 1 millimeter. So for the significant antibacterial activity, which are fungal treatments that produce ZOIs of larger than 1 millimeter, none were effective against E. coli, and there were 2 fungal treatments that were found for S. aureus. Oh, and this perfectly reflects the 2006 study about 30 to 80% of fungal strains having antibacterial activity. So, more results. This is the positive control again, and E. coli had a ZOI of 2.4, which is almost half of S. aureus' ZOI. And then looking over here, this fungus I'm going to call S3. It's of the order Polyploralis. It had a ZOI of 5.1 or 12, which is close, super close to the positive control. This fungus is, I'm going to call it S9, is of the order Xylarias, and it produced a ZOI of 1.0 millimeters. So why were the positive controls so different? It is probably because I used tobramycin, which is a hydrophilic antibiotic, and E. coli is a gram-negative bacteria, meaning it has an outer membrane that is especially selective against hydrophilic compounds in its environment. Therefore, tobramycin just isn't as good at penetrating into E. coli as it is penetrating into S. aureus. So the inhibition, or the growth inhibition, is not going to be as high. This could also explain why E. coli, or there weren't any fungal treatments with significant antibacterial activity against E. coli because of the extra layer of selective membrane. So why were S3 and S9 so good at inhibiting growth? One explanation is that I used distilled water for the extraction. This means that really only hydrophilic antibiotics are able to seep into the extraction. And this is also evinced by the lack of ZOI against E. coli, which is good at keeping out hydrophilic antibiotics for these 2 fungi. Another possible reason is that these 2 could just have higher concentration of fungi. S9 was super groundable, so many parts of the fungus was in the extract. Therefore, it probably, the fungal treatment was probably more potent. And then looking at S3, there's this white growth over here. And definitely it is not E. coli. And I suspect that it's the fungal mycelium because it looks really similar to what I saw under the fruiting body when I harvested it. And that means that S3 was in high enough concentrations for the fungus to develop and grow mycelium. All right, and another noticeable thing was S1, this fungus, and it had noticeable growth inhibition, but the ZOI was 0. And this also correlates to the non-circular ZOI, which probably means that it developed resistance. But because the bacterial concentration is so similar, like regardless of the distance, like say from here to here, that means that resistance developed way early on. And I only incubated them for like 24 hours. So that's super little time for them to develop resistance. And honestly, that's a sobering reminder of our arms race against antimicrobial resistance. But in spite of that, even put up against uninclusive methods, 35% of fungal treatments inhibited E. coli growth and 39% of fungal treatments inhibited S. aureus growth. So to all you nail biters and people that eat spoiled food, there's hope. But until then, let's just treasure our loved ones. I want to thank Naomi and Emmy for being my advisors, Cassie for peer reviewing, MBI for the lab space, and an informal thank you to Melanie and Ewan for listening to me until 1 a.m. yesterday. Thank you. don't jump all at once.
Yes.
So I actually don't know, for the existing antibiotics that are out there, what percentage of them ballpark come from a fungal background? Is that something that you discovered in your research?
Yeah, I was actually trying to search that up, but I couldn't find like really any answers. But I suspect like maybe I'd say less than 50%. 50%. Yeah.
How do antimicrobials actually work?
Well, a lot of them have super different mechanisms. Like some of them might attack the cell wall, some might attack like protein production. It's a huge variety. Yeah.
Yeah.
How did you know what to do to test this? How did you develop these methods?
Oh, girl, so many Google searches. I was so lost. But I'd say short answer, like 5 hours of Google. Yeah.
Do you think that the substances that have antibiotic properties in fungi are actually adapted, evolved substances to fight bacterial infection in the fungi themselves?
Bacterial infection. I don't know much about bacterial infection in fungi, but they do compete for the same resources, so I would say yes, there's always been an arms race between fungi and bacteria in the wild. Certainly, yeah. Same reason.
Is there a question? Okay. Maria. Thank you, Maria. Muy bien. Our next speaker is Emma Allende from UC Santa Barbara, and she will be talking about antifungal properties of mucal substances on the skin of neurons.
Hi everyone, my name is Emma, and today I will be sharing my findings about my research about antifungal properties of toxins and their potential resistance to diseases. So amphibians face unique vulnerabilities due to their biology and the environments that they inhabit. Their semi-permeable skin allows for vital processes like respiration and water exchange, but also makes them highly susceptible to environmental contaminants and pathogens. One of the most significant threats to amphibians globally right now is the chytrid fungus, Batrachochytrium dendrobatitis, or BD. The impact of BD cannot be overstated. It's directly linked to the decline of amphibian populations worldwide, with over 500 species currently being affected to varying degrees. In some cases, such as the golden toad, which I'm sure many of you are aware of, it has driven species to extinction. BD infects amphibians through their skin, disrupting their ability to regulate essential processes and often leading to death. Amphibian skin also hosts a diverse microbiome composed of bacteria, fungi, and other microbes. And among these microbes are species that can play a crucial protective role against pathogens. So cane toads in particular possess unique natural defenses that help protect them against various threats. One of their primary defenses is the secretion of toxins from their parotid glands, which are located right behind their eyes. These toxins are known for their potent chemical properties, which deter predators. However, recent studies, including my own, suggest that they may also exhibit antifungal properties. And this brings us to an important research gap. While there's evidence that cane toad toxins could play a role in inhibiting fungal growth, their specific antifungal mechanisms, particularly in the context of ET infections, remains largely unexplored. Cane toads are notably resistant to chytridiomycosis, the disease caused by BD, which brings me to my central question. Do cane toad toxins contain antifungal properties that protect them from fungal diseases? So to answer my essential question, I designed a study that would allow me to observe cane toad toxins and its interactions with fungal spores on amphibian microbiomes. I conducted nightly surveys across several locations in Monteverde, including these 5 right here. During my fieldwork, I used an approach to collect data and samples from amphibians. First, I located amphibians searching areas with suitable habitat conditions, and then once I found an amphibian, I swabbed its skin to collect samples for fungal analysis, ensuring minimal disturbance to the animal. I then recorded key details, including species location, and site descriptions. In addition to skin swabs, I also collected cane toad toxins from a singular toad by stimulating its parotid glands. After completing my field sampling, I then moved to my laboratory phase of my research, which was focused on analyzing the fungal samples and testing the effects of the cane toad toxins on these fungal samples. To begin, I sterilized all the petri dishes and prepared a potato dextrose agar plates, which is a nutrient-rich medium ideal for cultivating fungal growth. I then cultured the swabs collected from amphibians onto these agar plates to promote the growth of any fungi present on their skin. These plates were then incubated at about 21 degrees Celsius for 48 hours, providing optimal conditions for fungal growth. And then once the incubation period was complete, I examined the fungal growth under a microscope and documented the extent of these colonies grown. So I divided my experiment into 2 treatment groups and included a control for comparison. In treatment group number 1, I allowed fungal colonies to incubate for 48 hours before applying cane toad toxins to them to one side of the petri dish. And this approach allowed me to monitor the toxin's effects on already established fungal growth. For treatment group number 2, toxins were applied at the same time of inoculation, which allowed me to, and these were alongside the swab samples, and this allowed me to observe whether the toxins could inhibit fungal growth from the very start. Additionally, I also included control plates where no toxins were applied at all, and these controls provided a baseline for fungal growth in the absence of cane toad toxins, and allowed me to directly compare the results and better understand the toxin's impact. So here I have my results from both treatment groups. On the left side of each petri dish where the cane toad toxins were applied, the fungal growth was completely inhibited. You can see that right here. This is consistent across both treatment groups regardless of whether the toxins were added after incubation or before incubation. You can see this most clearly in certain dishes where the division between treated and untreated areas is very stark. The untreated side shows significant fungal growth, whereas the treated side remains clear and very smooth, demonstrating the effectiveness of the cane toad toxin in combating fungal growth. These results strongly suggest that toxins do exhibit potent antifungal properties, validating my hypothesis of their potential role. So out of the 8 amphibian swabs I tested, the results were consistent with my hypothesis. In 6 of the petri dishes, the toxins caused complete inhibition of fungal growth where they were applied. In one dish, there was slight inhibition, and in another, the toxin had no observable effect. And the overall trend remained clear. In all 8 dishes, no fungal growth was observed directly where the toxins were placed. So my results strongly support the idea that cane toad toxins exhibit antifungal properties. Significant fungal growth inhibition was observed, with most toxin-treated sides of the petri dishes showing either reduced growth or complete inhibition. This aligns with the hypothesis that these toxins could play a protective role against fungal pathogens. So when compared to previous research, these findings suggest that Amphibian reliance, which are the primary compounds in the cane toad toxins, may have stronger antifungal effects than earlier studies have indicated. While prior research reported around 50% inhibition, my study observed near complete suppression in most cases. There have also been studies that have delved into the idea that bufadionolides actually enhance the growth of anti-BD bacteria, which provides a multifaceted defense mechanism against fungal diseases like chytridiomycosis. The significant inhibition of fungal growth observed in my research suggests that these toxins could play a role in mitigating the impact of chytridiomycosis. Although more research is needed to fully understand the mechanisms involved, these findings open up exciting possibilities for cane toads and further exploration into biological defenses against fungal pathogens that threaten amphibian populations globally. Before I conclude, I would like to quickly acknowledge these people for helping me throughout my project and really making this research possible. So thank you to Federico for being my primary advisor, Naomi for my secondary advisor, Frank for teaching me so much, same with Emmy, and then to my host family parents, my parents, and then all the staff at the Institute and the station.
Lots of time for questions. Yes?
How did you go about extracting the toxins? That sounds pretty easy.
So as I said, I stimulated the paratoid glands, and then it's like essentially milking it for its toxins, and then I collected it and put it in these test tubes, and then I stored it in the refrigerator.
How do you think this can be applied for like amphibian conservation? Because like you can't really apply the toxins directly to like amphibian skin, right? Because it's toxic.
Yeah,
So
that's a good question. I think it's more about learning more about which amphibians are more affected and why, because that's kind of an unanswered question right now in the amphibian world, because they're not really sure of like which amphibians are affected. So it's just like learning more about the disease.
What was the method you used to apply the cane toad toxin to the petri dishes, and how did you like measure the results of it when it produced like the inhibiting growth?
So I used swabs, and I did it on half of the petri dish. I just put it over the already. So first I swabbed. I put the swabs of the amphibians on the petri dishes, and then I put the cane toad toxin swab over that to see the inhibition. Yeah.
Do you think the development of these antifungal properties in cane toads are like as a defense against the chytrid fungus or other fungi, or are they, I don't know, like?
I think it could be some of both. It's originally, like the toxins are originally for defense against predators, but there are recent studies that have found that the toxins are producing anti-BD properties, which is very interesting. So yeah, maybe some of both. Yeah.
Do you think that these antifungal properties could have pharmaceutical applications as well?
Yeah, I do think so. They're currently looking into using this in the medical field. Cane toads are a big topic in that area. So I think it could be really interesting for novel discoveries.
I was wondering, like, chemically, how you think I can't remember exactly the pronounce the word of the toxin would be affecting the fungal, like where the actual mechanism is.
That's a good question. I think that would require further research and better materials than we have here. I have very limited materials, but that would be a very interesting topic to research.
Thank you. Our next speakers are Jaden Malanese from UC Berkeley and Holden Shivers from UC Santa Barbara. They will be talking about antibiotic effects of plasma from American crocodiles.
Hello everyone, my name is Jaden Manalesi.
And I'm Holden Shivers.
And our research project is on the antibiotic effects of the American crocodile plasma against Escherichia coli. Staphylococcus aureus and Staphylococcus epidermidis.
Cool. So the background for this project is we were listening to a conservationist speak about his work in the Amazon. He was mentioning when the extraction of a blotfly is failed, it leaves an open wound for infection. And the way they treat that out in the field is by using an indigenous method of applying blood from a black caiman to the site. So that got us thinking of maybe there is some sort of like intrinsic property of crocodile blood that has antibiotic properties and could be used as an antibacterial. We looked further into it, we found that there actually is very recent research done demonstrating that there is promise of a new source of antibiotic inside crocodile plasma.
This led us to our research question: does plasma from Crocodilus acutus present antibiotic properties? So over the course of our study, we were challenged with 3 challenges in regards to our methods. The first was safely capturing crocodiles in Kwahini Kill. Second was humanely performing a blood draw on the crocodiles out in the field. And lastly was conducting lab work to test the crocodile plasma for antibiotic properties. So in regards to the capture, we were accompanied by Madai and Stephen, both of whom are locals from Kwahini Kill, who have experience capturing and handling crocodiles. Here's a video of our process. Sorry it's so small, it should be bigger. But we would go out at night and we would use our headlamps to look for the eye glimpse of the crocodiles, and then we would safely and slowly approach them and grab them by the neck just behind the jaws so they can't bite us. And then out in the mangroves, we would utilize a gill net and encircle the crocodile, either going around it completely or encircling it or trapping it against the mangroves. And yeah, there's a video of holding, gaining control of the crocodile. As soon as we gain control, we would tape the mouth shut so that it can't bite us, and then also make sure not to tape the nostrils so that the crocodile could still breathe properly.
So the next challenge we were faced with is how to perform a blood draw on a crocodile. I have previous experience doing in vivo research on mice, and I've drawn blood a couple of times from there, but obviously that's completely different than a crocodile. I consulted with a crocodile researcher, Davinia Benito, and she instructed us on the most humane way to draw blood from a crocodile and how to get the right amount of blood for our samples. She told us that the best way to do this is to insert a syringe at 45 degrees into the post-occipital venous sinus, which is located in the middle of these scales here. That's roughly on the scruff of the neck, like if you were to put that on a human. And you insert that until you get a red flash of blood in the syringe at high negative pressure. So here we clean out the site with iodine, make sure it's sterile. Insert the syringe at a 45-degree angle, and once you see a little bit of red in the syringe, you apply negative pressure until you have roughly 1 milliliter of blood from the crocodile. After you have a milliliter of blood, you're going to pull out the syringe from the crocodile. You're going to reapply iodine on the site to make sure it's sterile again. And then you're going to take off all the tape around the crocodile's snout so it can go back to living its life. You make sure it's responsive, happy, and healthy, and you put it back in the same spot that you extracted it from. So this is a release. All of our releases went perfectly fine. All the crocodiles responded really well. And this is us applying the blood to a 4 milliliter ETA tube. This prevents the blood from clotting so that you can keep it on ice and transport it back to a centrifuge and centrifuge out the plasma that we would use for the lab portion of our experiment.
So for our lab methods, we decided to run an agar disc diffusion assay where we use 4 different treatments. Obviously the crocodile plasma, then we use DI water as a control, and then we also use the antibiotic Trazodex to compare the plasma to something we know that works at inhibiting the growth of bacteria. And lastly, we use my own plasma, which we drew from a clinic here in Monteverde, separated the plasma from the blood, and we use this as a comparison to make sure that the antibiotic properties found in crocodile plasma aren't just common attributes found in other animals.
So this is a video of us applying the treatment to all the different filter papers. Essentially, it's the same experiment that when you ran, where you grow a uniform lawn of bacteria, then you separate it out into 4 quadrats. We ran this experiment blind, which means that one of us knew the location of each treatment and had a key. and the other person measured the actual zones of inhibition. So we try to remove our own bias in that method. This is us making a uniform line of bacteria right here, and this is us applying the treatments to all different corners of the dish. And then I applied the treatments, and Jaden measured them with calipers to determine the zone of inhibition, not knowing which treatment was which, and then I applied the key and collected the data afterwards.
And these are our results after incubating our dishes for 18 hours at 37 degrees Celsius.
So this leads us into the results section of our study.
So this chart shows the antibiotic performance of crocodile versus human plasma. On the y-axis, you have zones of inhibition in millimeters, and on the x-axis, you have the different bacteria cultures we tested it against. The takeaway from this graph is that crocodile plasma significantly outperforms human plasma. And as you can see, for S. aureus and S. epidermidis, there were no signs of inhibition for my plasma. However, for the crocodile plasma, there was clear evidence. And for E. coli, there were signs of inhibition for both. However, the crocodile was more strong at inhibiting the growth of the bacteria.
So after we compared it to human plasma, we decided to compare it to a traditional pharmaceutical antibiotic, Trasidex, which is the same one that we used in her experiment. So on the y-axis, again, we have zones of inhibition, and we have the different cultures we compared it against. So through statistical analysis, we found that the performance of crocodile plasma and Trasidex had no significant difference against the cultures E. coli and S. epidermis, and that the p-value is greater than 0.05. So statistically, they have comparable performance. We did find a significant difference against the strain S. aureus, where the p-value was greater than 0.05, but was less than 0.05. And crocodile plasma performed roughly 74.3% as well as the antibiotic. So this leads us into our discussion, and we believe this is solid evidence that crocodile plasma presents some sort of antibiotic properties. For one, crocodile plasma consistently produced zones of inhibition on every trial that we ran. It was comparable in performance to an antibiotic against 2 out of 3 of the bacterial cultures that we tested, and it significantly outperformed human plasma in all bacterial cultures tested.
So our results are consistent with prior research in this topic. These findings align with previous studies on the antibacterial properties of crocodilian blood, particularly in the American alligator and the Siamese crocodile. Further investigation should research and examine if properties can be attributed to a specific compound, and if so, the potency, structure, and mechanism of said compound or compounds should be examined further. In conclusion, this study demonstrates that American crocodile plasma has significant antibiotic properties, supporting its potential as a novel source of antimicrobial agents in the fight against antibiotic resistance.
We believe that further research is warranted to develop a greater understanding of these compounds' properties, and we want to isolate the bioactive compounds that are causing this zone of inhibition right here, which is on the crocodile plasma.
We want to acknowledge and say a special thanks to these individuals who made our project possible: Dr. Frank Joyce, Naomi Solano, Davinia Beneto, Jose Flores, aka Medaya, Stephen Lara, Jordan Barnes, and last but not least, our homestead parents, Santos and Paulo Campo Vegas.
Any questions? Grace?
It appears that crocodiles are caught really small. Are there ethical considerations about taking a milliliter of blood from a baby crocodile?
Yes, we consulted with Davinia Beneto before performing this thing, and she gave us all the parameters of how we should perform this experiment. She actually advised us to go for small crocodiles and not to go for larger crocodiles, because the larger ones can get stressed when you handle them, and they can actually kill themselves from anaphylactic shock. So you go for small ones for their own safety, and additionally you go for it for your own safety, because no one wants to jump on the back of a 3-meter crocodile. Will?
How many crocodiles did you sample?
So we sampled 4 crocodiles over the course of the study.
And did you get blood from every single crocodile?
Yep. Claire?
If all of your crocodiles were smaller / younger, do you think there could be a difference in the effective
So we studied, the biggest one we caught was the guy that the guy is holding right here. It was the one I caught in the video. It was 118 centimeters. So that's probably going to be a year and a half older than the one that we showed in the blood extraction video. And across all studies we compared it, there was no comparable difference. Maybe if you get into an older crocodile, they might have a more developed immune system and might have like a more developed response to bacteria. But from what we can tell, it was kind of uniform across the different crocodiles. Yeah.
What led to the choice either by yourself or by previous research for using plasma rather than whole blood?
We didn't use whole blood and decided to use plasma because there was previous studies that were done on the Siamese crocodile in Thailand, and they used plasma, and we wanted to keep it consistent with what they did so we could compare our results to theirs, because there's really only 2 other studies in existence that have done it on like other crocodilians, and that's the American alligator and Siamese crocodile. So the most closely related was Siamese crocodile, and we like to compare results to that.
Another study has done it on the serum of crocodiles. However, due to our limited resources and supplies, it was more feasible to get the plasma.
Thank you. Thank you. We're going to take a 10-minute break. For people who are in the next session, please make sure that your presentation is on UN's computer. Okay, so don't disappear. There are refreshments, and you know where the bathrooms are.
So in this second section, we will talk about biology of sea creatures, and we have Claire Bustler from University of Santa Barbara, and his subject is active strategies of covering behaviors of sea urchins. Hi, my name is Claire Bustler, and I'm going to talk to you today about the adaptive strategies in covering behavior in Toxodonustis roseus across variable surge conditions. So covering behavior is the tendency of urchins to grab loose objects on the ocean floor and use their tube feet to secure them on their body. And there's been multiple identified functions as for why they do this, including protection from UV radiation, camouflage, and predator avoidance. And they'll use a variety of items that they can find available, including algae, rocks, shells, and coral. And my study organism, Toxodonustis roseus, is one of the urchins that exhibits this behavior, as seen in these pictures here. And here's an example video of them exhibiting the covering behavior. And in addition to the previous stated factors, surge protection has also been identified as one of the reasons that these urchins cover themselves, since the added weight and stability from the materials can help stabilize them to the substrate, protecting them from surge. And in this case, surge is defined as the upward and forward movement in the water caused by waves and the tide. And while it is known that it is a mechanism to protect against surge, the mechanisms and material preference that enable them to adapt to variable surge conditions is unclear. And so this led me to my central question: what are the adaptive strategies of the covering behavior in Toxodonustis roseus across variable surge conditions? And I predicted that in stronger surge areas, urchins would preference heavier items, and larger and heavier urchins would be able to carry more and heavier items as well. So I visited 5 study sites in the Gulf of Santa Elena, and they're all listed right here and on the map right there. And the surge at each site was determined by comparing the sites to one another and with guidance from local dive experts in Quajinikio. And this was done on a qualitative scale from low to high, and which was like an overall measurement of surge at each site. And then each day I gave the sites a quantitative scale from 1 to 5, with 5 being the strongest surge. And for my sampling procedure, I snorkeled at each of the locations for a few hours every day, and when I identified an urchin, I would mark it with a buoy and take a picture of it, and then I would take the urchin back to the boat in a container and weigh the urchin with its covering material on it, and then I would remove all the covering material, put it in a bag, and weigh the mass of the urchin itself and the diameter. And then when I got back to land at Costa Verde, I would weigh the collective mass of all the items that the urchins were carrying and the individual mass of each item. And then here for my results, this is a box plot comparing the ratio of the collective item mass to the urchin mass as a ratio to the quantitative surge level. And the red dots on here represent outliers, and as the surge level increases from 1 to 5 of intensity, there is a notable decrease in the median value of the collective item mass to the urchin mass ratio, which suggests that the urchins in area of high surge levels actually tend to carry less relative mass compared to their own body weight. And then this is another box plot comparing the combined mass to the location grouped by surge. And you can see that medium surges, which is Isla Muñecos, northeast in this case, shows higher combined masses compared to the high and low surge locations. And interestingly, high surge locations are, again, lower. And more specifically, Bajo Rojo, which was an area of really high surge, had the lowest median combined mass, although it had high surge and was significantly different compared to all the other sites. And then this is a scatter plot comparing the number of items to the urchin mass grouped by surge level, and you can see an increasing trend for all of the surge levels. As mass increases, the number of items also increase on the urchins. And then this is another one comparing the collective item mass to the urchin mass grouped by surge level. And for low and medium surge, you can again see this positive relationship that heavier urchins have more weight of the items they're holding. But this relationship breaks down in areas of high surge where urchin mass no longer becomes a predictor of collective item mass.
And then this is a histogram representing the count of items as a factor of the item mass grouped by the locations. And this graph on the right is just a zoomed in version of this graph on the left, only from 50 to 250 grams of item mass, so that you can see a more zoomed in version of the actual count of items. And looking at this, you can see that there's a higher presence of lighter items at all locations, at all locations, and very few super heavy items found on the urchins. And so, contrary to my initial prediction, the study did not find clear correlation between surge strength and preference for heavier items. Instead, in high surge conditions, urchins tended to carry fewer and lighter materials, and this could be due to an energetic trade-off, as urchins in high surge environments may have increased need for anchorage. And so instead of prioritizing trying to find materials to weigh themselves down, they would secure themselves to bigger boulders and stable substrates. And by doing so, they limit the availability of surface area on their bodies and for them to actually be able to pick up items. And referring back to earlier where Bajo Rojo had a really low combined mass even though it had a high surge, Bajo Rojo was an environment that exhibited extremely stable substrate and had a lot of large boulders, and all the urchins that I found there were jammed underneath or between the large boulders and exhibited really low amount of items on top of them. And then in medium surge, it was the most unique environment and surprising result in my opinion, because it had the highest collective item mass and the greatest number of items used for covering, which could be because surge is not as extreme as in the high surge conditions, which allow the urchins to carry more items without expending excessive energy or risking dislodgement, but simultaneously their moderate surge may be enough to create stress and a threat to encourage covering behavior as a productive strategy. Back to my prediction about urchin size, it was seen that as the urchin size increased, the amount of items and the weight of materials that they carried also increased, but this broke down again in high surge conditions, which could be due to the prioritizing of stability rather than actually looking to find materials to cover themselves. And for item preference, there was a higher proportion of lighter items than heavier, which could be due to an energy requirement of picking up items, and it could be easier to pick up smaller items rather than heavier items. And it also could be due to availability of items found. Something important to note is that the heavier items that were collected were all picked up by the larger urchins that weighed more and had a larger diameter, which suggests that there could be a threshold to the mass of an item that an urchin can or is willing to pick up. And some limitations of my study, the biggest one that could be addressed in the future is having a more robust surge measurement tool, because I did all of mine based on comparison, but having a tool that could give you a real number and comparing between that may give you more consistent and more consistent results. And then future studies could investigate the energetic trade-off associated with selecting, carrying, and maintaining covering material, which could be conducted both in the wild by tracking urchins over a long period of time, or in a controlled lab condition where you could gain more precise insights into the impact of surge and observing covering behavior. And this would help determine if item preference is based off specific characteristics or by chance in surge. And so, in conclusion, this study suggests that the adaptive strategies of Toxinustis roseus in response to surge conditions are highly variable, and that in high surge environments, urchins appear to prioritize stability and energy efficiency, selecting for lighter or more readily available materials rather than physically anchoring them onto substrate. And this highlights how urchins adapt their covering behavior based on surge conditions, individual characteristics, and even factors beyond this, providing insight into how Toxinustis roseus may respond to environmental changes and the resilience of benthic species in fluctuating ocean conditions. And for my acknowledgements, I'd like to thank Frank and Emmy for being my primary and secondary advisors and giving me a lot of great feedback on my work. For Leo for helping me on the boat, and this project probably wouldn't have been possible without his help. To the Lara family and Diving Center Kwahini Keel for taking us out on their boat every day. The Castro Campos family, who was my homestay family, for providing me a nice environment to come home to. The Monteverde Institute for hosting us right now, and then all of my friends and family here and back home.
so when I figured out what the sites I wanted to use I spoke with the dive experts who work there, and they gave me like relative on a qualitative scale, low to high, what the surge levels were in comparison to the sites. And then each day that I went, I gave it a qualitative or a quantitative scale from 1 to 5, so that I would have 2 ways of comparing the surges. Will.
Did you ever find it difficult snorkeling in these high surge areas?
Yeah, I actually thought I was going to drown the first 2 days because I was so scared, and it wasn't fun, but it's okay. Oh yeah. Or blonde.
Okay. Did the urchins have a favorite item? Like, did you find some more than others?
Yeah, the majority of the urchins, when I grouped them by site, there was another graph I did that I didn't include. Pretty much all of them had a majority of rocks as their covering material, except, again, in Bajo Rojo, which had a majority of shells. And I think this could be due to the bottom composition seen at all of them, because both the Isla Muñeca sites, Playa Hachal and Isla David, they all had like rocky and sandy substrate with a lot of loose items, but Bajo Rojo was primarily really stable, solid, like rock substrate without a lot of loose materials and giant boulders. So that might be why there were less rocks than compared to the other sites.
Other question?
Yeah. Do you know if there's any like other factors that influence their collection rate, or like other reasons why they may collect things
Outside of like the surge and
yeah.
Well, a lot of the studies I looked into said, and like past studies from students I've done here have looked into surge and diel activity because they're thought to be more active at night, but at night you might not need as much protection because it's dark out, so that would be something interesting to study. But then a lot of other studies have seen that it could be for UV protection since they are found in some shallow areas. And then another interesting one was that they sometimes collect algae pieces or like leaves and stuff that are on the ocean, which has been thought to be used as a food source later. I didn't see any urchins with algae on them, so for me it was hard to tell, and that's also another study.
Cool. Well, thank you so much.
So we have Nina Hall from University Berkeley, and she's going to talk about associations between non-spine organs and small reef species.
Hi everyone, I'm Nina, and I focus on small fish living with the needle sea urchins. So basically what inspired me to do this project was when we were in Quijina Keel in October. I observed juvenile Cortez rainbow wrasse swimming through the spines of needle sea urchins. And then after further research, I figured out that these urchins are Diadema mexicanum. And then I also looked into a previous study done by a previous UAP student named Caitlin O'Brien in 2010. She found 12 species of association incidence between 30% and 70%. So my research questions were which reef species associate with needle sea urchins and in what proximity, and that was just inspired by my original observations. And then I wanted to compare my results to O'Brien's study in 2010. I know it's hard to see. Oh, you can't even see it. There's a fishy. So basically what I did was I went to 2 of each of these sites each day. And then when I got in the water, I would bring a buoy with me, and the buoy was made with a rock or a weight tied to like an empty water bottle on the other end. So then I would mark a site I wanted to study, and then I would go back to the boat and get my GoPro, or actually Frank's GoPro, and I would tie it to this contraption that Leo made for me. It was basically one of those stools that were in Casa Verde with a PVC pipe tied to the end of it, and then I would record the aggregation for 20 minutes. So I had a total of 25 videos. So to extract data from my videos, I watched 3 minutes in each video, and every time I saw a fish swim close to the urchin aggregation, I would record it as an observation. So this is which video I was looking at the arrival and the departure time, and then I would calculate the total time I saw it near the aggregation, and then I would record the species, and then I made these proximity grades to classify my observations. So these are my proximity grades. The first one is around, so that's when the fish is within 10 centimeters of the spine. There's a little fish right there. And then near is when the fish is like really touching the spines but not in between them. And then I is when they are in between the spines or like inside of the aggregation. So for my results, I made this graph that shows the average time I observed each species in any proximity to the urchins. And then, so basically, like, these ones are the ones that I decided were the most associated, and the fish on the other end are the species that I decided were the least associated. And then after some statistical analysis, I found that there is a statistically significant difference between the association levels of these fish. And then, so I found that there is the statistical significance difference was between the cardinal fish and Cortez rainbow wrasse, Acapulco gregory, and shark-nosed puffers, with cardinal fish being the most associated, and those other 3 being less associated. So this graph shows it shows like the total time I observed the fish split up into the proximity grades I observed them in. So you can see, like, for example, this is a Acapulco gregory juvenile, and you can see the least amount of time I observed it around the urchin aggregation, and then the most amount of time I observed it was in the urchin aggregation. And then, so some of the species aren't on this graph because every time I observed them, they were, like, the Spencer wrasse was, every time I observed it, it was 100% around the aggregation, and then the same for the barred serrano. And then for cardinal fish, I always observed it inside the aggregation, and same with the redhead goby. So this table shows my results compared to O'Brien's results, and the lines highlighted in turquoise are the results that we found that we got the same for, and then all the other ones are things that were different between our results. Something I found really interesting was that the flag cabria and the Pacific mutton hamlet I didn't see, and O'Brien found that they were of really high association level. Possible reasons for this difference is my method, I used a GoPro, so it was like kind of further back, so I could see fish like swimming above, which was really interesting, and like I could see that space above. But GoPros, like, they don't focus very well from, like, you can't really like see inside the urchins very well. Whereas Caitlin used the quadrat method, so basically she would like go down into the water and she would look at, she could like look really closely, sorry, I just saw, and that distracted me. So these are the fish that I found of low association, medium, and then high. So a reason why fish might be swimming in between the spines is for predator protection. So urchins are known to, when a predator is close by, they're known to wave their spines around. And if a fish is nearby, it could feel the urchin moving its spines around, and it could indicate for it to go deeper into the spines and seek shelter. It's important to note that urchins are grazers, but it's also important to focus on the fact that they could be microhabitats for different fish species. This is just like my main idea of my project. It's just, it's important to focus on all aspects of species roles in their ecosystems. And these are my acknowledgements.
So time for questions.
Oh, sorry.
Did you find, or I don't know if you're looking for this, but was there a difference with like body size? Like, was there a certain body size that would go?
Yeah, bigger fish can't fit in between the spines. So a lot of my fish that, I don't know if I can go, it's kind of hard to see, but like that fish, like you're not gonna find him in between the spines, this guy, but he still does hang out, which is interesting. And then you also can't really see, but there's a little blue dot right here, and that's an Acapulco Gregory juvenile, and those ones are in between the spines. Yes?
Do you think this is mostly like a defense against predation, or is there also some benefit, like extra food or something?
Yeah, I think it could be like some benefit, maybe extra food, but I think most of the theories and like the studies that have been done say it's mostly just like to escape predation.
How much did you see crabs within the urchins, and what inspired you to like include crab among your fish?
I wanted to include that crab because Caitlin included it, and I thought it'd be interesting to compare. And then also he was like really associated. So when I was observing in like my 3-minute sections of the videos, he was there the whole time. So
Was it just one time that you saw him?
Yeah.
Sorry, you mentioned this and I am lost, but if there are even, did you notice something about the number of urchins? Like one urchin or a group of urchins making a difference or not?
No, I have the data, but I didn't analyze it.
Okay, thank you so much.
How do you learn to eject this thing? Or can I just take that? It's on this thing? This thing. Oh wait, how does this work? Oh.
The orange one and the black thing.
You have to click both of those.
Igual, o sea, posiblemente no sea, pero All right, so we have Mika. She's going to talk to us about parrotfish occurrence in relationship to the benthic substrate. She's from University of Berkeley.
Hi everyone, my name is Nikita, and today I'm going to talk about parrotfish. So a little background on parrotfish. They're bright and colorful fish that occur in tropical and subtropical regions all over the world. They're called parrotfish because of their beak-like mouths, which they use to scrape algae off of coral. So they're known to be corallivores and algae grazers, but research shows that they may not be eating coral on purpose. It's more to get the algae off of it, and maybe by accident or necessity they also eat the coral. And because of this, they could be an indicator of a healthy reef because of their mutualism with the algae. So basically they eat, they're shown to eat algae off of coral and around coral. And algae is coral's competitor for sunlight and space. And in return, coral will offer a safe place to have shelter. Parrotfish also display site fidelity, which essentially means that they don't tend to move from a site. They stay in one area for most of their lifespan. And there's evidence that the higher the structural complexity of coral, the higher the rate of fidelity. So just the more, the bigger that the coral reef is and the more intricate it is, so it offers it better hiding spots, and so they want to stay longer. And a little bit about coral reefs, they host a fourth of marine life, even though they're only less than 1% of the ocean floor. And because of this, they are essential to understand for biodiversity and conservation. So this leads me to my research question of what types of substrate do parrotfish prefer? And my prediction is that they prefer algae and coral. So I was in Quijiniquil in northwestern Costa Rica, and these were the 6 sites that I went to, and we went during early morning slash early afternoon, or sorry, late morning slash early afternoon. And here are my methods. So for each site, I would line a transect of about 30 meters, and I took a picture every 5 meters, so 6 photos per site. And I tried to take the picture about 1 meter above the ocean floor. I chose a meter because of visibility conditions, so this allowed me to be far enough away that I could capture a larger area, but still close enough that I could see. So after I took these pictures, I then would survey the transect, so I would swim up and down the transect looking for parrotfish, noting what direction they were swimming in and what substrate I found them near. The reason I noted which direction they were swimming in is to make sure that I didn't count them twice. And I also took videos and pictures every time I saw a parrotfish to later identify them. I then created a grid using an app called Grid Number to kind of mimic a quadrant. And the total area of my pictures ended up being 3.3 meters by 2 meters, and each individual rectangle ended up being around 60 centimeters by 40 centimeters. And I would mark each rectangle as what the majority substrate was. And then I identified the fish using a Smithsonian Tropical Research Institute website. So here's an example of what my pictures looked like. So for this one, for example, would be like 92% coral, and then this one majority algae, that one bare rock, and that one sand. And it's also important to note that the algae that I found was on rock and not growing out of the benthic floor. And so I did this for each site, for each picture. And my results are that I found one species of parrotfish, the Scaruscoban, or the Luchin parrotfish, and I found a total of 154 of them. And so here is that number broken down into site. So as you can see, I saw the most in Isla San Jose, and I saw none at Playa Sortija. And then it kind of just more evenly distributes at the other sites. I saw 76 at Isla San Jose. And here is that benthic substrate cover per site. So as you can see, Playa Sortija, which had 0 parrotfish, has mostly sand, where Isla San Jose, that had 76, has algae and coral. And then there's different levels of algae and coral per site. And then this is where I saw the parrotfish near every time I saw an individual or a group of them. So most of the time I saw them near algae, and then coral, and then sand and rock were about even. So my results show that there's a moderate correlation between algae and parrotfish and coral and parrotfish, meaning that with the association of, with the display of algae, there's more likely to be parrotfish, same with coral, and there's a negative correlation between sand and parrotfish and bare rock and parrotfish, which means that with the presence of one, there's likely to not be the presence of the other. And here are those numbers played out, and as you can see, algae has the highest correlational coefficient. So on to my discussion. Parrotfish live near algae and coral and are found in healthy reef systems. And all this, tied to the fact that they display site fidelity, means that they could be an indicator of a healthy reef system. It was interesting to see that in sites that just had algae, there were less of them than sites that had both. So they very clearly prioritize or prefer both. And I think a potential future study would be to, well, first compare places that are marine protected and not. Isla San Jose is marine protected, so that could have accounted for my high number of parrotfish. And then to kind of do this more long term, to see coral degradation in action and see if parrotfish contribute to that at all. And then to also do this in a variety of sites to see how parrotfish differ, like if they have individual or species preferences. So yeah, thank you so much. That's my presentation.
Any questions?
Yes. Clarify, what is benthic substrate?
It's just the ocean floor.
Okay. And did you ever find like where there was algae on coral, and then what you would do in that situation?
That I didn't come across as often, but I would probably categorize that as algae, because it's the algae that's drawing them more to, like, it's more so the algae cover that's probably drawing them more to that habitat.
It's possible I just missed this, but did you visit the sites more than once, or were you only able to visit each site once?
I visited each site just once.
Any other question? Frank?
What do you think accounts for the difference in numbers observed between Isla San Jose and the other areas, like specifically?
Specifically, I think the fact that it's marine protected was the main driver. I think that they have the opportunity to grow larger and like more abundant, and then they have the resources to do so. And in the other areas, it's either closer to land or like you're able to fish there, which could attribute for that.
Is there anything about parrotfish and the way they're hunted that makes them especially vulnerable?
Yes, so they are especially vulnerable at nighttime when they have they basically make a mucus bubble. It's like just basically their spit, and they are able to make it in an hour, which is super cool. And it basically makes them a really easy target because all the fishermen have to do is just stab into it and that's it.
Thank you.
You good?
Yeah, I got it.
Well, so we asked Jonathan Seawards, junior refugee behaviors observed in Pacific North sharks in the Gulf of Santa Elena, Guanacaste, from University Santa Barbara.
Good morning, friends and biology enthusiasts. Today I want to share my research on the diurnal refuge behavior in Pacific nurse sharks that I observed in the Gulf of Santa Elena in Guajini Kilo. This is the species in the focus of my study, the Gangliomastoma unami. It's especially notable, unlike other sharks, it utilizes a strategy called vocal pumping, which allows it to oxygenate its gills without continuously swimming. That, in combination with adapted pectoral fins that allow it to navigate the substrate of the ocean floor, it exhibits sedentary diurnal behavior shown in these two images. The right one is pretty dark, sorry. But as found in previous studies, such as the work done by Sergio Madrigal-Mora and Mario Espinoza, there's been many observations of aggregation of these individuals, sometimes up to 30 individuals, especially at specific sites. These sharks are known to exhibit high rates of site fidelity, and so they often return to a specific location to rest. Another strategy they use to rest during the day is concealed habitat, which I observed at many locations throughout my study. Especially in juveniles, because they're more able to fit inside of a concealed habitat. This drove me to ask my central question, what are the patterns of diurnal refuge behavior in shallow waters within the population of geonami in the Gulf of Santa Elena? I wanted to define refuge behavior as just any strategy that an organism uses to avoid predation. There may be other reasons why these sharks rest in these patterns, but definitely it does help them avoid predation because they do have some predators. They're not fully apex predators, they're mesopredators, so there is evidence of them being eaten by larger sharks, such as tiger sharks and even juveniles with crocodiles and egrets. This was my study area. The large markers are where I found sharks. Smaller areas where I did not, the little circles, but Isla Muñecos, El Chal, Isla San Jose, and Playa Sortija are my main spots of focus. Playa Sortija is indicated in blue because that was where the large aggregation site was. And that was the only site where I observed aggregations of 5 individuals or more, which I classified as a large aggregation. My methods basically were, we would drive a snorkel boat, we would drive a small outboard boat out to these locations. I would snorkel between hours of 9 a.m. and 3 p.m., canvassing the area for likely spots where these sharks might be sleeping or resting. I chose a depth range of 1 to 8 meters to facilitate me being able to observe them with freediving. Some of the spots were upwards of 25 feet deep,
and I had trouble getting stable video of them, as you'll see in my images. And the data I wanted to collect was how many sharks were occupying a resting site, and if they were occupying it multiple days, which you'll see in my results later. I thought it was the next slide. This is, once I would find a shark in a specific resting spot, I would create a map to help me more easily return to the spot in a following trip. I visited sites multiple times, and so I wanted to know where the spots I had seen them were so I could recheck for shark presence. These are the pictures I was talking about. They were hard to get. The GoPro did not like the lighting, and I had limited breath, but as you can see, these sharks are taking refuge in coral concealed habitats. And in the right image, there is two, even though you're not able to see the other one. I promise there's two. I also observed some site sharing, which corroborates with research done by Alex Martin in 2021 during this program, where they studied the associations between fish and nurse sharks in Quahini Keel. And so that was an interesting finding. I don't have any quantitative data on that, but it was worth noting. This table pretty much embodies my results of my findings throughout my study. My formatting got messed up. But as you can see in the green highlighted cells, that was when I observed recurring occupancy, i.e. A shark inhabiting a refuge site multiple days in a row or reoccurring. The red was when I just observed a shark once, rechecked it, and there wasn't a shark there. And the gray was when I was only able to observe that site one time, so I wasn't able to check for reoccurring presence. I found a total of 38 sharks, which was actually, I was really happy about that because the first few days we were in Quahini Keel, the visibility was very poor, and I was getting skeptical whether this project was feasible or not. As you can see there, the cells with 5 and 15 in them were in Playa Sortija, where I saw the large aggregations. And those were in the sandy substrate of Playa Sortija. Most of the time that I observed sharks in concealed habitat, it was either 1 or 2 individuals. So Playa Sortija was the only location with large aggregations. To further investigate their behavior in these resting sites, I tested the shark's response to food by placing a box with cut sardines in it. A box with cut sardines in it, about a meter away from the shark resting site. This photo was from actually Cassie's data because this shark was investigating her trial, but in my findings, I found the next slide. Yes. 3 of my 5 trial sharks left. Am I out of time? Okay. 3 of the sharks left, and 2 did not. As a takeaway, I concluded that sharks use concealed habitats as resting sites. They exhibit site fidelity with conspecifics and other species, and they showed a varied response to food. I would like to acknowledge the Lara family, Leo, the other EAP scientists for helping me find my sharks, Frank, Joyce, Emmy, and my homestay family, Mata Mata Casada family. Thank you.
Any question?
Do you think that you only found like 1 or 2 sharks under in like tight spaces because of like space area relation, like aggregation were there because you only fit 2 sharks in the area?
Yeah, actually, I do believe that because when I was scuba diving on the field trip, I observed upwards of 8 sharks in a larger cave, and those features, like a larger cave, aren't exactly available at the depths I was snorkeling at, and so that definitely played a role in my data. Yeah.
Based on some
sharks being there multiple days and some not being there on different days, would you, how long do you think these sharks will come to a certain place for, like, and stay there?
Sorry, can you rephrase your question? How long do they come to a site? You mean like throughout their life? Do they like typically frequent sites?
Like how some of your data had sharks at a certain spot on multiple days, and then some, like the red ones, were not. You think from that you'd be able to estimate like how long they'll stay in a certain refuge site for?
Right. So one important thing I didn't say is that I wasn't able to like tag and identify individuals. So these are just like me checking a spot for a nurse shark inside of it. But I think where they rest has probably something to do with their nocturnal hunting strategy, of sort of when they're done hunting, finding the nearest spot. But they're known to have really good spatial awareness, so I'm sure they're like aware of favorable resting sites and probably choose those instinctively. Yeah.
We have Cassandra Ryan from University of Santa Barbara, and we fish richness and abundance in protected and unprotected marine areas in northwest Costa Rica.
Hi, I'm Cassie. I'm a little sick right now, so just forgive me for that. I'm studying fish abundance and richness in protected and unprotected areas. So human actions from overfishing to ocean acidification to pollution have all degraded marine environments in a multitude of ways. So the need to protect marine areas has become increasingly necessary. Specifically in Costa Rica, ACG, Area de Conservación Guanacaste, has 43,000 hectares of marine protected area. And in these sites, there is no mineral extraction, there's no fishing of any kind, and there's no plant or animal removal. But while this area was designed to be a fully protected no-take sector of ACG, they have very limited funding and inadequate enforcement. For example, ACG has gone through budget cuts over the last couple of years, and they also only have access to 10 functioning patrol boats. The other half of them do not function. So this lack of enforcement allows fishermen to go to the Islas Murciélagos, part of the protected area, at night and hit it hard with fishing. So I wanted to ask the question, what is the difference in fish biodiversity in marine protected areas and unprotected areas? Just to see how effective these enforcement laws are and if it is actually directly correlated with that. So in order to do that, I set up a weighted GoPro on the seafloor and had it filming a bait box filled with sardines. And this would attract carnivorous or omnivorous fish. And so it did act as a limitation because I could not attract herbivorous fish unless they just happened to pass by in the camera's frame. And I would film for 10 minutes, and I would tally the total amount of fish that I would see in one video at one site, and then the total amount of species I would see in one video in one site. And I went to a total of 8 unprotected sites and 3 protected sites. And here's just an image of my setup. Here are the study areas. Down here are the protected areas we went to in the Islas Murciélagos. They're about 24 kilometers from Guajiniquil. I saw a total of 2,610 individual fish and 52 species over 110 minutes of recorded video. And then here are my results for the fish richness, which is the amount of species that I would see. So in this bar graph here, we have site on the x-axis and then number of species on the y-axis. And these dark blue bars are the protected areas. I did a one-way ANOVA and found that P is 0.0044, so there is a significant difference in fish richness in marine protected areas and non-marine protected areas. And then you can see that over here that the average number of species I saw in a protected area was 20.7, and the average I saw in an unprotected area was 11.1 species. And then as for fish abundance, once again, site is going to be on the x-axis, and then on the y-axis is the total individuals that I saw in a site. These dark blue, once again, are protected areas. And I did a one-way ANOVA and found that the p-value was less than 0.05, and so that means there is a significant difference between the two. And then you can see that once again in the average. So for a protected area, on average, I saw 436.7 fish, and in an unprotected area, on average, 162.5 fish. And these are just some species of note. Actually all four of these were, I'm focusing on the protected areas. So this is a nurse shark, and as John said in his presentation, it did approach my bait, and so it's really cool to see that I saw, for like 5 minutes, the nurse shark was attacking the bait, which was pretty cool. And then also a spotted eagle ray came in the background of one of my videos at Isla San Jose. And then down here, this fish is a milkfish, and it's a very rare fish to be seen in Costa Rica, and I saw 9 of them at Isla San Jose. In the video, there was actually more than that, but I only got 9 in the video. They aren't particularly fish in Costa Rica, but in Southeast Asia, they are fish. So it's not really an issue here, but they are rare in Costa Rica. And then, back to what Nikita was saying, here's a blue-chin parrotfish. And I did see these in other sites, but there was 66 that I saw on my video in Isla San Jose, and that's definitely an indicator of ecosystem health because blue-chin parrotfish are commercially fished a lot in Costa Rica because they have like a nice fleshy body, and so it's good to see that there is 66 of them, the most that I saw out of anywhere at Isla San Jose. And then I'm comparing my study with 3 different studies. First, Gilbertson and Robertson in 2017 did a very similar study to me. They set up baited camera traps in different sites, and they found that there was no difference in fish abundance among sites. And now I've done the same thing 7 years later, and I have found that there is a difference between the sites. And I'm not saying that's 100% attributed to governmental protection. It's likely due to a variety of factors such as the increase in coral and maybe lesser fishing going on there. But it is interesting to see that now you can find a difference in the abundance. And then in 2010, Michael Hazen was looking at coral at Isla San Jose, which is part of the marine protected areas. And he noticed that the coral there mainly consisted of dead coral, which he attributed to a large coral die-off the year before in 2009. And then when we went to the Isla Murciélagos, I saw that the coral there was mainly living. And so over the past 14 years, the coral has regenerated, and I think that's really cool. And then one final thing, in 2022, Haley Fox did a study, very similar, surveying fish diversity just around northwest Costa Rica, and she found that fish diversity correlates with coral coverage and not necessarily governmental protection alone. And so that is a big factor that there was a lot of coral in these areas, in areas like Bajo Rojo, where there was a lot of fish and a lot of different species, did have the highest coral coverage of the unprotected areas. In conclusion, there is a significant difference in both fish abundance and fish richness between protected and unprotected areas. Thank you to Frank, and Naomi as my advisors, and thank you to Miriam Collado for letting me stay at her house. Thank you to Leo, you're the best, Minor for driving the boat, and all the cats that I saw.
Questions? Connor.
How long did it take you to count 2600?
A really long time. It took me about 2 hours per 10-minute video to like watch the fish and count them and ID them.
Any other questions?
What was your favorite cat?
Probably the one on this slide. She would meow really loudly, and I named her Cement because she's gray.
And some other question there? No? Yeah?
How did you choose the location, like once you were in there?
Yeah, so for all of the unprotected sites, it was entirely at random. I would just kind of hop in the water and then swim around for a bit and I'd be like, okay, whatever, here's fine. But then once we went to the Islas Murciélagos, Frank helped me, and then he was like, oh, here's a good place to put it, let's put it here. And so, yeah.
Frank?
What would you say to a fisherman that you would speak with directly who is coming out of the marine protected area with a boatload of fish?
Do I have to talk to them?
No, you don't have to talk to them.
So am I like boarding their boat or like what are the logistics of this?
You could say, you know, hello, when's Yang, got any fish for sale, or you guys are bad. I mean, at some point you may actually encounter this, not necessarily in Costa Rica, but maybe in California.
I'm a very non-confrontational person, so I probably wouldn't say anything, but I'm thinking in my head, that's not very good.
What are you?
Casi outside the protected area, among the species that you saw or counted, do you have some that could be doing less well or more endangered?
Like endangered fish in unprotected areas?
Yes.
Yeah, at Playa Sortija, I don't remember the exact name of the fish, but there was a rare fish there that I saw a couple times, but that was like one of the only fish that I saw at Playa Sortija. There was only, I think, either 4 or 6 species there. So that's interesting. I should look more into that.
Do you know anything about how this corresponds to particular areas in other regions of the world?
I'm assuming and hoping that it's similar, that other areas also have significantly higher abundance and richness in those areas, but also protection laws can be implemented because they are not doing well, so it would be more beneficial to look at like a long-term study to see how that changes.
Next, William from University of Berkeley, and he will talk about chemical composition variability between modern and marine gastropods.
Hi, I'm Will, and this is analyzing cross-species chemical composition variability between modern and Holocene gastropod shells. Ocean acidification poses a major threat to all marine calcifying organisms. Gastropods, one of the largest groups of marine calcifying organisms, are especially vulnerable. Not only do they require substantial carbonate to form their shells, but they're also extremely hunted for their shells and their meat. Gastropoda is one of the largest classes and most ancient classes of marine multicellular organisms, approximately 500 million years old, and they're also critical for ecosystems as they provide nutrition, for other organisms, nutrient cycling, and habitat for creatures like crabs and other mollusks like octopus. Gastropod shells are made up of calcium carbonate. The two main forms of calcium carbonate in these shells are argonite and calcite. Gastropod shells are very complex with different forms of calcium carbonate, and it's separated by layers. Argonite is most likely in the inside part of the shell, and it has a variety of special properties, including the iridescence shown in abalone shell, as well as special fracture resistance. Carbonic acid, a product of ocean acidification, deteriorates calcium carbonate in gastropod shells. However, there is a variety of structures in gastropod shells, and some have properties like resistance to dissolution and resistance to pH. As shown right here, here are different forms of calcite and argonite in one gastropod species in the Pacific. My focus for my project was 4 species of marine gastropods in northern Guanacaste off of the coast. On the top right A is Neely-Ringas, top left is Strombus gelatus, also known as the Eastern Pacific giant conch. The bottom right is Coriophila costata, and the bottom left is Homolocantha multicrispata. My question that I wanted to answer was, what are the chemical and structural variations of marine gastropods of different species and ages? For my methods, I did a variety of things. To collect my samples, I snorkeled at 7 different sites and went to one island to collect mid-Holocene shells to analyze the difference between Holocene and modern shell composition. To collect my Holocene shells, I went to Isla San Jose, which is the location of a midden dating back to the mid-Holocene, and I found shells adjacent to this time period to collect samples. I also collected live samples by taking individuals from the seafloor and using a handsaw to take a 3 centimeter by 3 centimeter sample of its shell. Then, back at the lab, I organized the shells and made seawater of 3 different pHs to test the effect of acidity on pH structure, on shell structure. My control pH was 8.26. My first experimental pH was 7, and my second experimental pH was 5. I lowered the pHs by adding acetic acid in various concentrations, and then recording it with a pH meter. Then I broke the pieces of the shells into 3, and I put each piece in a shell of the various pHs, and then I took data by weighing each shell piece before and taking pH data throughout the experiment. For my results, in total, I sampled 36 individuals, 19 H. multicrispata, 6 Coriophila costata, 5 Strombus, and 6 Neely-Ringas. One of the main hurdles of my project was being able to accurately calculate the amount of calcium carbonate that dissolved from the shells into the surrounding water. And the limitation that I struggled with was the scale, as it didn't have enough accuracy to calculate the very minuscule differences that these shells would face to find a significance. So what I did was, analyzing the pH data, I calculated the hydronium concentration of the final and first measurements. Then I multiplied that by the liters that I had it in, and then I used stoichiometry to account for how much calcium carbonate would be needed to neutralize the hydronium to get from the first to the final pH. And then I converted the moles of calcium carbonate to grams, and then I divided that by the original mass to get a percent of the shell that was lost through the experiment. My results, this first graph shows H. multicrispata, my most abundant shell, in its response to pH 5 and 7. And this is basically analyzing the difference of this species' modern shells versus Holocene shells. And what I found was that there was a large difference between modern and Holocene shells, both at pH 5 and pH 7. I also found, on the y-axis is the percent mass lost through my experiment, and then I found that the modern shells at pH 5 had on average 0.000669% loss, and the Holocene shell had 0.000112% loss. Using a ANOVA, I calculated that this was a significant difference. My next graph is a cross-species analysis between all of the species, and I found that the largest difference was between H. multicrispata and Crocodile costata, and that I used a t-test to determine that this was also a significant difference between species. Basically, modern samples had a larger percent of their mass dissolved into the surroundings than their mid-Holocene counterparts. And then using the cross-species analysis, I concluded that both age and species type played a statistically significant role in the percent difference in mass of the shell. Crocodile costata had a statistically significant percent of the mass lost at pH 5 than Homolocantha, and it could serve as an indication of gastropod species most vulnerable to ocean acidification in the area. An environmental factor that could have influenced my findings regarding the Holocene versus modern shells is carbon flux ion availability. Since the Industrial Revolution, the ocean has increased by 30% in acidity, and so this could have reduced the amount of carbonate available for gastropods to create their shells, thus reducing the chemical density of their shells. Further, another explanation could be the removal of biogenic calcium from the local biogeochemical systems through fishing and removing vertebrate skeletons from the substrate, thus reducing the amount of available calcium that these organisms have to create their shells. This is basically juxtaposing how ancient the shells are versus how much they have lost in mass. My conclusion is that due to the threat of ocean acidification to gastropods, it's critical to identify the variations in shell chemistry, and I was able to identify that gastropod shell composition is different depending on what time period individuals are from and what species they are. And I'd like to acknowledge Frank Joyce, I'd like to acknowledge Leo for helping me for my project, Naomi, Emmy, Federico, my homestay mom Miriam, and I want to thank the people that helped me with my project, Cassie, Grace, Claire, and John for your help finding my shells. That's all.
Any questions?
Yes.
It's like the outlook, on when are we reaching the pH, worsening pH levels to the point where you see significant loss in the shells?
So the ICPP reported that we will reduce our pH, the ocean pH, we would reduce to 7.8 by the end of the century, which is a drastic difference in pH. Most of my graphs were regarding pH 5, but I also did tests on pH 7, and those also showed statistically significant differences for shells.
Any other question? Frank?
Which was the rationale in the study to compare with mid-end shells?
The rationale was basically trying to see if there are any observed changes because the ocean has acidified by 30%, so it could basically compare a trajectory of marine gastropod decline and not being able to form their shells properly.
How do you compensate or explain the weathering that's happened on the shells of the Holocene compared to more modern shells?
Yes, this slide, answers it a little bit. Basically, this one explanation for the weathering is that because my Holocene samples have been around for 7,000 years, they could have already had a significant loss in calcium through leaching and degradation. And so during the experiment, they could have proportionally lost less mass because there was less calcium to begin with. And then my modern shells would have had more calcium to begin with because they're more recent and they had more available calcium through maintenance. Any other questions? Yes.
Can you tell us a little bit about your saw?
I had a variety of saws. One of them rusted and broke in my hand, and then I had to use saw bits for a little bit. Yeah, on the boat. Rocking.
Okay, all right, so, muchas gracias.
Grupo de APS la justificación era como tratar de sacarlo de la playa, no sacarlo del medio.
Vea, Fran, sería como ponerlo así.
No vale nada. Pero creo que uno es mejor que otra cosa. ¿Qué pasa si pone aquí?
O sea, lo que pasa es que hay que mover el cuadro para que creo que hay que ponerlo así. Ajá, o yo ponerlo así.
¿Cuál prefiere?
Prefiero que muevas el pollo. Porque es que si lo pongo, es que igual tendría que ponerlo así para que se vea. Si la pongo así de frente.
Pero ¿qué tal si nadie hace a ver si se puede ver ahí? ¿Qué tal así?
Sí, esa es la otra opción. Lo que pasa es que a mi dice la velocidad se queda blanca, pero es que usualmente, bueno, con ella tengo calores que son demasiados, pero usualmente lo que hacen es que me pasan la Ok, entonces voy a... Entonces, Francine, tal vez cambiamos de lugar. Sí. Ya tenía mucho frío en la mañana, pero vertebrates, very understudied vertebrates, and we're gonna come back here to Monteverde and learn. We're gonna keep learning about gastropods, but this time it's terrestrial gastropods by UN1 from UC Berkeley. Thank you.
Hi everyone, my topic is leaf-associated arboreal mollusks, microhabitat preference, and species survey in Monteverde region. So mollusks derive from nemertine tuilarian basal clades, and they're the second most diverse invertebrate group. And all terrestrial mollusks are gastropods, but they're actually a highly polyphyletic group, because as you can see here, these are all the major subclass, and we can find terrestrial groups in Cynogastropoda, Neuratimorpha, and Panpulmonata. And some of the terrestrial groups, they decide to live on a specialized lifestyle. These include arboreal, and then further, they can be leaf-associated. Usually for leaf-associated gastropods, they're small size, and they highly rely on their microhabitat, which are also called phylosphere. So the final goal of this study is to assemble microhabitat preference among species. But to do that, I need to first survey and filter all the leaf-associated species. So my hypothesis is to find difference between major clades, Neuratimorpha, Stygomatophora, Veronicelidae, and also find species level distinction and difference within the clade Stygomatophora. This order is also the most diverse terrestrial group. Okay, so for my methods, I visit 3 locations: Crandall Reserve, Estación Biología, and Santa Elena Town. In the field, I will measure these microhabitat parameters, temperature, and height from the ground, because I'm looking at arboreal species, and also epiphyllous or epiphytic coverage, the percentage, and also the relative humidity. For individual, I also search a lot of literatures, and also I look at GBIF, the website, to do species identification. Also, I will include the activity status, like are they sleep, estivation phase, or are they feeding, or are they semi-active, meaning they retract their eye stalk. Also, I will record the host types. So to get approximate percentage of epiphyllous coverage, I will put the leaf onto a standardized grid. So this is how I get the estimate. Also, I will record whenever I encounter a mollusk species, I will record the temperature and humidity with the hygrometer. And also, one important thing is that how I identify plant species is to Hola, Hilario. I'll interweave my discussion presentation into my results. It's very cool. First, this is how I filter leaf-associated species. As you can see here, for this species, Antibremias sulphurus, it's 100% of the substrata found is on the trunk. So this I will rate it as arboreal but not leaf-associated. However, Drymaeus genus and Helicina monteverdens, Ticoconus costaricans are rated as leaf-associated, whereas the Veronicelidae and Euglandina are also not leaf-associated. Okay, so this DCA detrended correspondence analyzed around by our language, and this is a multivariate gradient analyzed to show the clustered environmental condition, like plotting them in different locations. And one cluster means they're the sites are ecologically similar, and the ordination represents the environmental gradient combination. The longer the arrow means there's stronger gradient that influence these sites. Yeah. And each site is one plant morpho species and grouped by calculated parameters average. And one interesting thing I found out recently is that DCA results actually can show beta diversity by looking at the projection of these points to each axis. For example, on the x-axis, there's shorter or narrow range. That means there's less diversity in their preference of epiphyllous coverage. However, when we look at the y-axis, there's wider range, means there's more pronounced species turnover. There's more diversity, not in a macro habitat, but in micro habitat aspect. We can see there's relative humidity, height from the ground, temperature. They display this wide range of preference. And statistic tasks I involved in these studies are ANOVA and post-hoc tasks. For these tasks, I only include 5 most abundant species, so the DF here is all 4, and results are all significant. So that means the species have their own preferred microhabitats. In contrast to the conventional view that all snails and slugs, they want humid environment, and yeah. And also I do the pairwise comparison task, shows that Helicina monteviridens, this species, prefer really low temperatures compared to others, and also the Ticoconus Costa Ricans have low height preference and high humidity preference. Okay, so this is kind of discussion part, but I'll make them in all one section. And this is one of my most favorite gastropod I found in Costa Rica. It's called Helicina monteviridensis, and it is only found on Rubiaceae plant. It has a really strange population distribution. You can find them in large quantity in forest area, but you cannot find them in Santa Elena town. And this species also exhibit mimicry. If you look further away, there's a red spot on the tip of the shell. It resembles insect gulls. And also, can you find where's the snail here? So it's a little bit out of focus. Oh, wait, okay, so. And this species is in the family Euconialidae, and the genus is Haberconus. And it can only be found on Rubiaceae and Peperaceae. As you can see, there's a ruler. Most of them have this 2 meter length of their shell. So it's pretty hard to find them. And they can only be found on the understory of the leaf. And also this alien-looking gastropod is actually not a snail. It's a semi-slug. Because you can see here, there's a mantle covering this shell so that the body or the food cannot fully retract to their shell. And this makes them really vulnerable to desiccation. So that's why this species prefer high humidity. And similar to the previous one, it can only be found on the understory of the leaf, meaning the downside of the leaf, not the surface. And he also liked a Rubiaceae plant. And I did an auxiliary study on species composition, and these species are characterized by if they are endemic, native, or invasive. Santa Elena have a lot of invasive species. And this is a Venn diagram showing the distribution and the overlap. These are all the invasive species. And we can see in the DCA plot that antifungal growth is more important than abiotic environmental variables. I forgot to mention that by default, the DCA program shows that the x-axis is the most important environmental gradient, which is the epiphytic growth. And I found overall not crowded microhabitat application because each size cluster support no more than 2 species. And indeed, I found species level preference, and also I explained previously about that appropriate species, Halicina multivariances, and there's a lot of invasive species right now. This is a species count with a taxonomic group, yeah. And acknowledgement. Thank you, Amy, for offering meaningful advice in my article, and thank you, Federico, for arranging hygrometer and ruler, the measurement equipment, and thank you, Francini, for accompanying me to do nighttime research, and thank you, Miguel and Connor, to carry the ladder for me over long distance, and thank you, Roxanne, for translating Spanish literature to me. Thank you.
Thank you. There's time for like about one question.
What does that say?
It means nature is full of charm and we should always be humble.
Okay, second question.
How do you think that plant specificity occurs?
So I read some articles. It shows that some slug species, they have this mechanism to detox the volatile chemicals that a plant release to protect themselves, so they're able to live together or directly feed on the plant. But there's no record for the Halicina multivariances and Ticonus Costa Ricans, but I think that will be a really good future research.
Thank you. Okay, now we start with the role of arthropods in particular, also very understudied. So Zach Beckholm is going to teach us about these very common but very underlooked and understudied hanging flies in spider webs. Thank you, Zach.
So I studied the temporal abundance and spider silk preference of these very obscure thread-hanging flies here in Montevideo. This is the fly right here. So first of all, Diptera, as many of you know, is a very diverse order, and it includes all of the flies that we know. A very small portion of these flies have been documented to intentionally hang from spider silk. So rather than being caught in a spider web, they will actually use it to rest on. Many of the thread-hanging flies are in the family Cecidomyidae, which is commonly referred to as gall midges. These are very small, long-bodied flies. In terms of the reasons for this behavior, it is relatively unknown. Some studies have documented, first of all, resting behavior. So they will rest on these webs, and it can be used to protect them from predators who hunt on foliage. So by being separated from plants on these very thin, delicate threads, these larger bodied predators cannot get to them. Another proposed explanation is kleptoparasitism, which is basically parasitism except klepto, which means stealing, so they will steal prey from spiders out of their webs. This is the behavior in question. So here's the fly right here. Notice how the two front limbs are used to hold on to this single spider thread, and the other four limbs are outstretched in a planar fashion. It's important to mention before we go any further that there are various spider silk types. So a single spider can produce up to 7 different types of silk, all with differing functions. The two that I'm going to focus on are flagelliform and ampullate silk. So flagelliform silk is used in the sticky capture threads. Within the web, you think of the traditional fly catching web, that's the sticky flagelliform silk. There's also the non-sticky ampullate silk, and this is used for web support lines and spider safety lines. So spider safety lines are employed by spiders when they're just wandering around. They will leave a trail behind them, and in case they get threatened and have to ditch a leaf or something, they will always have a safety line to return to. So the two questions I focused on to kind of catalog the natural history of these flies is firstly, at what times of day are hanging flies most abundant on spider threads? And number two, on what type of thread do these flies prefer to hang? To answer these questions, I set up a transect right up here in the Crandall Reserve. It was 29 meters on the right side of this trail right here. And I sampled this area 3 times a day at 2:15 p.m., 5:15 p.m., which is exactly when the sun set, and 7:15 p.m. to simulate afternoon, sunset, and night periods. In each of these sampling periods, I counted the hanging flies that I encountered in a 35-minute window, and I, again, sampled each of these 3 times for a total of 9 surveys of the transect. I also noted the thread type on which each fly was hanging. I will go into that in depth here. The first type is called a dragline thread, which is kind of what I was referring to with the safety lines. So as you can see here, there's a single thread and this guy hanging from it. So safety line, again, used by spiders to prevent them falling. And I would define this for my study as a single thread, which is not associated with a web, but is simply between 2 environmental factors, usually leaves. The second categorical output I have is support thread. And this is a thread that connects a web to its surroundings. So as you can see here, the organized web spiral, and there's some foliage over here. And this is a support thread between the foliage holding the web up. And this is where I found this guy. The third categorical output I have is a web capture thread. So this is a thread that is within either a organized architecture web structure or a high density of crossing threads. This one's kind of hard to see, but this guy is right in the middle of all these crossing threads. I also additionally tried to catalog some of the diversity here of hanging flies in Monteverde, and so I collected them after sampling their abundance and their location. I collected them in Eppendorf tubes with alcohol, so they would die painlessly and quickly. And then I distinguished them under a microscope based on factors such as genital morphology. You can see some different looking fly genitals here, or antenna shape. So there's different antenna shapes here. The results for my time of day aspects of my study was that there was no significant change in abundance at different time periods, although the means indicated greater results at night and dusk. The sample size and variance did not allow for a significant p-value after running an ANOVA test. For the results of my thread type aspect of my survey, however, hanging flies were found to be significantly more abundant on the dragline threads than they were in the capture areas. So this is evidenced by the 54 midges found in draglines, 28 found on support, and 9 found in the capture area. And in terms of the diversity, I found a total of 20 morphotypes. As expected, most of these were gall midges, although there was one unexpected Caroplatidae, which is this guy with the long abdomen. So for the time of day, although I didn't find any significant difference, I think this is caused by a couple different factors, one of which is that my study did include many different species, genera, and as I mentioned, even families. And it's been documented that within Cecidomyidae, there's quite a variance in activity levels throughout the day between species. So it's very possible that I cataloged different species that have different day-night activity levels, and that kind of formed a more homogeneous distribution. The reason this is important, and I really want to see more research be done on this, is because time-based abundance can indicate the activity levels for the natural history of these flies. So if we assume that, that hanging on these threads is primarily a resting behavior, then finding more on threads would indicate a less active time for the flies. In terms of thread type, there was a increased abundance as you get further away from a web. So drag lines are the least associated with a web, followed secondly by support, and then finally, obviously, being in a web, very close to spiders. So I think maybe this is to avoid spiders, or it could also be to avoid the sticky capture silk. As I mentioned, flagelliform is more common in these capture areas. So somehow these flies are either avoiding spiders or they are avoiding the sticky silk itself, which is very interesting. My data also suggests that thread hanging in these cases is used primarily for resting than for kleptoparasitizing. And this is because kleptoparasitic cecidomyids are known to associate very, very closely with their hosts. In some cases, they will even land on the spiders to avoid expending energy. For a future study, I would like to track which species of these flies prefer which type of thread, and if they even have a host specificity with spiders. I noticed in all of the instances in which the flies were found in the capture area, this occurred within a certain family of spider web. They were all within the Fulcidae family of spiders, so there may be some sort of specificity with Fulcidae spiders. I would like to acknowledge Amy Triana for all her help, Natalie Sanchez as my secondary advisor, Nina for being a great reviewer, and my host mom Ingrid, as well as the Institute here and the Cranbrook Reserve for letting me use their translate there. Thank you.
All right, there's time for questions.
Yes.
How do they avoid getting eaten by the spider? Or like, does the spider benefit in any way from having them around so it doesn't want to eat them?
So there have been studies on that, and no, it seems that it is a parasitic and not a mutualism relationship. They avoid, so that's part of why I think they will associate less with the webs and more on the kind of abandoned, used drag lines, is because they do not want to be near a potential predator. So I did see in a couple cases what looked like these flies being caught and eaten by spiders.
Next study, if you compare a bilateral area with different types of the silk or the line, do you think they are located in what is more common to find, or do you think they select where they are going, or they find a certainly common line type rather than select?
You mean like the type of silk? Yes, I believe there is some sort of mechanism for picking the non-sticky silk, especially because they could, in some cases, they can do trial and error where they land, but if it's sticky, then they're kind of in trouble. If they land on a sticky web, they're kind of stuck. So I believe there is some sort of adaptation, but that's not been documented yet.
Did you see them when they land?
Yeah, I saw some leave when I came and then land back again. They did seem to repeat the same landing site. Need more research for that.
Just, I'm curious to hear your thoughts on this. So sometimes they do get snagged in spider webs, presumably.
Yes.
And so, how do you suppose they are detecting spider webs that they wish to rest on? versus they go cruising along and get snagged in a web? I mean, are they doing it by vision? Do they have echolocation? What are they doing to detect these things?
I would love to answer that. They are severely understudied, so that's not really known from what I can tell. They do have, I can tell you, they do have very large eyes, and they seem to have really adaptive vision. So I would think it's vision.
Any other questions? No? Thank you, Zach. Our next presenter actually relates from UCLA. She's going to teach us about the actual spiders where the hanging flies were perched on, which are Gentilla radiata, very abundant around here in Monteverde. Thank you, Annie.
Hi, I'm Annalise Phillips, and I will be presenting on the impact of urbanization on the abundance, size, and substrate use of Tangella radiata webs in Monteverde, which is a species of spider. To begin, I'll just give some context on urban ecology, which is a branch of ecology that focuses on species, ecosystems, and ecological processes and how they are impacted by human development. Spiders are pretty understudied within this region, and there are also, within the studies that have been conducted, very mixed results, as mostly show an overall decrease in biodiversity, but some species have been shown to thrive in urbanized environments where some cannot survive. And many of you already know a lot of context about Monteverde, but just to go over it briefly, there is development here since the 1950s. It's famous for its cloud forests, its protected reserves, but there is development such as in areas like Santa Elena, where there are lots of ecotourism infrastructure and concrete buildings, but it is predominantly rural. After spending a lot of time here, one species that stood out to me was Tangella radiata, which is a species within the Tangelidae family. They are endemic to Costa Rica and build permanent webs that serve primarily for hunting and protection. They are also nocturnal hunters and primarily put prey on smaller insects by using a cribbolate silk that, rather than a glue-like mechanism that you might think of typically for silk, it is sort of like Velcro that entangles the prey. And this is just a little more overview of their web architecture. On the area labeled A, this is the threads of the upper tangle, which primarily serve for structural support. B is the protective tunnel, where the spider spends most of its time for protection and to stay out of the sight of prey. And then C is the horizontal sheet that makes up a majority of the web where the prey is caught and where this cribbolate silk that I mentioned is. And after observing them, I was curious to note that I was able to find them in all different areas of Monteverde, including the deep forests and also in more urbanized areas. So this leads to my study and my central question of how does development in Monteverde influence the abundance and size of Tangella radiata webs, and what are its their effects on their substrate availability. So for methods, I conducted 30 transects across 3 levels of development here, each measuring 25 meters. During these transects, I measured the web abundance, and for each web, I measured the length and width to calculate an area, and also the substrate type, which is the material that the web is built on. For later analysis, I used a chi-squared test for abundance, an ANOVA test, and a post-hoc Tukey's HSD test for size. I categorized 3 different levels of urbanization, the first being no development, which was the dense forest near the Estación Biológica of Monteverde. Moderate development, which was the neighborhoods surrounding Bajo del Tigre, which featured like dirt roads and buildings, but with lots of space in between and a lot of vegetation still. And then lastly was high development, which was areas near Santa Elena, which introduced a lot more concrete structures and buildings within closer proximity to each other. And then for my results, within the no development section, I observed 144 webs. In moderate development, I observed 176 webs, and in the high development, I observed 74 webs. For the chi-squared test, the value was 41.44 with a p-value of less than 0.1 and a sample size of 394 webs. So this showed significant differences between all three values. For the web size, the average for no development was 440.63 centimeters squared. For moderate development, it was 336.49 centimeters squared. And for high development, it was 73.86 centimeters squared. And I used a ANOVA test to observe differences between these three values, and then a post hoc Tukey's test to observe that there was a difference between each three of these groups. And then for substrate use, I observed 7 different types of substrates that I found the webs on. Within the no development area, I only found them on tree roots and dirt walls, which were all natural substances. And then for moderate development, I had the highest diversity with a mix of human altered substances, such as a dirt wall with wooden fence built onto it, or natural substances such as tree roots. And then with high development, I only found them on human altered substances such as a stone wall or wooden planks used for construction. And then to move into my discussion, this table summarizes the main patterns that I observed from my results. So to begin, despite my hypothesis that I would expect to find the most webs within the no abundance area, I actually observed the most within moderate development. However, within the no urbanization area, I found the largest webs, and they were significantly less variable, which I think shows that while there were less webs, there was maybe more consistent access to resources because the webs were able to be larger. However, I didn't measure the exact size of the spiders or the prey abundance. I measured the size of the webs to use as an indicator for size of spiders and age of how old and how big they could create their web. And then for substrate diversity, I had the lowest substrate diversity here, which is what I expected, as there was only 2 types, the tree roots and the dirt walls, which may suggest that these are their favored types of substrates to build on. And then for moderate development, as I said, I found the highest number of webs. However, they were significantly smaller than the no development, but also significantly higher than the high development areas. And furthermore, there was the greatest diversity and variability in the size of the webs, which may show an uneven distribution of abundance of prey throughout these areas, as fragmentation due to disruption from humans may cause this. And overall, it just may be less favorable than the no development area, despite having more webs. And I found the highest substrate diversity here. As I said, it was on a mix of human altered and on natural substances, which I think shows how these spiders can adjust to these moderately developed areas and build on substances that they may not be used to. And then lastly, for high development, I found the smallest and fewest webs here, which was what I expected to see. However, they were still able to, I was able to find quite a bit of webs here, showing that they can survive in these areas despite them being the most developed within Monteverde. And furthermore, there was the middle amount of substrates I found, and they were only found on human altered substances, further showing their ability to adjust to their original habitats being altered. And just to conclude, sorry the font is so small, I don't know why it did that, but main takeaway is that throughout all different urbanization levels in Monteverde, I was able to find these spider species which show their flexibility and ability to adjust to different levels of urbanization, and that population can still be supported. For future research, I would want to do a more direct measurement of prey availability and also of spider size, so that I could make more conclusive points on that. And also measure patterns of other species of spiders to have a broader understanding of urbanization and how it impacts arthropods within Monteverde. And before I conclude, I would like to acknowledge a few people, first being Leticia and Javier Solis Mendez for welcoming me to their home. You guys provided great support for me. Thank you so much. Amelia Triana for being my primary advisor and providing invaluable insights. Natalie Sanchez for being my secondary advisor. And then just all the staff here with UCEAP, Naomi Solano, Federico Chinchilla, and Frank Joyce, thank you for making this program happen and teaching me so much in general about biodiversity. Thank you.
Thank you, Annie. There's enough time for questions.
Yes.
Do you think the substrate, the biggest factor in the size, decreasement, and abundance of the webs being the substrates or other human developments like large amounts and like just movement? Do you think that could be a bigger impact, or what do you think?
Well, I think it's interesting because within the most developed areas, there were trees available and more natural substrates, but I didn't find them there. I mostly found them in like areas that could only shelter much smaller webs. So that does make me wonder if this is a preference of these webs can only, if these spiders can only support much smaller webs, if that's why they're choosing these areas, or if it is something to do with what is actually available. So I think that's an interesting question, and I also wonder. Yes.
Do you think it's within the realm of possibility that the urban spiders don't, or the spiders that are with human infrastructure actually begin or have begun to prefer those habitats, and so a spider that grows up, as it were, is produced in an urban habitat does not go wandering off to find some patch of woods, but stays in the urban environment because apparently there are good places to grow webs.
Yeah, I think within like the highest level of development that I observed, I would say that they did not say that they would prefer that yet, because like I said, there were much less webs, and also they were significantly smaller, and majority of the webs I found were in like clusters. Like one transect I did had I think 22 webs, another had like 10, and then the rest had like 5 or 6. So I think I would say no, but then I do wonder with the moderately urbanized area how I found so many that in that case I would say it's potentially that they would prefer that, because like within Baja del Tigre where I did my transects, there's still so much vegetation and access to prey, I would assume, just based on how many webs could be supported. But I also have doubts with that, as I said, like the webs were much more variable in size. So many of the webs did not grow to be as big as within the natural untouched environment.
Cool, thank you.
Any other question? Thank you. Okay, our next presenter, Andrea Saldarma, she's going to teach us about moths, and particularly their wing scales and how they could function for bad deterrence here in there. Thank you very much.
Hi, my name is Gray. Studying the comparative morphological analysis of moth wing scales in relation to bat evasion strategies. This right here is a photo of scales close up. Okay, so bats attack moths. It's a predator-prey relationship, and moths have multiple ways of evading that predation, such as absorbing the sound waves through the echolocation that the bat produces, or responding in a series of clicks that kind of warns the bat that they are toxic, or they can hear the echolocation through a tympanic membrane and then move away, or like play dead, stay still, or none of the above, and they're heavily predated. So my research goal was examine how moth wing scale structures and arrangement may contribute to bat evasion strategies. There have been a few studies that have looked how it works with the thorax scales, but never with the wing, which I thought was kind of funny. But the reasoning for that was that the thorax is more robust, so that would attack like a meatier part of the moth. I would think that the moth wing would be better because it's more surface area that would interact with the sound. So why this is important? It could help us understand sound mitigation in nature, and also help us with potential applications in noise and sound management, like let's say in a music room or anything like that. So methods I used a light trap with a black light and an LED light, and collected many moths. Euthanized them via freezing, so to kind of kill them quickly, then dissected their right forewing on each of the species. Used the center of the wing, and for a few reasons, one of which is there was less damage to the center of the wing. If they run into things, it's normally the outside of their wing. And also, again, more surface area that would interact with the sound waves of the echolocation. I observed these right forewings, so the top wings, under a microscope and in many different magnifications, but for data collection, I always did the same magnification, or I multiplied to get consistent data. So the traits that I measured were scale shape, and these were determined by amounts of serrations. So, so this has like one peak and one peak. This is 2, 3, 4, 5, or more is what I was calling it. And so there were 6 groups, A through F, and then scale size was, I used a software called ImageJ, and again, same magnification for all of them, which is really important. Could trace scales through photographs that I took through a microscope, and could get an average of scale sizes on each of the species that I examined. And then overlap, I had my own measurement scheme, which I'll show you in a bit, to get numerical data on the amount of overlap, 0 being not overlap at all, with both horizontal and vertical overlap. So scales would be like this or like this, that would be 0. And 3 would be completely on top of each other. So this is a lot of words, but basically it was saying that I examined in depth 8 species and 4 families, so that's 2 individuals for each family. I chose these families because there was previous data on what these families did to deter that predation. So Arabidae was known for absorbing and responding with a form of click. Geometridae has the tympanic membrane, so hearing. Noctuidae was absorbing and hearing, and Sphingidae was hearing and responding. So you can see there's overlaps between the two. Overlaps between like hearing in Geometridae and Noctuidae and Sphingidae, and that was helpful to kind of look at data and see what traits could be overlapping with those 3 families. And I identified to the genus level at least of each of the species. So again, these were the shapes of scales, and I aligned them with the columns so it might be easier to see of the species that I collected and what I found in the totals. And I predicted that each family would have about the same scale shape because they're part of the same family, but I found that to not really be the case. The only one was Arabidae that had the same scale type of C, which is this one right here, and that was pretty unison and in line, which I'll show you in the next slide. But the rest of them really surprised me, especially Geometridae, which it has only 1 overlapping scale type, and the rest of them had, and the 2 species had 3 types of scales that didn't overlap with each other, which was pretty cool to me. So next, I'm going to show you some images of these individuals and magnifications of their scales. So this is, again, the scale type C, which has like the laceration in the middle. Geometridae, which had no overlapping, it had D, but it was assumed to be a lot different. You can see here is like a triangular shape, and here is like a 3-peak. So these were all taken through a microscope. So the next, I did area of scales. And again, I did this with image J. This is a photo of what image J looks like. I can use a tracing icon and choose the exact scale. And I made sure to do that with the right magnification for each of them. These were the averages of sizes in nanometers. I did it obviously in micrometers and then had to convert because they were so small. But these were the averages, which led me to my data. And there's a lot going on in this, but basically, I found that most families were statistically different. Most species were statistically different from the other species in their family, except for Geometridae, which is AC. I did an ANOVA test and TUPE test with the help of my professor because this was quite a complicated one, seeing as we go up to D, E with the letters. But yeah, so Geometridae was the only one that really stood out to me as being the only one that was statistically similar to its pair. And across the families, some had overlaps as well, which I wasn't expecting. Overlap of scales, like I mentioned, there were 2 different ways of determining them. I decided to do 10 scales per individual, randomly selected. Sometimes I also asked people who didn't even know what I was doing to select 10 scales, so I really was a blind study as to what, we had limited bias as to the scales I chose. This is what I got, that for horizontal scales, which is the blue columns that you see, the x-axis is the species and the y is the degree of scale overlap with the data that I generated. With horizontal, everything was statistically significant to each other. Families would have stronger overlaps with the data that I studied, and that was not the case. So thank you to Emmy, Naomi, Estacion, Instituto, and my host family, and everyone else that helped me with catching moths and dealing with them. It was a lot, but yeah, thank you.
Thank you, Dre. There is enough time for questions.
You said that the bats will respond with like feelings themselves.
The moths.
Yeah, the moths will respond to the bats to display like toxicity, and that's not through the scales. It's them like they're feeling ultrasound from bats and then responding with a click, and the bat then understands that as toxicity.
The bat, so it's a warning symbol that's like, hi, I'm toxic, don't waste your energy on me. A lot of them that do produce that sound aren't actually toxic even, but the bat rather not waste its energy trying to figure out if it's lying or not.
Sorry, I was just missing. How do they make that sound?
I'm not quite sure. I didn't study the sound production, but I know that it's interpreted through scales in the tympanic membrane. So I'm guessing produced via mouth, but I don't know. That'd be my best guess.
Another question? Okay.
Andrea, can you find variation in the type of scales among the wings of the same moth? Or the scales in the moth, are they always being one type, or there is variation among the wings?
So to make sure that I was concise and didn't, if I studied the whole wing, it would take me months. So I decided that I would only do the center of the wing, like I said, because of surface area and then also less damage on average for all of the species I collected. So I know that with a lot of species that have like a tail, like there's like tail scales like in the wing, yeah. It's like the edge part of the wing for the people that don't know about bone. There's like normally different scale types, but because I was only studying the middle, I didn't want to include it unless it happened to be in that part of the center wing. But because of the surface area, I was like, that probably isn't going to affect it as much as what the majority of the wing is is made of, which is the scales that I mentioned here. Frank?
This is a question, maybe just a suggestion. When you do your final submission, you might check out some of the literature on the micromorphology of owl feathers, because it's a different thing. They're trying to not make detectable noise so their prey don't pick them up. But there's some interesting parallels between owl feathers and your scales.
Okay. Thank you, Dre. Okay, our next presenter is Rox Chávez. She's going to teach us about these very charismatic wiggles, but very, very understudied here in Monterrey. Thank you, Rox.
Hi everyone, my name is Roxana, and my topic will be about the natural history and female mate selection of the Lixus address we were in Monterrey. I first became interested in the species during a orientation walk when I first arrived to Monteverde. And in this orientation walk, my group saw a weevil on the plant Simposyum, and when it was held, we noticed that the coloration on this weevil seemed to rub off easily. This was very interesting to me, so I began to do more research, and I found nothing specific about the coloration or the species. The only thing I found was their taxonomic grouping classifications about its species Lexus apterus and its order Coleoptera and family Corcolionidae. There's other information like that these weevils are herbivores and their interactions with plants can be mutualistic due to their ability to contribute to pollination. This species is distinguished by the yellow and red wax-like substance that covers their body, and they are also found. I only found them in the high elevations, which is near the TV towers. And this presentation, I will cover the main points of their life cycle and try to answer the questions: can they fly, and does the wax-like powder found on Lixus apterus weevils influence female mate choice? The methods. So during the 2-week span of research, I would go to the TV towers in Monteverde and look for the plant Simposyum, where I collected 12 weevils and gathered branches from this plant. I also froze a pair of weevils in order to look under their elytra for wings. In the first, also made sure that in the 12, at least one of the 12 weevils collected contained at least one male of the collected weevils contained its color. And I placed the colored male and a male without color in the plastic container as shown in the image. And then I placed a female. This was considered one trial, and I did 9 trials in total. And in every trial, there was a different female. So let's end discussion. What I found was that Simposyum is their primary habitat and host plants. I also found that this was their mating season, and they like to mate on leaves and branches. They also lay eggs within the pith of the plant's branches, as shown in the second picture. And I found that in the first picture you can see 2 small holes. One of them is slightly covered, and that is where the egg or larva is found. And in the second hole, you can see that it's open. That's where you will not find the larva. It's no longer inside the branch. And the wing morphology, I also studied the wing morphology and found that after looking at the elytra, that they don't have a big enough wing for flight. It's underdeveloped. And it's important because their name or epithets after us means without wings, which now it makes sense why this is their species name. And although the species name should have given it away, there was no information stating this fact specifically. And this leaves us with the question of how are they found in high elevations if they can't fly? And then we have the coloration. As you can see in the first picture, you can see a bright yellow on the side of the elytra and a bright red at the end of the elytra and abdomen. When I took a sample of this wax-like substance, I learned that it was not pollen, but it rather looked, under the microscope, it looked like small sticks. And I was not able to find what specifically the wax-like substance was. But I learned that when I left them in the clear container for 2 days, they seemed to regain their coloration, which suggested that they probably produced this wax-like substance. And this is a weevil without coloration. So this is what the male without coloration looks like. And then during the sexual selection or the mate choice trials, I found that the female preferred the male with color 7 out of the 9 trials, and the females preferred the male without coloration 2 times out of the 9 trials. This suggested that coloration plays a role in female mate choice, which could indicate reproductive fitness. And although it may seem like a very popular trend, my trial sample is a bit too small for me to specifically say that it's significant. And then during the trials, I also observed that there was male-to-male mounting behavior in 2 out of the 9 trials, which suggested that males try to mate with others regardless of their sex. Also found that female behavior of leg rubbing and antennal movements can suggest that that's an acceptance and mating, and they're ready to mate. And also found that they are very aggressive. For instance, during 2 trials, I observed a male that used its snout to push against the elytra of a competing male. And so I did not figure out why they produce the coloration, but it seems like it's very important for their sexual selection, and that might be one of the main reasons why they produce it. And some limitations were time and the constant rain. And I also found that my study might not be fully significant due to my inability to find any past research on the ability of weevils to see color. Yeah. And thank you to Emilia and Naomi and for guiding me throughout this project, and Francine for going with me to TV Towers and the Monteverde Institute, Estación Biológica, and UN for pinning my insects, and the Familia Ugantes Salazar for making me lunch so I could take to the TV Towers.
There's time for questions.
So you said that they do, like, you think they do produce the whatever the dust waxy stuff was. Did the non-colored male try and gain color to compete with the colored male, or did that not happen?
No.
No.
They it wasn't until like I left them in the container that I saw that they were gaining their color back. They need time to produce it.
During the mating trials, was there always the plant throughout the, was it always present, or would they mate without the plant?
They mated with the plants inside of it. Yeah. Yeah.
What do you think would be like the most interesting future direction?
I think seeing if they can actually see the color, or if there's like any other way they know that the male has the coloration.
Why do you think that the coloration gets rubbed off?
I noticed that you could easily, I think because it's not part of them, so it's like, it's just like a powder. So like even when I would go up to TV Towers and it was raining, some of them did not have their color. Yeah.
Is something that like comes and goes?
Yes, that's what I know.
Are you using the word coloration as sort of a proxy for the substance, whatever it is? Like, you could also say colored substance, right?
Yeah.
Okay. So it's not necessarily the color per se that seems to be making a difference. Cool. Thank you.
Okay, we have some news. We're gonna eat lunch first and then finish this session. So sorry, Brian and Blanca, you're gonna have to wait.
Gastropod.
You know my insect snails? Smallpox. I love snails.
We're gonna keep learning about this abundant but very undescribed species. And now we're going to go back to Guajini Q and learn about firefly larvae by Brian Sirudom from UCEAP. Thank you, Brian.
Hi, so I'll be talking about the microhabitat selection, substrate preference, and abundance of firefly larvae along the mangroves of Guajini Q. So just for an introduction, the specific larvae I'm studying is the larvae of the Florida tidal firefly. And this was actually, they reside in Florida and the Bahamas, but they were most recently, as of last year, discovered in the mangroves of Guajini Q. And so they're very understudied, and they have 4 specific life stages: egg, larvae, pupae, and adult. The adults are distinguishable by their very ovascular shape, as well as the yellow margins on the ends of their elytra. And the larvae themselves are surveyed through the distinguishable 2 small light organs on their back dorsal plate that kind of looks like their tail. And so just more context is that the larval stage is the longest stage for the adult for the firefly. It's 3.5 weeks or months. And they're also predators of intertidal snails. So they have been known to be captive to eat anything that they could, like opportunistically, like dead matter in plants, but they mainly eat intertidal snails. And so another thing to also know is my study was conducted after a flood had happened in Guajini Q, and the mangroves were flushed with a lot of fresh water. And so the questions I'm asking are, what are the microhabitat preferences? How do substrates play a part? and what is the relative abundance of these larval fireflies along the mangroves of Quantum. And so for my methodology, for approximately 10 days, I went to the mangroves every night and just observed the natural history of the larval firefly. And I did this to better understand where they might be, and also for every individual detection, I took a photo and identified what substrate they were on. And then during the second week, I did 7 transects. I did about 1 to 2 each day, usually during low to mid tide. And these transects took about an hour each, and each quadrat was 1 by 1 meter. And I took 5 minutes per quadrat, just looking for vegetation, larval abundance, and the substrates they were on. And furthermore, I built a terrarium, as you can see over there. And I added just vegetation to simulate a mangrove ecosystem to see if I can identify and see behaviors of larvae to better understand where they are in the mangroves. And so this was my study site. I used the GPS to map the coordinates of each transect I did. And even though I did it fairly randomly, I just laid out my quadrat and started conducting my survey. You can see there's like some distinguishable area where towards the bottom, that's where the tide comes in more normally, and towards the top, it's way drier. And my hypothesis also is that they're going to gravitate towards more damp vegetation, and that their abundance will be low because of the flood. And so for my results, the most common substrate is wet soil. It's not much of a shocker, that's the most abundant, and that's where the most cover of the mangroves is. It's wet soil and mud. But I do want to acknowledge that they were also present in sedge and decomposing wood, as well as other substrates, and that with the availability of wet soil, they were still, and other substrates around them, they were still just staying in the soil. And I think I can attribute this partially to the diversity of the different compositions. So there was like clumps of mud, there's holes made by fiddler crabs, and there's also just like different moisture and health qualities within the soil. So even though there was a lot of things around, I always primarily found them in the mud. And so I also did a vegetation index of just the percentages and distribution of all the larvae I found in my transects, and I found that they were mainly in pneumatophores, sedge, leaf litter, and decomposing wood, which once again supports the fact that they're in these damp microhabitat areas where there might be good nutrients and potential prey that they can eat. And so out of the 56 larvae, or in total I observed 56 larvae of the firefly for the Florida intertidal, and they were on 7 different substrates, and this was pretty prevalent. And for my transects, I assessed it based off of 2 variables, or like 2 different factors. The first one was I started my quadrat 0 meters from the river, and I went up to 10 meters away from the river, and there was no significant relationship at all between the river line and abundance, so I wrote that out. And then I also did my transects based off of their proximity to the intertidal area and farther out in the mangrove. And as you can see in the left model, where with the individual transects, transect 1, which had the most abundant amount of larvae, and this is an area that's constantly flushed with with the mangrove water during the high tide. But overall, there was a low abundance. I only found 21 larvae in the 7 transects I did, and 77 quadrats, and I think this is possibly due to the fact that there was a flood, or there might just be a low total population. And also, I didn't collect any experimental data for my terrarium, but I did observe a lot of behaviors of the larvae that they displayed. For instance, even though they love to be in the mud, they're always clean or spotless, so they clean themselves up. I witnessed them literally getting the mud off of their body. And they also coil up, which is hypothesized to possibly be because when water comes at them, they can just float away. They moved into loose soil in the terrarium, so just whenever the soil they were able to kind of nudge their way into a burrow, they were able to do that, and they also move into holes that were made artificially, and I've witnessed them in the mangroves going into holes of fiddler crabs. Other additional observations were that they climb, they walk along substrates, and something I found really interesting was I submerged one, like I just like put in a container and just put water, and it was just walking as if like nothing had happened. It wasn't trying to swim or anything. So that might be an interesting reason as to why they are in the intertidal area. And they also move along edges, which might be some energy conservation. And so basically my conclusion is that I think larval diet preference could play a huge part in microhabitat preference, in that they choose to be in areas that have excellent cover, moisture, and nutrients, and that their substrate preference also reflect that because they are usually in moist materials such as wet soil, decomposing wood, and sedge, and that their low larval abundance may be due to post-flood conditions or the general low population, which I'll get more into right now. So the Florida intertidal firefly larvae are heavily understudied, and they're also an endangered species. So the fact that they're here in Guayaquil, Costa Rica is a very significant thing and should leave space for more study. And I think future studies can focus on like the salinity, nutrient, and moisture of the soil content, as well as their feeding ecology. And I think because of like increased severity and frequency of tropical storms, that it's even more important to study like fireflies and firefly larvae, especially because over the years their populations have dwindled. And I also want to just thank my primary advisor, Emmy, and also my secondary advisor, Frank, for helping me just learn more about this species. I want to thank Grace for coming to the mangroves with me every night, even at like 11 p.m. or 12 a.m. I want to thank my homestay family because I didn't actually have a key, so they always had to open the door for me every night. Monteverde Institute, and then this is my homestay dog, that one right here, that's Toby. So thank you to Toby and all the dogs for finding me cute.
There's enough time for questions.
You said that the diet might impact the habitat choice. Are they eating the mud, or like what are they eating?
No, they're, well, like past studies have only found that they eat intertidal snails, but in captivity, studies have found that they'll eat any type of plant matter or organic matter in captivity.
How recently did you say they were first identified?
Well, in Costa Rica last fall. By Emmy. Sasha.
What dog was your favorite?
Probably Toby. Yeah, he was my homestay dog, and he was really excited to see me every night. So.
Any other question? Thank you, Brian. Okay, our last presentation of this session by Blanca Saiter from UC Berkeley. She will teach us about also a very understudied location, which is the canopy of Monteverde and the arthropods that are in there. Thank you, Blanca.
Hi everyone, I'm Blanca, and I'm going to talk about vertical stratification of beetles and other arthropods in the ficus tree here in Monteverde. And by vertical stratification, I mean the different layers of habitat that exist. And in this case, we're looking at the understory of the forest and then in the canopy. But there are other layers also, like people will study the middle. In this case, we're only doing two. So I'm going to start with a quote by William B. Yet another continent of life remains to be discovered, not upon Earth, but 100 to 200 feet above it. There awaits a rich harvest for the naturalist who overcomes the obstacles and mounts to the summits of jungle trees. So I do want to mention that this concept of discovery is a bit colonial and coming from the lens of the Western scientific community, kind of like Europeans thinking they discovered things that indigenous people knew about already. But it is true that there is a lot in the canopy that we don't know, and there's a huge gap in the knowledge in the scientific literature. So on that note, I want to talk about the importance of studying arthropods and insects. Insects are the most diverse group of animals in the world. They make up more species than all other animals combined. And about 80% of them are undescribed. So huge gap of knowledge there in the insect community. And forest canopies may harbor up to 1/2 of these species. And Dr. Nalini Nadkarni is a pioneer in canopy research here in Monteverde. And she describes the canopy as a keystone component of forest ecosystems. So it's a really important place to focus our efforts. So that leads me to my research questions. I have 2 of them. The first is general arthropod composition. So what arthropod communities are present in the canopy and in the understory of the Spicus Turkheimii tree, and how do they differ from one another? And then I'm looking more specifically at beetles, because in my humble and correct opinion, they're the best order of insect. And so my question is, what morphospecies of beetle is found in the canopy and the understory of this tree, and how, which location supports more diversity and abundance? And I expect there to be different communities, essentially, of arthropods, so I expect to see vertical stratification. So the collection site was by the Elida Lodge in the Monteverde Whole Foods. This beautiful ficus tree. The middle image is of the understory vegetation, and then the image on the right is in the canopy. And I used 2 collecting methods. So the first one was branch feeding, and I essentially held an insect net below the vegetation, and then I smacked it a bunch of times with a stick, and the arthropods would fall into the net, and then I would transfer them into a plastic bag. And I did 30 seconds in each location, so in the canopy and in the understory, and I did that twice, so a total of 1 minute of collection. And then I also set fermentation traps, which are a mixture of beer, molasses, rotten fruit, water, yeast, really delicious stuff, and insects love it. So I set one of those traps in the understory using this jug, and then also one in the canopy. And I put 500 milliliters of the mixture in each one, and they were set for 96 hours. So our results, in the canopy we had 139 specimens in 10 different orders, and in the understory we had 122 specimens in 8 different orders. So you can see Diptera here are the flies, and those were the most common in both locations. They were more abundant in the understory. And then other important differences between the 2 locations. There were 4 orders that were only found in the canopy, and that's Lepidoptera, which are the butterflies and moths, Blatodia are the cockroaches, Orthoptera, crickets, and grasshoppers, and then Refuge are like really creepy looking arachnids. And it's important to notice that obviously these insects also exist, or arthropods exist on the understory as well, but they were not found in the traps. In the understory, there were 2 orders that were not found in the canopy. The Psychoptera, or the book lice, and the Amphipoda are actually crustacean. And this is the relative abundance of both of the locations. So I like this because you can kind of compare. Here you see how Diptera, the flies, were just dominant in the understory. And in the canopy, they were very common as well, but you see more of other orders, and they're more abundant in the canopy. So looking more closely at the Coleoptera, I collected 8 families of beetles, and there were 17 morphospecies that I identified, and 39 specimens total. So in the canopy, there were 30 beetles. There was over 75% of the beetles collected in the study were in the canopy. And only 9 were collected in the understory, and the most common family were Metadulidae, which are sap beetles. So looking at the vertical stratification and the differences between the 2 locations, the canopy had 10 morphospecies that were distinct and only found in the canopy, not in the understory. The understory had 5 morphospecies that were not found in the canopy, and then there were only 2 morphospecies that were shared, so they were found in both the canopy and the understory. So the best part is looking at these beautiful beetles. So these are the morphospecies that were found in the canopy. There are 5 families represented here. Most of them are Nidodulids, so all of these are Nidodulidae, this one as well. And then we have some other families here. This is an interesting one because it's a carabid, which is a ground beetle, but it was found in the canopy. So you can see a really big diversity here of morphotypes and different beetles, which is really fun to see. These are the understory morphospecies. So here we have a weevil with interesting texture. This one was found in the vegetation during branch beating. And then both of these down here are Nidodulids, also the same family. These are the ones that were found in both locations. And this is represented by 2 families, Nidodulidae and Tenebrionidae. So, the canopy overall supported a higher diversity and abundance of arthropods, and there was clear stratification of arthropod communities that were found in the canopy and in the understory. One thing that's important to mention is that different collection methods will target different taxa, and insects and arthropods are so diverse and they live in so many different microhabitats that it depends, like whatever method you use will determine what kind of things you're going to find. So obviously I wasn't going to find things that live under the bark or something like that using fermentation traps and beat sheet methods. But for example, with beating, branch beating, there were families of beetles that were found using that method that were not found in fermentation traps. So it just kind of speaks to the importance of diversifying your collection methods. And future directions for future studies. I think it would be important to sample multiple trees. Because of time, I was only able to sample 1 tree. So having a larger sample size and more data would be very valuable. Another thing to note is that it's the rainy season, so the insects that are out right now are going to be different than maybe in like May and June. So it'd be really interesting to see how the study would turn out in the dry season. Excuse me. And then another thing I'm interested in is comparing different forest types. So primary growth forest, old growth, compared to secondary growth. I think that this study does speak to the importance of having old, tall trees with epiphytes and like a healthy community in the canopy. Because in this study, the understory vegetation, which was younger, did not have as much diversity as this big, old, tall ficus tree. So in conclusion, the forest canopy is an important microhabitat that supports a unique biotic community, and forest canopies are an important part of biodiversity conservation, and more studies are needed to understand the taxonomy and habitat specializations of canopy-dwelling organisms. So thank you to Emmy, my advisor, Francini. I really could not have done this project without her. She spent so much time with me and taught me so much, and she even donated some of her own gear and everything. So thank you, Francini. Thank you, Naomi, for being my secondary advisor. Thank you to Eladio and Anais, my homestay family, for making sure I was fed and had enough coffee to go out into the field. And thank you to the Elida Lodge, Monteverde Institute, the station, and UCEAP for the materials and access to the study site.
Thank you, Blanca. There is enough time for questions.
How high did you go up the tree and how did you get there?
14 meters high, and Francini was really kind enough to let me learn, or she taught me a lot about tree climbing with the way that scientists do it with like a rope, and then you have a harness, and you have an ascender on your leg and everything, and then you like climb up the rope. And in this case, the tree, the ficus tree is a strangler fig, so they're hollow on the inside because they grow around the host tree. So the inside was hollow, so we were able to climb up, like basically like rock climbing, but we still had like helmets and ropes and stuff, so yeah.
What was your favorite outdoor rock climbing?
Ooh, okay, let's look back at the beetles. I really like this guy. I think he has really, these are his jaws, or mandibles. I think that's awesome. I also really like this beetle here. It's an Anthissidae, which is like an ant mimic. And it was very hairy. I don't know if you can tell here, but yeah, I really like the Nidodulids. I think they're super cute. I love their little antennae.
Blanca, do you know have been done some experiments in which canopy beetles move to the bottom, and bottom beetles move to the ground, Did you find anything like that to try to figure out, or how do you explain this specification?
So one of the studies I found was from Nalini and Jack Longino actually, and they were looking at ants and stuff, and also kind of like leaf litter and soil, and that's another thing about like, there's so many different habitats that like they were focusing on kind of the soil, and they found more density in the canopy than in the understory. I'm not sure about the like movement and like how much the insects are moving around, but I guess my explanation is just that it's a different microhabitat. I mean, the vegetation in the canopy, I was branch feeding from a epiphytic cactus, and then there's also like the ficus tree leaves themselves, and there's kind of like more foliage. I think it's interesting that there were so many Lepidoptera up there, maybe just because they're like flying or they're taking refuge during the day in the leaves. So I think there's lots of explanations.
More questions? Thank you, Blanca. Alright, I hope you enjoyed learning about arthropods and vertebrates, but now it's the turn to switch to the vertebrates. And Faye will be in the vertebrates.
Thank you. Thank you, Emilia. So, what I'm going to do is to be echándole un ojo a Frank to know if we are going to make a break or not, but we're going to start with the section number 4, which is about behavior and ecology of vertebrates. And first speaker, Grace Miller from UC Los Angeles, is going to speak about behavior of cocodrilos. So, Grace,
Aquí. Hi everybody, I'm Grace, and I'm going to be talking about responses of American crocodiles, Crocodilus acutus, to conspecific crocodilian and hominid juvenile distress calls. So a lot of people think that crocodiles are just dangerous reptiles, but they are way more complex than that. Crocodilians are the most vocal of all reptiles, with complex territorial, breeding, and distress calls. Juvenile crocodiles chirp and scream for their mothers, and adult crocodiles participate in bellowing to establish territory. Also, mothers are very protective of their babies. They'll watch over babies for months after birth and carry them around in their mouth, as you can see in the picture on the right. Additionally, there are past studies that show crocodiles reacting to the vocalizations of other animals. One study in Morocco showed now crocodiles responding to the distress calls of baby monkeys as well as baby humans. And another study with now crocodiles showed that they both understood and would respond to the distress calls of spectacled caimans, which you can see on the top. However, there's a very big research gap when it comes to American crocodiles' vocalizations, and that is one of the reasons why it is the focus of my study. I also conducted an interview with Dr. Miriam Butcher, who is a crocodile expert, to help determine what a predatory versus a protective response for crocodiles would look like. She said that parallel movement towards an object of interest, which in this case would be my speaker, signifies predatory response. She calls this an investigative cruise, where the crocodile goes past something and can either escape or go closer. Where she said perpendicular or direct movement would be a protective response. So my research question was, what are the responses of American crocodiles to the distress calls of juvenile American crocodiles, juvenile spectacled caimans, and human infants? My predicted responses were that the American crocodile distress call would show both protective and predatory responses, whereas the spectacled caiman and human infant calls would only show predatory responses. I hypothesized that the white noise, my control sound, would have no response because it doesn't have a frequency or a pattern that mimics any animal sound. And so my study sites, I looked at crocodiles at 7 different study sites. 4 of them were in the mangroves along the Rio Quahini Keel. One was near a bridge along the Rio Quahini Keel. One was at a pond by Playa Quacho Porcacho. And one study site was in Tomas Bay, the site of the mariculture project in Quahini Keel. And so my methods, I sampled 32 crocodiles. When I saw a crocodile, I would turn on my speaker and I would play one of 4 distress calls. The first one being the American crocodile distress call, which sounds something like this.
also would play the spectacled caiman distress call. This one's a little bit loud. The human infant distress call. Or the white noise, which just sounds, okay. That one's glitching. It doesn't sound like that really. And then I would train my binoculars on the crocodiles and watch them for 10 minutes, recording anything that I saw with a voice recorder, whether this is behavior or movement, and this is me observing the crocodiles. And when it comes to analyzing the data, immediate responses of the crocodiles to the calls is very important. So for the American crocodile distress call, the crocodiles 33% of the time remain stationary, which may sound like a lot, but is the lowest out of any of the calls. Also, 33% of the time the crocodiles actually appeared after the sound was played, which means that they surfaced from the water. And one time the crocodile was seen swimming directly towards the speaker. In terms of the spectacled caiman distress call, 75% of the time the crocodiles stayed completely stationary. For the human distress call, 50% of the time they stayed stationary, and one time they did appear when the sound was played. And as for the white noise, 66% of the time the crocodiles did not move, and one time the crocodile actually turned away from the speaker and started moving away, which was a response unique only to the white noise. I also analyzed movement responses over the 10-minute period. The American crocodile distress call had the most movement responses with 15 total, and 9 times the crocodile moved closer. There was a similar pattern with the human infant noise. 7 times it moved closer to the speaker, and then the spectacled caiman showed the opposite, where 5 times out of 8 the crocodile would move farther away from the speaker. And as for the white noise, the crocodile moved about equally in each direction. And then I had one observation that was very significant that caused me to perform more tests. I was sitting eating my lunch, no crocodiles in sight, and I decided that I wanted to just play the American crocodile distress call. When I did, a large crocodile came out of the woods, jumped into the water, and began swimming towards the speaker. It only turned parallel to me when it saw me. Before that, it was swimming directly at the speaker, which indicates potentially a protective response. And this observation caused me to do more testing specifically with that conspecific American crocodile distress call. I tested it, playing it for 3 to 5 minutes, and analyzed 6 crocodiles with that playback. 5 out of 6 times, the crocodile immediately started moving towards the speaker. 3 of these times, it moved directly towards the sound, and twice it moved parallel towards the sound. And analyzing their movement responses in total, 12 times out of 13, the crocodile moved towards the speaker. Only once did it not move at all, and it never moved away from the speaker with these additional tests. And so the overall responses show that the American crocodile distress call shows both protective and predatory responses. The spectacled caiman, the crocodiles displayed some disinterest. For the human infant, they displayed interest, and the white noise control showed no consistent response. So what does this mean? American crocodiles exhibit both protective and predatory behavior towards juvenile conspecific distress calls. Behaviors such as surfacing or appearing when the sound is played, as well as moving closer, parallel towards the sound, suggest intrigue or predation. But my additional testing should suggest an innate protective response of these crocodiles, considering that it is not nesting season and there are no baby crocodiles in Bikini Kiel. This shows that the crocodiles would respond to babies that are not their own and when they know they do not have babies to protect. Further research could be done with male versus female crocodiles to see if potentially mother crocodiles or female crocodiles would show a more protective response. As for the spectacled caiman, there was some displayed disinterest but no consistent pattern. This could be explained by a lack of territorial overlap between caimans and crocodiles. I saw no caimans where I saw crocodiles in Bikini Kiel, so I saw no overlap. And then in terms of the human infant distress call, there was interest, but again, no consistent pattern. This could be because the crocodiles may have a pre-existing fear of humans due to the fact that they are hunted and poached in Bikini Kiel, which could affect the consistency of human-infant responses. And this study is important because it provides behavioral insight into a very understudied species that is becoming more and more prominent in Bikini Kiel and the Nicoya Peninsula. And in conclusion, American crocodiles show both predatory and protective behavior in response to juvenile distress calls. This suggests that in addition to viewing juveniles as potential prey, an innate protective instinct exists. And I would like like to thank Bryant for coming with me to the mangroves every single night, Frank for being my primary advisor, Emmy for being my secondary advisor, Leo for helping me out with my research, Flora Laura Victor for opening her house to me, Will for also joining me in the mangroves, and Dr. Miriam Butcher for providing essential insight into my study. Thank you.
Okay, time for questions.
Were the treatments done to all individuals you saw, or do you have a preference to only use the mosquito individuals?
There's not any way to distinguish the gender on crocodiles unless you would catch them and look at the cloaca. So any crocodile I saw, I tested with 1 sound per crocodile.
What would you do if, like, one crocodile stayed for a while? Would you move spots to go to find like another crocodile, or how was that?
Yeah, so if I saw one crocodile and then played the sound, I would watch it for 10 minutes, and that was sort of the testing period. After those 10 minutes, sometimes I would experiment with other sounds and see what would happen. And then in the scenario where there were multiple crocodiles like that were in the same place, I would just play the same sound for all of them at the same time, and I would have usually somebody there with me also keeping track of their behavior. Yes?
Did the crocodiles ever respond? Did you ever like to stop using?
Yeah, so they love the red hot chili peppers, specifically scar tissue. I accidentally tested that a couple times. They responded great.
Bruce, do you know if the crocodiles can hear sounds from above water while the crocodile or the seabird is underwater?
The crocodiles underwater?
Yeah.
I talked to Miriam Butcher about this. She said that yes, they can hear to some degree. It's unknown whether it's like the vibrations that they hear or if they can actually hear the noise. And she said it also depends on like their distance and if there's any debris or stuff in the water. But she said that they can hear.
Muy bien. Oh, last question.
Can you demonstrate the big American crocodile sound?
I can try.
That's it.
That's very cheap.
Somebody's computer, whose computer is this? So, welcome next speaker. Conner Tequeda from UC Santa Barbara and Miguel Suarez Evia from UC Santa Cruz. They're very dynamic pair working, jumping streams and attempting to measure rocks in the stream.
Thank you, FC, for the introduction. My name is Conner Tequeda.
And my name is Miguel Suarez-Evia.
And our study is on the effects of environmental factors and stream characteristics on anaerobic activity at stream sites. Just some general context for our presentation. The term anaerobic refers to toads and frogs, And over time, anurans have evolved adaptations in order to reproduce in streams. The benefits of reproducing in streams are that streams are more constant supply of water versus ponds that are more susceptible to desiccation or drying out. And so they have, or a con of laying eggs in streams is that eggs may get washed out by the flow rate. So in order to deal with this, they have adapted several adaptations, such as the glass frog, in which they lay the eggs on top of vegetation above the streams, and then when the eggs hatch into tadpoles, the tadpoles will drop down to continue their life cycle. Other species of frogs will attach their eggs to the underside of rocks in order to make sure that their eggs don't get washed away by the stream. And they have also adapted, some species of frogs have adapted calling behaviors, in which the males will call out to the females to find a mate. There's actually been studies shown in which the males will actually call out to each other in a type of competition, and the loudest caller will eventually get the mate.
Yeah, and in preparation for this study, we identified a handful of environmental factors and stream characteristics. These can be split up into two basic groups. The first being the macroecological features, which are more on a broader scale than just the stream habitat themselves. They include the likes of precipitation and elevation. And then we also looked at microclimatic features, which are more specific to each stream habitat. And these include stream size, temperature of the water, and pH of the water.
And so the central question of our study was how do environmental factors influence anaran abundance and calling rate within stream ecosystems.
So getting into our methods, we picked out 4 different study sites for our study. We laid out 50-meter transects, 2 of them at each site, except for one, which Connor will get to in a second. Our first site was at Casa de Arte, which is centered in Cerro Plano. Our second was here, behind the Institute at Crandall Reserve.
Our third survey site was the Simposyum biologica stream. These 2 transects were along the same stream, however, just 10 minutes apart from each other. Something to notice about this stream was that the high precipitation events in November likely caused there to be less vegetation, and the vegetation in Kennedy got washed out, so the streams are a little bit more open. And our 4th survey site was San Gerardo. Due to logistical constraints, we could unfortunately not get a second transect. However, something to notice about this survey site was that there was an increased amount of canopy cover, as you can see in the picture.
So here we have a little diagram of the different characteristics that we took at each site. We took all these variables every time we visited, except for 3, canopy cover percentage, furthest distance from stream to vegetation, and elevation we took once at each site. Every night we would go survey one site, and we would do a 45-minute visual survey. We would walk the length of the 50-meter transects, looking for as many frogs as we could find, taking note on species and abundance. And then we would also take a 5-minute recording of calling activity to get a count on calls.
And so in analyzing our data, we used a generalized linear model, or GLM, to compare predictor variables to response variables. And basically what this means is the predictor variables would be the environmental factors that Miguel just talked about, such as canopy cover, pH, flow rate, things like that, compared to the response variables being species richness, abundance, and calling rate. These were useful to see the environmental characteristics, which ones were most predictive of anaran presence and activity, and we ran 42 models in total, and yeah.
So here we have a list of the anaran species that we found during our study period. On the left we have the ting frog, or Diasporus diastema, emerald glass frog, Espadarana proseblepon, brilliant forest frog, Lithobates worzewiski, and then the rufous-eyed brook frog, Duomenohyloguphioculus.
Going to our results, over our 12 survey nights, we observed a total of 69 observations of frogs. The abbreviations of the sites read as CDA is Casa del Arte, CR is Crandall, EB is Simposyum biologica, SG is San Gerardo. As you can see, the site with the most amount of observations would be San Gerardo, and the species that we observed the most was Espadarana proseblepon, or the emerald glass frog.
So continuing on with our results, here we have some graphs that we got from our generalized linear models. Essentially, like Connor was saying, we ran 42 different variables and combinations of variables, and the model popped out the variable that was most predictive of both annual in abundance and annual in call rate. So on the x-axis we have temperature, and then on the y-axis we have abundance and calling rate. And as you can see, as temperature increases, we see a trend of more frog abundance and more calls when the temperature is higher.
In explaining this correlation between water temperature and increased abundance and calling rate, our study suggests that colder water can produce breeding activity if it's not within the suitable range. And this is backed up by previous data in which elevated temperatures have shown to stimulate calling behavior because it influences their muscle function along with their energy expenditure. And something else we found was that precipitation was assumed to be a predictive variable for calling rate and abundance. However, due to logistical constraints, we could not find precipitation data for San Gerardo. However, a general trend that we did find was that on nights where there are higher amounts of precipitation, there were more sightings and observations of frogs, as well as more calls heard.
And lastly, we did note differences between stream habitats in the presence or absence of certain species. This is exemplified by the emerald glass frog that seemed to prefer habitats that had less of a distance from stream to vegetation, and also higher canopy cover. And this intuitively makes sense as the glass frogs like to call from vegetation for mates and to prevent from desiccation and predation. So yeah, this is just to say that even though water temperature was the variable that we found was most predictive of abundance and calling rate, it's not to say that other characteristics of the stream are not important for whether or not certain species are present at sites.
In conclusion, in pursuit of our question on how environmental factors and stream characteristics influence enderins, we found that water temperature is most critical in predicting their calling activity and abundance. And some interesting just future directions that we could have taken with our study. Unfortunately, we did not find a significant correlation with the other variables, things like canopy cover and flow rates and pH, things like that. So really, past studies have found that a correlation between canopy cover, so doing a study on predicting how specific types of vegetation or the absence or presence of palms, things like that, could affect enderin abundance, as well as seeing how the different amounts of pH or things like that.
Adding on to that, I think it would be interesting to see how a study like this would pair up in like a different time period. So potentially during like the wet season when it's different factors are affecting the enderins, and also different frogs reproduce at different stages. So there could be different species that we find doing it at a different time.
Yeah, and lastly, a lot of people helped us with the project, and we couldn't have done it without them. We'd like to give a huge thank you to our advisors, Federico Chinchilla and Natalie Sanchez.
Our homestay family, Elion, Karen, Ethan, and Luan.
The frog experts, Eladio Cruz and Matt Moran.
Our wonderful peers for helping us with that project.
And our family and friends at home who supported us from afar. Thank you.
Very good use of the time. And now we have time for questions.
I don't know what.
Are you recording, Colin?
So I know that there are some frogs that prefer like faster currents or slower currents. Did you think about that, take that into account observation-wise, or how do you think that could affect your data?
In terms of our project as a whole, we focused more on like the larger abundance, but we did notice that at one of our slow-moving sites, Casa de Arte, there was a higher abundance of the brilliant forest frog. And we didn't find that number of species like as large as those anywhere else. So we think that's one that was preferred still habitats. While we know that past studies have found that emerald and glass frogs prefer faster-moving streams because they have their eggs on top of vegetation above faster-moving streams. So.
And also the Rufus-eyed brook frog, which is only found at the San Gerardo streams, that was the stream that was fastest moving, which is interesting that was the site that it was present.
A little space. Did you graph show the data for the stream temperature? Can you put that graph up again? So, again, so take those temperatures in the very same place, or is that the range of a bunch of different streams in different places?
So for each transect, we did a temperature calculation. We took the temperature at each transect every single night that we went. So we would do like 4 transects a night, and then 4 different taking of the temperature.
So do you have any sense of like what else accounts for the differences in temperatures that you took in streams, and then what else might be associated with that, correlated with that?
Well, we only surveyed one site a night, so that was kind of the difference. Like each night was a different temperature at the sites, or like, so one night we would do Casa de Arte and then take the temperature of the water there, and then the next night we would do Crandall and Estacion, and then so it was like a combination of all those nights, and then different points are the different survey nights.
So the temperatures are basically a proxy for the site.
Yeah, they were taken at each site, at each transect.
Yeah, okay. I'm probably not making myself clear. I'll ask after the break.
Okay. Okay.
What was y'all's favorite frog?
I think the emerald glass frog.
Yeah, I think the emerald glass frog is pretty cool. We also observe its calls a lot. They have like the 3 like dink dink dink. So those are pretty cool to observe. And they're also see-through, which is cool.
Sorry, could you try and do that call?
They're like ding ding ding. Like that. So,
Gracias. So everybody, next speaker, we are keep on jumping between Monteverde and Cotton Hill. Y ahora we're going to listen to Amethyst Douglas from UC Santa Cruz, speaking about rodents in islands. Welcome, Amethyst.
Hi everyone. My name is Amethyst, and for my project I investigated the presence of rodents on islands. And just a little bit of an introduction for my project. The 2 main components are rodents and islands. So a little bit about rodents. They are herbivores, and they can have a significant impact on the flora of the ecosystems that they inhabit. And then they are also predators. Usually they are opportunistic predators, meaning that they will eat whatever is around them and available. So in some cases this can be small animals, insects, and then in some cases also birds, their young and eggs. And then some notable species are Rattus rattus and Rattus norvegicus. Both of these are invasive species and can have significant impacts on the environments that they are in. And then islands are important because they are isolated habitats, so sometimes they have predators and sometimes they do not. So that is really important for seabirds that can use these areas for nesting, breeding areas, and then also species that are vulnerable to predation. And this is important because it's really important to understand marine habitats and then seabird conservation. And then it's necessary to understand the habitat dynamics with and without predators and the interspecific interactions that occur in those communities. And then this is also really important information for conservation organizations like the ACG and for conservation practices. So for my project, my main question was, do rodents occur on islands near Kahini Keel? And I predicted that rodents would occur on islands that are closest to the land. And so for my methods, I used a total of 30 traps. This consisted of 2 different traps. I used 15 Sherman traps, which are about 9 inches long, and they're a sheet metal trap for use for smaller rodents. And then I also used tomahawk traps, which are a wire cage. I also used 2 types of bait. One was sliced yucca with tuna on top, and the other was a mix of oats, rice, and honey. And I used these baits in the traps. And then I trapped for a total of 5 nights. And so I would go to 1 location per night. And this included 3 islands, and then 1 mainland location that I surveyed twice. And so this is the first study location. This is the smallest island. This is Isla David. You can also see that this island is connected to mainland at extreme low tides, but it is separated at high tide. And the yellow polygon is the area that the traps were set. This is Isla Muñecos, which is a larger island in the relatively same area. Yellow polygon is where the traps were set as well. This is Isla Bolaños. This is an island that's protected by the ACG. This is also home to a colony of the magnificent frigate birds, and they nest on the left side of the island on the ground. And the 2 areas is where I set my traps. And then this is the mainland site. I divided traps into 3 areas. Area A is in the camp / beach area. Area B is along a trail in the forest. And then area C is around one of the buildings. And I distributed traps evenly between the 3 sites at this place. And so the results of my surveys, I found no rodents detected on the islands. So to break it down by island, I found 27% of the traps on Isla Muñecos where they contained Bercheracinus lateralis, which is a land crab. And then in 47% of traps at Isla David, I found Conobita capresis, which is a hermit crab. And then at Isla Bolaños, I found 83% of the traps filled with either hermit crabs or land crabs. And to compare, at Fungal, on the 2nd night of trapping, I identified 2 species of rodent. The first is Rattus rattus, and the second is Leoma salvinii. I found 1 Rattus rattus individual, which is the one pictured on the far right. And the second species, I found 2 of those, and that is the one that is pictured that I'm holding right there. And just to mention, at Fungal, there were no rodents found the first night of trapping. And between the 2 nights of surveying at Fungal, there was only 0.05% of traps filled with bycatch, so anything that is not a rodent. And then this is a bar graph that's just showing what was in each of the traps. The x-axis is the location. Each location is split into 2 bars. 1 bar represents what was found in Sherman traps, and the other represents what was found in the tomahawk traps. And then it just shows the percentage. So everything in blue are hermit crabs. The orange is the crab. And then this 1 bar right here in the middle, you can see a little bit, that's the rodents that I found. And the green represents the 2 opossums that I found at Fungal. And then so just a little explanation about these results. So there was a really high number of Bercheracinus crabs, and it turns out that these are actually, these can be used as an indicator species of the presence of rodents, because they're actually very vulnerable to predation by rodents, especially Rattus rattus, which are invasive. And so the high numbers of these crabs that I found, specifically on Isla Bolaños and Isla Muñecos, represents that, or it supports the idea that there are no rodents present. And then it's worth noting that I surveyed Fungal twice, and the first night of surveying, there were no rodents found, although we do know that there are rodents present. It's important to know that just because there are no captures, it does not mean that there are no rodents present. And so some of the challenges that I faced with this project was the high number of bycatch. So especially at all of the island sites, all of the crabs and hermit crabs that we found prevented the traps from being open to rodents if they were present. And then the weather before this survey occurred could have some influence on the rodents' activity, which influences if they were found in traps or not. And then also the accessibility of study sites, and then the limitations that we had setting the traps in limited areas or places that have dense vegetation. And so in conclusion, there were no rodents detected on the islands, and to further support this, the high number of crabs that were present in the traps suggests that there are not rodents present on these islands. And this is really important, especially in the conservation context. These invasive rat species can have a significant impact on what is able to use these islands as protected space. And especially for islands like Isla Bolaños that have the frigate birds nesting there, it's really important that there are not rodents predating their eggs. And yeah, so this is just very important for seabirds in general. And then I would like to thank Frank and Federico for being my advisors, and Leo for helping so much on my project. The Lara family for providing the transportation to islands, and then for everyone who came out with me to scale islands or go out really early in the morning to check traps. Thank you so much for all the help.
We have time for questions.
How did you decide what food to put in the traps, or was it random?
So I actually asked my advisors about that, and both of them have experience with small mammal trapping and trapping rodents, and so Federico suggested using a mixture of oats, rice, and vanilla, and Frank suggested using yuca slices with canned tuna on top, and so I said that we will use both to see which one works better. Yes.
How do you think you would run the study again to reduce the chance of bycatch?
Yeah, I would definitely, I was reading a study that suggested putting traps on like plastic bins. So you flip a plastic bin upside down and then you place the trap on top of it, but so that it's only like 5 inches or less off the ground. So it's just enough where the crabs can't get in, but it's still accessible to rodents. And so I would definitely do that next time. Yes.
Out of your 3 rodents captured, is there a preference between the dates that they were for?
I think that all 3 of the rodents that I found at Fungal were in the Sherman traps that had the oats and vanilla mix. So I don't know if that's because of the bait mixture or because they were smaller rodents that they went into those traps. So there's some variables. It's a little unclear.
Go ahead.
For traps that you put on the mainland, was there a reason why some of them were like around, or why the area where you put them was around versus some others?
Yeah, so I wanted to split the areas up because the camp area, there was a lot of people and there was like trash cans present, and so I wanted to trap there because I was thinking that it'd be more likely that rats would be present. And then the area that you're thinking about that's like a zigzag, that was just along a trail area, and so we ended up just setting traps along the trail just to get some surveying in the forest as well.
Was there any crabs captured at Fungal or no?
There were. I believe there were 2 crabs captured. Yeah.
Okay. Gracias, Beatriz. Okay, thank you. And let's keep on going with this, the most interesting group of animals. Next speaker, Marisa Islam from UC Berkeley. She will speak about spider monkey intentional gestural communication in this species.
Hi everyone, I'm Marisa and I'll be talking about intentional gestural communication in the Central American spider monkey. So intentional communication can be described as a voluntary action in which the signaler is aware of the receiver's intentional state and has a desire to deliberately modify that state. And so as seen in this figure, this is the signaler, and the signaler is aware of the receiver's intentional state and produces a signal in order to carry out a specific response. Intentional communication was once thought to be unique to human language, but in recent studies it's been found to be present in other primate communication systems. One such communication system is gestural communication. In addition to vocalizations, a lot of primate species also use gestures, which are body movements used to communicate with conspecifics. So that could be if they wanted to request grooming or initiating play. Something that sets it apart from just other random body movements that they might make is that the signal cannot carry out the response of the receiver directly. It has been shown to be used intentionally and in goal-oriented manners, and it also has been described to be in 3 different modalities, which is tactile, which is when there's physical contact made between the signaler and the receiver; visual, which is when there's different body postures that are visibly perceived by the receiver; and then auditory, produced sounds, not by the vocal cords, but by the different body parts. That would be like the hands, if someone was clapping. To provide like a visualization of what a gesture could be, these are common examples of gestures that have been found in great ape species. In order to assess intentionality, there's a common and widely accepted framework that has been used in great ape species studies, and that includes 3 different conditions. The first being audience checking, which is when before the signaler makes the gesture, they'll look to the receiver and inspect what intentional state they're at. They'll then wait for a response from the receiver, and then if their signal was unsuccessful or if they didn't receive a response, they may persistently use that gesture. So I chose to study the Central American spider monkey, and a large reason for that is that there's only one study that has been done investigating intentional gestural communication in spider monkeys, which was done in Mexico. They found 52 gestures, but apart from that study, all other intentional gestural communication studies have been done in either great ape species or other old world monkeys. So there's a large gap in knowledge and understanding as to like how prevalent intentional gestural communication is across species and how it evolved in New World monkeys. So this led me to my research questions. The first being, what is the composition of the spider monkey gestural repertoire? What proportion of the gestures are produced intentionally, and what modality is more commonly used intentionally? And then lastly, if it varies across wild and captive environments. So the first population that I observed was a wild population in Cuatro por Cuatro in Guayaquil, Costa Rica, shown by the green box, and then the red indicates where I observed spider monkey groups. I observed them across 4 days between noon and 5 p.m. And I typically found them in groups of 2 to 8 individuals that were usually 10 to 15 meters off the trail. I categorized them as being semi-habituated, and that was because the trail is commonly visited by humans, cars, horses, dogs, and cats. The other population was a captive population that I observed in Las Pumas Sanctuary in Guanacaste, and I observed them on November 28th from 8 a.m. to noon. They have 6 spider monkeys there, 3 males and 3 females, and they were all well habituated to humans. So for my procedure, I would first identify the signaler and the directed receiver. I would then identify the body movement and determine whether it was a gesture and describe the context that it occurred in. I would categorize the modality of the gesture. I would assign intentionality to gestures that displayed one of the 3 conditions, so either audience checking, response waiting, or persistent gesturing, and then I recorded the response. And so for my results, I found 40 observations of gestures with 17 different types. 9 of these were tactile, 6 were visual, and then 2 were both auditory and visual, multimodal. The responses to the gestures were also categorized to be either affiliative, which is if they're showing a desire to interact with that individual, or agonistic, which is when they would either avoid or it would be an aggressive behavior. In regards to intentionality, 90% or 36 out of the 40 gestures that I observed were produced intentionally. And if you look at the heat map, all of the intentional gestures did, the signaler did display audience checking. The agonistic gestures, in this case, were object throw and object shake. Those were the only gestures in which the signaler also did persistent gesturing. For modalities, the tactile gestures were the most commonly produced, and all but 1 tactile and 3 visual were produced intentionally. And so intentionally was not significantly different across modalities, which shows that all modalities show intentionality pretty equally. When comparing wild versus captive environments, the captive population I observed 18 gestures, and in the wild I observed 22 gestures. The captive spider monkeys displayed significantly more gestures that responded with affiliative responses, whereas the wild spider monkeys produced gestures that were more agonistic. And then lastly, in addition to conspecific gestures, I also noticed gestures that were being made to other species. So in the case of the captive spider monkeys, 22% of their gestures were made to other species, which was an iguana and a capuchin, which were in their same enclosure. And then 36% of the gestures of the wild spider monkeys were directed towards me when I was observing them. So, something that was interesting is that the heterospecific gestures of the captive spider monkeys were entirely affiliative, while those of the wild individuals were agonistic. I'm not going to go over this too briefly, but these were all the gestures that I observed and described. And something that was interesting is that with the study done on spider monkeys in Mexico, approximately half of the gestures that I observed also were in their study as well. So for the discussion, the predominance of tactile gestures does correlate with their ecology because they're very arboreal. There tends to be a lot of vegetation and a lot of leaves that might be obstructing their views. And so tactile gestures can allow them to still produce a successful gesture despite any visual obstacles. Affiliative gestures reflects that they're very social animals and that it helps them maintain group dynamics and interactions. Audience checking may serve as the backbone for intentionality, and that's because I saw them across all intentional gestures, and then response weighting and persistent gesturing may be like kind of a backup option if the first try doesn't work. Signalers only displayed persistent gesturing during agonistic gestures, so that suggests that in times of like heightened urgency or aggressive and avoidant behaviors, they need to gesture more in order to get their gesture across. And then because they were able to gesture across all the modalities, that shows that they're highly flexible and adapted to different social and environmental contexts. And then the agonistic behavior being more frequent in the wild populations suggests that that could be due to just increased competition for different resources, such as food. So in conclusion, the Central American spider monkey uses intentional gestures to communicate with both conspecifics and heterospecifics, and thus it's an important study species for investigating gestural communication in New World monkeys. Some future studies could look more into how it develops with age, how the receiver is able to encode a signal, just a better idea of the gestural repertoire, and then further effects of captivity on intentional communication. I'd like to thank my advisors, Natalie and Frank, Leo for all his help in Pahinikio, Sasha, Brian, and Claire for helping me hunt for monkeys, and Claire, thank you for the photos, the Castro Campos family for hosting me in Pahinikio, and then the team at Las Plumas for allowing me to conduct this study.
Now we are in time for questions.
Can you show us your favorite gesture?
I like the side wrapping one. It's like a side hug. They would like give each other hugs.
What were the gestures they were doing at you?
Oh, they would like to do object throw. There was one day that I was observing them and I think maybe the baby was like too close to me, because then they all of a sudden got really angry and they started throwing.
Did you find gestures in common between Las Pumas and Puerto Rico?
There were a few. The most common was definitely the grooming gesture, present body, and object shake. Those were the most common. And a couple of the embrace ones were also in both populations, but I noticed like 5 new ones in Las Pumas.
Did you find monkeys every time you went out looking for them, or what was your success rate?
No, that is not good. I went out at least like 12 of the days in Coyhaique, and I only saw them on 4 of them. So I saw howler monkeys every day, but I did not see those spider monkeys.
Did you notice at all that in some cases, if there was another individual who was not the target, but sort of intercepted a communication signal, that the sender would sort of resend the signal by moving or making sure that the proper recipient actually received it? And that's the first question. The second is, why spider monkeys rather than like Cebus or howlers?
The first question, I noticed that in Las Pumas, there was one instance where I remember one of the monkeys seemed like it wanted to get groomed, and it kind of like laid out its body to get groomed, but its receivers were just turned around, and so it kind of went around to go in front of it, and then it did it. So yes, I did observe that in Las Pumas, and then I wanted to do spider monkeys because of that one paper that I found. I couldn't find any evidence that there's been gestural communication in capuchins or in howler monkeys, so I just felt like focusing on spider monkeys would be the best way to go.
Any other question? Thank you, Marisa. So, if you notice, I asked with a gesture to Frank if we keep going, and he said with a gesture, yes. So another gesture that I saw was Emilia sleeping, so I just opened the door for making more. Usually when you feel sick, you want to see the doctor, or maybe better asking Eladio. But next time I would like to find out what a monkey is telling me to take. So next speaker from UC Los Angeles. Wait. This is not one. Next speaker, Miles Fisher from UC Los Angeles. He will speak about so far macrognosis in white-faced capuchin monkeys.
Hi, I'm Miles, and I'm going to be talking about zoopharmacognosy in white-faced capuchins, a Cebus imitator. So what does zoopharmacognosy mean, you might ask? It comes from the Greek, and it means animal medicine knowledge. And you might have seen some examples of this in your own life. Have you ever seen a dog eating grass? That's to soothe their stomach, and that's an example. Other examples are elephants actually will treat their wounds with olive flowers. And in primates, previous studies have been done to show fur rubbing with millipedes is used as an insect repellent, and they will chew the bitter pith of certain fruits to remove parasites, and they actually will change their diets in response to injuries or traumatic events. And so a little bit about capuchins. They're found in Central America, distributed from Nicaragua to Panama. They're omnivores, and they're considered to be the most intelligent of all the New World monkeys, which is important in advanced cognitive behaviors like zoopharmacognosy. So my central question was, how do white-faced capuchins self-medicate in the Monteverde region of Costa Rica? So what I did, first of all, this study took place from the 29th of October to the 20th of November. And first, I identified sites of high capuchin activity, including some reserves, Reserva Bajo del Tigre, Reserva Curicancha, and Reserva Conmemorativa Rachel and White Crandall. And I spoke with guides and staff there to identify sites of high capuchin activity, and I set up camera traps there. And every day I would go starting at 9 a.m. And go walk around the trails, ask the guides if they'd seen monkeys, communicate with other students. Because of the weather, it wasn't always possible, but when I did find capuchins, I would follow them, taking notes on their description of the individuals and their behavior, and I marked all of the plants that they ate with flagging tape, and I returned a few days later with Eladio Cruz, who helped me to identify them. So I found 14 different species that the capuchins ate, 13 species of plants, and 1 wasp. These were taken on the 29th of October, the 15th of November, and the 20th of November. And these behaviors can be broken down into 4 general categories: fruit eating, live leaf eating, dead leaf eating, and other behaviors. So first of all, fruit eating is not necessarily the most relevant behavior to zoopharmacognosy, as fruit does comprise like a really big part of capuchins' diets. It's rich in sugar, dietary fiber, and a lot of nutrients. But these do have medicinal properties. Not to say that's why the capuchins were eating them, but they've been used extensively in indigenous culture as stomach pain relief, relief for diabetes, diarrhea, and wound care, and that's due to a large range of organic compounds. And similarly, live leaves also just comprise a big part of their diets, again, really high in fiber and nutrients, so these aren't necessarily relevant, but they do, some of them do have medicinal use, such as cestrum, which has a lot of saponins, lignans, and alkaloids, and has been used to treat fever, inflammation, and as an antimicrobial. And so getting into the more relevant findings, I observed capuchins eating a lot of dead leaves. And quick disclaimer, it is common for capuchins to forage through dead leaves looking for insects and larvae, but from my observations it appeared that they were actually eating the dead leaves themselves, but further observation would be required to confirm this. But the 2 of the first plants I observed were Yucca gigantea and Drosenia fragrans, which are both, especially yucca, very culturally relevant in a lot of indigenous cultures, being used to treat a whole range of ailments, from joint pain, bleeding, inflammations, dandruff, even gonorrhea, and used as a laxative, similarly to Drosenia, which has steroidal saponins and linalool, which is a terpene that can have relaxant and calming effects. It can even be used to treat HIV. And the third plant in the dead leaf category was Cecropia obtusifolia, commonly known as guaramo. And again, this is very extensively used in indigenous cultures, and importantly, it is actually made into a sedative tea in El Salvador, which leads well into my next plant, which is, in my opinion, the most interesting finding in my study. I observed, as you can see in these pictures, an adult male capuchin break the stem of an Iochroma arborescens plant and actually eat the structural tissue of the stem, which is primarily made of cellulose, which capuchins can't digest. And so it's really unlikely that the capuchin would have gotten any nutritional value from this plant, meaning there was likely some other motivation for eating this plant. And looking into it, this plant's actually used in Brazil as a narcotic, and it's thought to be psychoactive, which is really interesting because if in fact the capuchin is receiving some sort of psychoactive effect from this plant, it could lead to insights into addiction, how the capuchin brain works, and possible more research needs to be done there for sure. But it also has a wealth of other uses, treating colic, liver infections, and is anti-tumor and antifungal as well. And so I observed 4 other behaviors that couldn't really be categorized as well. First one was bark eating in Yuglins, which is a member of the walnut family. Again, used to treat a whole range of ailments, and this also would not have much nutritional value for the capuchins, so very interesting trying to figure out the motivations there. And similarly, a species in the genus Tenatia of flower, which does not have any documented medical use, but further research is required. And the final 2 behaviors were stick breaking and biting in Montanoa guatemalensis, which was very interesting to observe. The capuchins would just simply bite the stick and break it. They wouldn't swallow, they wouldn't chew, they wouldn't ingest it in any way. And a few hypotheses for this could be trying to treat some sort of dental issue, some oral problems, or simply out of boredom. Not really sure. And finally, adult wasp eating. It's really common for monkeys to raid the nests of wasps in search of the larvae, which is really high in protein. But to see them actually eat the adult wasps, which are not high in protein at all, is really interesting. And I spoke with locals and staff about this, and it wasn't a behavior they'd actually ever seen before, which is really interesting. The only literature on this subject was a study saying that in Mexico these wasps are actually boiled and can be used to cure the scares and to help with menopause. So again, no real conclusions can be drawn from that, but promise for future studies. And so what I found, the vast majority of the species had significant medicinal value. But this leads to this really difficult question of how do we determine the intention behind? Is this purely dietary? Is this something they're aware of? They know the medicinal properties of these plants somehow, or could it be both? And I ran into some big limitations in terms of trying to figure this out. First of all, the weather. We got hit by Tropical Storm Sarah during the observation period, which greatly reduced the activity rate of the capuchins. So it was really hard to see them. I only actually found them for 2 days out of the entire period. And also just the time. A lot of studies on zoopharmacognosy are about analyzing long-term changes in dietary patterns, so that wasn't really possible. So I had to do my best to find ways of finding the motivations. But I think the 3 future most promising findings I had were the idea of sedative plants, including Icrom arborescens, Obtusifolia, Yersinia fragrans, and Cestrum, all of which having sedative properties could lead to some insights on the desire of capuchins to actually alter their mental state, which is really fascinating to me. And then the wasp eating and the stick biting, also not behaviors that have been observed or documented much, that have a lot of potential for future research. And so why is this important? Actually 25% of all 25% of all prescription drugs in the world are derived from plants, and over 60% of anti-tumor and antifungal drugs are derived from plants as well. So there's a vast amount of knowledge that these plants can give us, especially as antibiotic resistance is becoming a more pressing issue and cancer rates are on the rise. These plants have a lot to teach us, and looking to animals who have a really intrinsic knowledge of nature is, in my opinion, a great place to work. So some acknowledgements, huge shout out to all the UCEAP and the NBI staff, Frank, Naomi, Emmy for just giving us a great experience, being so supportive, Fede for so many hours spent driving me around, guiding me, and just being a hilarious and amazing person, Eladio for helping me identify these plants. These wouldn't have been possible without him, McKenna for spending hours trekking through the rain with me, and the people at Reservas Curicancha and Bajo del Tigre, and of course my homestay family, the Fonseca Matas.
Time for questions.
Did you only find them chewing on sticks of a specific type of plant? And it was you had multiple. Yeah. So that's really good. Yeah. Not just a moral fixation, but
that's what I would assume.
Did you observe this sending behavior multiple times?
No, it was only once. That's, I think, something that really I would like to pursue in terms of frequency of visitation. Seeing if they go back to that plant would be really critical into getting more insight out of this.
If you had to eat one of the plants before you
I gotta be Icrom arborescens.
If you were to do like another study with more resources and time, how would you try to distinguish between intentionality?
I think looking for patterns in dietary changes. A big thing was in other studies was identifying injured individuals and then seeing their diets before and after that's like an injury occurred, and I wasn't really able to do that as well. And then more nutritional analysis, going on a deeper dive into the chemistry behind these plants, seeing if they're actually getting any sort of nutrition out of them. And more use of camera traps.
Other than wasps, did you see them eating like any other kind of like live thing?
They would forage through bromeliads for insect larvae. I didn't include that here, but yes.
Piggybacking on Maurice's study, did you like observe them doing any behaviors that like have potential to maybe be gestures with future study? Or?
There was a lot of vocalizations that was really interesting. Gestural, I didn't really see as much, but a lot of vocalizations that was really intriguing.
Were you able to find any studies about like how the plants affect like were there any previous?
There was basically no literature on this. I did find a few studies that actually will eat toxic millipedes in, not capuchins, but other monkeys will in certain parts of Africa that supposedly gets them high, and they're also known to steal alcoholic drinks in the Caribbean. It is evidence for them trying to alter their mental state.
Gracias, Miles. So next time that you go to Baramigos, don't trust in the white-faced capuchin. When you listen Miles saying that it was feasible to follow and see the monkeys a couple times, it's because we had the rainiest November that Eladio can remember in his life, so it was very rainy. So when you guys were enjoying the volcano view or having fun on the beach, we were under the rain. And next speaker can state that regardless the weather, she can be full with mammals. So McKenna Sandwich from UC Santa Barbara is going to speak about interactions among mammals. Excuse me.
Alright, my name is McKenna Sandwich, and I investigated interspecific interactions between mammals in Monteverde, Costa Rica. I looked at interactions both between wild mammal species and between wild and domestic mammals. So as a preview, looking at the list of species that I saw, these first four are the most relevant to my study. So looking at those, the white-faced capuchins are extremely social creatures. They are omnivorous, with their diet mostly consisting of insects, small invertebrates, as well as fruits and occasionally small vertebrates. White-nosed coatis are also very social, and they're also omnivorous. Their diet is as well dominated by insects and small invertebrates, but they are opportunistic foragers, so they'll eat whatever they can find. A note on the capuchins, they're extractive foragers, which means they're picky. They kind of pick and choose what they want to eat because they have, as arboreal foragers, access to a lot of different food sources. Agoutis are very solitary animals. They have very well-defined home ranges, and so they have specific foraging sites that they tend to revisit frequently. Peccaries are also very social, and their diet consists mostly of plant matter of all different kinds, but they do also eat some insects and small invertebrates as well. Other species that I encountered during this investigation were squirrels of a variety of different species, an alingo and a kinkajou, a spiny pocket mouse, and then of course domestic mammals, so dogs and cats. The study sites that I went to in looking for these interactions began with foraging sites, as most past research revolves around foraging behaviors, especially those that are catalyzed by capuchins, where capuchins will be foraging in a tree, they'll drop fruit or other plant matter, and then other animals will go and forage off of that. I wanted to look a little bit beyond just foraging behaviors, but I knew that that would be the easiest place to start. So I went to natural foraging sites, such as fruiting trees, particularly fruiting guayaba trees, as well as what I called artificial foraging sites. So these would be sites where humans provided food for these animals regularly, being local businesses or residences where the residents would directly feed the animals, or where compost or other trash provided a site that animals knew that they could go back to. I also went to some national reserves and did some research around the research stations we stayed at in order to look for that more expanded range of interactions. So I visited the Cuicancha Reserve, the Bajo del Tigre Reserve, and the Cradle Reserve, all of which are very nearby the Institute. And I did research while we were at the Estación Biológica Monteverde and the Estación Biológica San Gerardo, so that I could do more passive observation rather than directly visiting a foraging site. I kind of just kept my eyes open, and I was looking for any of these mammals or interactions throughout this entire period of time. This is a look at the weather trends. As we were just saying, this has been an incredibly rainy November. This November, there were 964 millimeters of precipitation. The mean rainfall in November in Monteverde is 200 millimeters. So you can tell this was extraordinary. This lime green bar at the bottom shows our direct observation period. You can see Tropical Storm Sarah hit directly during that period, and immediately before there was a period of extreme rainfall as well. There's a trend in Monteverde in which there are fewer rainy days every year, but each rainy day is getting rainier and rainier, which means that there are extended dry periods which can cause a risk of starvation for mammal species, as well as these extreme weather events, which put a lot of pressure on them in terms of starvation and not having adequate shelter as well. That's me on the rainiest day, still going out to look for mammals, even though Federico told me it was useless. So my two big research questions were what types of interactions occur between wild mammals, and how does the presence of human, and particularly the introduction of domestic pets, affect mammal behavior? I did have a third research question, which was investigating the way that rainfall directly affects mammal behavior. For the purposes of the presentation, I did not include that. So for my methods, our observation period was from the 10th of November to the 24th of November, though I did have two outlying observation days, the 29th of October and the 4th of November, which we will get into. I did both direct observations through doing field walks every day, and as I said, just being constantly observing, as well as doing indirect observations through the usage of camera traps, of which I had two at the Estación Biológica Monteverde, one at a fruiting guayaba tree and one at a corner where we regularly saw coatis every morning. And I additionally used videos provided by my classmates when they encountered mammal interactions when I was not around. To define interactions, I use spatial and temporal proximity, basically saying if individuals of two or more species were in the same place in under 5 minutes, I would classify that as an interaction. My interactions fell under four main categories: neutralisms, amensalisms, commensalisms, and predation. Within the neutralisms, I saw sharing behavior, which would be individuals of two or more species foraging or sharing the same resource without displaying fear or aggression towards one another. Ignoring behavior, which would be when they were coexisting without having any sort of reaction to one another. And waiting, which would be when individuals of one species stood on the outside of an area until individuals of another species left and then proceeded to go and use that resource. The amensalisms I saw, I'm going to move forward just so you can see the distribution here. The amensalisms I saw were competition, not directly for food, but for space. And you can see 10 out of the 15 interactions I observed were all neutralisms, 3 were amensalisms, and then I had 2 outlying observations, 1 instance of commensalism and 1 instance of predation, which I will get into in a minute. Out of the neutral interactions, you can see that the majority of them, 9 out of those 10, involved agoutis, which are very non-confrontational animals. These 7 out of these 10 were observed via camera traps, because agoutis will run away the second that they see me. So most of these happened just via what I was seeing. There was some sharing behavior, but mostly they tended to ignore other animals. The amensalisms I saw were interesting because it was an agouti scaring a squirrel out of a tree, despite the fact that squirrels are arboreal foragers and agoutis are terrestrial foragers, so there was no direct resource competition, yet the squirrels still left and did not return to the area. Looking at the commensalism, this is an interaction that Miles actually just mentioned, and it's this picture here on the left. This is a day when capuchins came to the Estación Biológica Monteverde one morning and went to the areas where wasps were in diapause and began to knock them down. They were eating adult wasps, and the coatis on the ground were feasting on these adult wasps. There was no effect on the capuchins as a result of this behavior, but the coatis had a great benefit of this, and as Miles mentioned, this is an unobserved behavior. This was a very rare and unique occurrence, so it'd be something very interesting to try and observe in the future. This photo on the right over here is the one instance of predation that I saw. This was on the 20th of November, and what I witnessed was 3 juvenile capuchins chasing after a rat. They were competing for each other over it, pulling it out of each other's hands. One eventually won, ran up into a tree, and started eating it while it was still alive. While this is an example of predation, this was seen at an artificial foraging site. This was a place where capuchins already had access to plentiful resources, so this was not predation out of necessity. This was a supplement to their diet, and likely also a social behavior because of the hunting that they were doing as a team. Then, looking at the interactions between wild and domestic mammals, I had 6 of them. 3 were neutral and 3 were amensalistic. So the neutralisms that I witnessed here, these were all with capuchins and all at artificial foraging sites. So there was absolutely no variety in terms of what those interactions were. The neutralisms here, one of which a dog and a capuchin were nearby each other, neither could, they were there. And then when I say no reaction or with reaction here, by reaction I mean that one of the individuals fled the area as a result of this interaction. These defensive reactions that I witnessed by the dogs were mainly them barking in terms of pack protection behavior, essentially. And with the cats pursued these capuchins up a tree or followed them intensely. And here you can see on the left, this unfortunate kitty got punched out of a tree by the capuchins that it followed. I mean, that literally it fell out of the tree as a result of physical aggression by the capuchins. And then in the other instance between a cat and a capuchin, the capuchins got scared away. And then on the right, here's a dog who was barking obnoxiously at the capuchins, and the capuchins just did not. In terms of, I don't know why that got tiny. In terms of effects of human presence, well, there are supposed to be 4 bullet points on this slide. There is the big effect of resource displacement, which is that within the formation of these artificial foraging sites, this creates a reliance on humans by these mammals that otherwise wouldn't exist. While this means that these are artificial foraging sites where weather isn't going to affect the potential access to resources, which could be very helpful in terms of these increasing extreme weather events that I was talking about, it also means that these mammals are going to become reliant on human behavior and on human habits that might not be reliable and may result in these animals not knowing how to hunt or forage naturally if for some reason this artificial foraging site is taken away. Additionally, adding domestic animals to these ecosystems disrupts the predator-prey dynamics of the natural ecological communities because particularly dogs are very good, very competitive predators due to their diurnal and nocturnal nature, their pack behavior, and their high population density, coupled with their extreme tolerance to humans. Because of this, they can outcompete predators who are naturally occurring here very easily. And they also can pass diseases on to native mammal populations that otherwise would not exist, that these populations have no resistance to. One other effect of human presence is that humans act as a constant predator stimulus, creating vigilance behavior and increased stress in these mammals. As a result of this constant predator stimulus, small prey species can become habituated to human presence, and as a result, not react properly to actual predatory threats, decreasing their ability to defend themselves. Also, the expansion of urban areas, along with climate-force shifts in habitat, means that these animals overall have a decreased territory and an increased overlap and interaction with humans and domestic animals, which is causing changes in both the spatial and the temporal areas and habitats of these animals. In the future, it would be awesome to do longer studies, ideally in periods of less rainfall, and ideally with more investigation into artificial foraging sites, because I couldn't find any research done on that in particular, especially between animals that aren't monkeys. I would like to thank Federico Chinchilla for answering all of his answers, or all of my infinite questions, and him and Naomi as well for telling me you do not need to go out when it is pouring rain, it's not going to be worth it. And Frank Joyce for making all of this possible, to the Estación Biológica Monteverde and the Monteverde Institute for providing a space to work, to Miles for coming out in the rain with me all the time and texting me whenever he found monkeys, to Annie, Cassie, Dre, and Miguel for providing me with videos that were extremely valuable extra data points, and of course to the critters for letting me follow them.
We're still having time for questions?
Yeah.
Did you do the stress ball with the capuchin?
Nope. Didn't hear it.
So with mixed species flocks of birds, they actually tend to be organized, and they're birds that call and sort of get the flock going. Do you see any whiff of like some mammals sort of calling the fauna together, or any signaling between species such that the group, not the flock, but the group of mammals would somehow act like a mixed species flock?
No, I didn't actually see any interactions between groups of mammals. The only interactions that I saw between species were either between two individuals or a group and a individual. So there weren't any cases in which that really occurred. And because so many of the interactions were neutral, the interactions themselves were confined to the period that I observed and didn't really continue past or have any additional behaviors associated with that.
Bueno, gracias. Kenna. The next interactions among mammals is going to happen now, having a break. Of how many minutes, Frank? Yeah, 10 minutes break. Thank you.
It's a crazy video. Last
I'm not next.
Y ya empieza a transmitir.
is increasing in interesting presentation. And you may be thinking, wow, the last section, the fourth one, was the most interesting, but wait for this one. This is the fifth session, Biology of Birds and Bats. And first speaker is Melina Garcia from UCEAP, speaking about the correlation between flowers, death, beetle death, and hummingbirds.
Okay, hi everyone, my name is Melina, and my topic is about the death of flowers, buildings of hummingbirds, and general hummingbird feeding behavior. So essentially my question came from a set of pictures. So basically what you're looking at here is a hummingbird whose beak is like fully immersed in this long tubular flower versus a hummingbird who is feeding from this more sort of shallow, smaller flower. And the reason that this like piqued my interest is because there's a long history of like coevolution between hummingbirds, certain species of hummingbirds and certain species of flowers. And so my hypothesis was that hummingbirds with longer beaks would be feeding from flowers that are like longer, sort of like this one, and like tubular shaped. And in order to test my hypothesis, I performed an observational study, and these are the species that were involved. It was the green Amphibian, the green hermit, rufous-tailed, striped-tailed, and violet saberwing. And these are the flowers. So this one is absolutely everywhere. You can see it on your way to the Institute, from the Institute, like up through the Institute. This one is found sort of like up by the Belmar, on your way to the TV towers. And these other two species here were from my homestay's garden. So for methods, you can't really see it too well here, but there's a mist net. This is the pole. There's like very thin little lines where you can sort of see the mesh. And there's kind of two components to this study. The first part was obviously getting the observations, and the second part was taking measurements. So to get bird bill measurements, we had to set up these mist nets. And this is a little tough because of the weather. The mist netting can be dangerous for the birds because they can't thermoregulate. So we have to wait for nice conditions outside. And it's also a little dangerous to like take them out of the net. So you need to be like trained and experienced in doing this. And the easiest and safest way to do this is, well, this is a picture of like sort of what it looks like when a bird gets caught. I believe this was a woodpecker of sorts. But yeah, but it's best to leave it to professionals to do the Thank you, Wendy, for the picture of it. And then for methods, essentially we would get the birds out of the net and we'd use a caliper, which has like this little bit here. It like moves down, and you can like adjust it to the measurement of what you want to get the measurement of. It's just like a fancy ruler. It looks a little bit like that, if I have to describe it. Then for the flowers, it was very similar. So there's like this bit on the end that comes out, and it's the same measurement as this here. So I would basically insert this end here into the flower to get the depth of the flower. And that looks a little bit like that. So for my results, I found that these were the average bill lengths by species. So clearly the green hermit has the longest bill by far. And actually if you go back to the pictures of the birds, you can see that they have like a little bit of a different shape too, where like these two are more curved and these three are straight. So these were the results. These three were pretty similar, and the two with the curved beaks were a little bit longer. And these are the averages per species. For the flowers, I did a similar graph. The flower species is listed here in the legend, and the depth is up here in millimeters. Those are the numbers for that. These two species were pretty similar in length, and those were kind of the longer, more tubular shaped flowers, whereas this one, the Stachytarpeta, which is one you can see everywhere around here, is a lot shorter. And that's interesting for these findings because this scatter plot shows bill length versus flower depth, and these points are colored again by flower species. And like some interesting things to note about this graph is here in purple you can see that there's like a wide range of bill lengths that we're visiting this flower. On average, I'd say it was around like 16 millimeters, but you can see that there's like birds with almost 40 millimeter beaks visiting the flower. And that didn't surprise me because it's like kind of logical that a longer beak can feed or like can reach nectar in a shallower flower. What was surprising about these findings were these up here, because these flowers, the Strobilantes crucia and the Thunbergia erecta, are a lot longer. I would say like maybe over an inch or so. But the birds that were feeding from these flowers had like roughly 20 millimeter beaks, which is really interesting to me. And it, yeah. Aside from that, the entire graph is kind of a mess. And it did answer the question, the main question that I had, which was whether there was a correlation between the flower depth and hummingbird building here in Monteverde. The answer was no. As you can see, the R-squared value was 0.01, whereas 1 would be like a strong correlation and 0 is no correlation. There's like very, very strongly no correlation. However, I was lucky enough to see something that might explain these points up here when I went to the Hummingbird Gallery. So if you see here, I have a little video. This is a green-crowned brilliant male, and hopefully it's plain. You can see at the end he extends his tongue, and if we pause it, like right around there, you can see that it's like probably, I would estimate, more than double the length of the beak, which would explain this here. I would say most of these were green crown brilliants. And yeah, so I felt very fortunate to have gotten this video because it took away a lot of my doubts about the data that I was getting. For future studies, if this were to be repeated, I would say that it should be repeated at a different time of year, maybe when it's not so rainy. Main reason for the safety of the birds when mist netting, and also so that there's more potential for different flower species to be in bloom, because right now, overwhelmingly, the main flower species available is the Stachytarpeta, the little purple flower. And I think that most of the birds are going to that flower because of its abundance, and they're kind of acting like opportunists. Another thing that I would have wanted to test is the nectar content, the sugar content of the nectar found in these flower species, because maybe the nectar is sweeter in the Stachytarpeta, or just they might prefer sweeter nectar, less sweet nectar. And finally, for acknowledgements, I want to say thank you to my two advisors, Federico and Natalie, the Galeria Colibri, the MOSI project, and everyone involved, Luisa, Moreno, Diego Rodriguez, Victorino, no sé su apellido, Olivia, thank you for waking up with me all those early mornings, entertaining me and helping me cross-reference the data, and my homestay family, Salazar Ugalde. Thank you for letting me collect some of my data in your garden and for welcoming me into your home.
questions, please.
Melly, I have a slightly different interpretation of your data. And maybe you actually already said this or think this, but the long-billed birds can access short corolla flowers basically based on behavior. So if they can hover and manage to get their tongue into the flower, the little guys, if they want to go for a longer corolla, some of them make a little hole at the base of the corolla and rob the nectar. So I hope, in my view, that graph that you showed is not a mess at all. It shows your data, it shows what you found. And I'd be happy to explore other interpretations. You may have already suggested them, or maybe your other advisors say, no, that's not what's going on. But to me, that's not a mess.
Okay, thank you. Yes.
Is there, can you say anything about the bill length and the age of the bird? Do they start short and they get longer as they mature?
I'm not sure about age. What I do know is that these three species here have different morphology based on gender, and actually the green-crowned brilliant has different coloring for the juveniles as well. So that could potentially influence the length of the bill. I'm not sure if we could do that based on age.
Did you see any of the hummingbirds stealing nectar from a hole in the base of a flower?
I didn't.
No. Okay. Gracias. Iván, can I disconnect you? Thank you, Liwen, for your help and to let us using your computer. Next speaker from UC San Diego, Queen Gussie, is going to speak about similarities between bird vocalization in the context of their phylogenetic histories. Thank you, Queen.
Howdy y'all, I'm Quinn. I'm going to be talking about the similarities between bird vocalizations in the context of phylogenetic relation. So bioacoustics in general is a very up-and-coming area of study, and I'm all about filling gaps in knowledge, so this was really interesting to me. And in the case of birds in particular, there are a lot of social interactions that can be better understood by better understanding the specific origins and mechanisms of their calls. This includes like alarm calls toward predators, mating calls, calls to find their chicks, etc. Pretty much all forms of all areas of social interaction. Also understanding these calls and the exact mechanisms of them could help us understand the origin of them. like whether they're more learned or more innate. And if we are going to go the innate route, like given the genetic basis of a lot of innate behaviors, one could argue that if you have a closer genetic relation, you should potentially have a more similar call if even a little bit of that call is based in innate behavior. And that leads me to my question: is a closer relation correlated to a more similar call? So the methodology for this was pretty simple. I just selected 4 species based on availability and relation. I selected the Crocodile Reserve as my research site because of how well kept the environment was, and also because all of my species were there. To record the calls, I used just a recording device, an external microphone, and after returning from the field, I used Raven Pro just to measure all of the call aspects that I wanted to record. I analyzed everything with a NOVA test, 2-piece HSD test, and also linear regression analysis later on. And then for each of these values, I just interpreted them for statistical significance in whatever scenario I was in. So these were the 4 species that I settled on: the white-breasted woodwren, gray-breasted woodwren, rufous and white wren, and the white-throated thrush. So all of these species, their relations to each other can be described by their taxonomy or classifications, and this takes into account phylogenetic relations, so basically what species they're derived from, and how closely those species are related. So the white-breasted woodwren and the gray-breasted woodwren are both in the genus Cancrena. And then the rufous and white wren is not in that genus, but all 3 of these are in the family Troglodytidae, which is the wren family. And then the white-throated thrush is in the Turdidae family, so not the same one, but they're all in the same order, Passeriformis, which is songbirds, which is another reason that I chose these, because songbird vocalizations are particularly interesting. And the first thing that I wanted to show for results was just, I wanted to give you guys a general idea of what I would be looking at. So these are spectrograms for the calls, basically just a visualization of what I was hearing. On the x-axis, you can see just seconds, and then on the y-axis is kilohertz. And these are labeled with the names of the species: white-breasted woodwren, gray-breasted, rufous and white wren, and white-throated thrush. And the little blue boxes are just the boxes I drew to have Raven Pro measure the peak frequencies and everything that I wanted to record. So that's just a quick visualization. And as for the actual data that I got from these calls, the first call aspect I wanted to record was call duration. So here you can see on the x-axis the Latin names for all of the species, and this correlates to just white-breasted woodwren, gray-breasted woodwren, the rufous and white wren, and then the thrush. And then on the y-axis, you would call duration in seconds. And this is just a box and whisker plot, and I just put the dots next to it for, I guess, more information. And the n values are just how many calls I measured. So as for interpreting these results, the first thing you probably are going to notice is just visually, the 2 in the same genus are the most similar. While the one that's just in the same family is a little bit different, and the one that's in a different family but same order is a lot different. And this is corroborated by the statistical values of the 2-piece HSD test. Every single pairwise comparison is very statistically significant. All of them are very different, except for the 2 in the Cancrena genus, the 2 woodwrens. So those have no statistical difference between them. For all purposes, their call duration is essentially the same. And then the second aspect of pulse that I measured was the pulse interval. And this also has the same x-axis with the species and in values. And this time the y-axis is pulse interval in seconds. You'll see kind of a similar trend with this one, except instead of the 2 species in the same genus having a more similar value, it's actually Hinocarina leucosticta and Trilophyllus rufalus, which is the rufous and white wren and the white-breasted woodwren, respectively. So again, every single one of the pairs that could be compared were statistically significant, except for one pair, which was white-breasted wood wren and rufous and white wren did not have a statistical difference between their pulse intervals. The last call aspect that I measured was peak frequency. So again, the x-axis is the same with species and in values and peak frequency. This time is in hertz. You'll probably immediately notice that there isn't really a general trend with this one. There really isn't a lot of statistical significance compared to the other two for peak frequency. In fact, none of the possible pair combinations we compared had any statistical significance except for T. rufalvis and Hinocarina leucophrys, which is the gray-breasted wood wren and rufous and white wren. And then this pair, the Trilophyllus rufalvis and Turtus similis, are actually the only pair here that have a very statistically significant difference between them. And then to kind of combine all of this data into a more easily understandable format, the thing that I did was I took all of those significance values and I ranked them 1 through 6 for each of the 6 pairs or possible pair combinations that there were. And then I averaged those rankings per level of relation. So does it differentiate at the species level, does it differentiate at the genus level, or does it differentiate at the family level? And with these averages, you kind of get similarity scores, is what I dubbed it. So basically, the trend is pretty obvious, I feel like, in that you see these descending values, which it means more significant because 1 is the most significant ranking and 6 being the least. The descending values and the lowest values are the values with the farthest relation between them. So if you were to put these in, say, linear regression analysis, which I did, you come up with a correlation coefficient of 0.999, which is a very strong correlation. I do want to note that giving them rankings like this, 1 through 6, whereas you did have, decimal points with significant figures, very much erases some of the data. That is a limitation of this particular test, but given that I had the other tests, I decided that this was a good way to kind of get a condensed, overall view of things, just for the purpose of better understanding it. So basically what that value means is that not only is there very strong correlation, about 99.9% of the variance in the similarity scores can be explained by the different levels of relation. And to conclude with that data, is a closer relation between these birds correlated to a more similar call? Yes, very much so. 99.9% actually. At least for these 4 birds, there is a very, very strong correlation. And as for why this could be, the kind of 2 main possibilities are, 1, this closer genetic relation kind of resulted in a more similar phenotype or morphological similarity between the birds. So it could be that their physical syrinx is shaped the same way, so they make the same sounds. Syrinx is basically their equivalent to vocal cords. Or, as previously mentioned, it could be entirely psychological innate behavior. And a good future study for this would be to determine which of the 2 that is, or to just have a study that is taking into account a much broader range of species to try and discover or support current trends. And these are my acknowledgements. Thank you very much for your time.
Thank you. Now we have time for questions.
Will.
Did you incorporate, some birds will have different calls, like a social call or a song or whatever. How would you, do you only use one type of call for each bird?
Yeah, I used what was, I guess, defined at least on Xenocanto and Merlin as generic calls. So if it was an alarm call, I would not use it. And I think there was one other that was, I forget what it was classified as a particular type of call and I didn't use that one either. So I only used just generic calls. Yes?
How did you decide to look at call duration, pulse interval, and peak frequency? And what
actually wrote about that in my paper. Basically, it was consistency of the variable and ease of reporting. So for instance, I couldn't report intensity because that would depend on how close I was to the bird. But things like peak frequency as opposed to highest frequency and lowest frequency is more reliable because peak frequency is kind of more where the majority of the sound lies, as opposed to highest and lowest depends on where you physically draw the box, which introduces some human error. And that also, highest minus lowest is bandwidth. That's another thing you could look at, but again, human error. So the ones that I looked at were just kind of trying my best to get rid of the human error aspect, as well as to not have to physically catch birds.
Gracias, Queen. Okay. Next speaker, Olivia Turner. It's another person that wake up very early every day from UC Santa Barbara. She will speak about if you start feeling itchy, it's no coincidence, right?
Oh, there you go.
Welcome.
Thank you. My name is Olivia Turner, and I'm going to be talking about the morphological and life history traits that influence feather mite composition and abundance in the avifauna of Monteverde, Costa Rica. So birds host a wide variety of ectosymbionts in their feathers and skin, some of which you may know, like ticks and lice. And another type are these feather mites, which live in the barbules or in the shaft of the feathers, and they feed on gland oil, fungi, pollen, just debris that's in the feathers. No avian order is free from a feather mite association. So from penguins to pelicans, they all have feather mites. And given this prevalence though, there's very little research on the biological and ecological factors that play in the abundance and composition of these birds. So there's lots of differences between these two birds. For example, the one on the left has a longer wing, it looks a little chunkier, has a bigger bill, and it also has, you can't see it, but it has a bill overhang. And there's also factors that you can't see that are related to their life history. So for example, this ovenbird is a neotropical migrant, while this olive-streaked flycatcher is a resident species here in Costa Rica. They also have different foraging strategies, so they eat different things, and they forage at different levels in canopy. So I'm going to be looking at how all of these differences relate to their feather mite abundance and composition. So my first hypothesis is that bird morphology is going to impact the ability of a bird to house feather mites. So for example, a bird's body weight and its wing length, as that increases, you're going to get, I hypothesize there's going to be more feather mites because there's just more free real estate for the feather mites, as well as more resources. I also predict other morphological features involved with the bill are going to influence the ability of a bird to defend against feather mites or ectoparasites. So the presence of a bill overhang will allow them to preen better along the beak. So that's going to decrease their feather mite ability. I also predict that life history traits, such as just their family or taxonomy, their foraging patterns, and where they are in the canopy, as well as their migratory status, is going to impact what kind of parasites we see, and the amount due to contact rate transmission. So, for example, a bird on the floor is digging through leaf litter, so it's going to have more rates of transmission. And another factor that I predict is going to impact the load or abundance of mites is molt. So when a bird molts, it drops some feathers, and I predict that the newer feathers are going to have fewer parasites just due to time. I also said that composition, or the type of feather mites you find, are going to be different between migrants and resident species. So to do this, I set up a series of mist nets to help me catch the birds. They're pretty much invisible nets that birds just kind of fly into. And then once caught, I took morphological measurements of the wing, the weight, and the bill length. And then I looked for ectoparasites. And to do this, you pretty much take the bird, you hold it up to the sunlight, and there'll be a bunch of little dots if it has ectosymbionts or feather mites. And then, according to these dots, I give it a score. So for each feather, I got a score from 0 to 3, 3 having the most, 0 being none. And then I counted the score for each of the 10 primaries and added that all up to get the total ectoparasite abundance. Then, if a bird had parasites or ectosymbionts, I would pull an unharmful feather to later identify what this was using a compound microscope in the lab. So in total, we caught a total of 72 birds from 36 different species and 10 families. And they were all in the orders Passeriformes, or songbirds, and Apodiformes. The only ones in Apodiformes we caught were hummingbirds. And of the 72, 10 were migrants, and of those 10 migrants, 9 of them had ectosymbionts. And of the 62 residents, 44 had ectosymbionts. So 75% of the total birds I collected had these ectosymbionts. And the only ectosymbionts I found were feather mites. And they were all in the family Proctophyllodidae. And if I was able to further identify the subfamily, there were some in subfamily Pterodactylae and Proctophyllodidae. So I found that, so on the x-axis we have wing chord and weight, and on the y-axis we have ectoparasite score. So I found that both of these morphological factors significantly and positively related to ectoparasite score, which aligned with my hypothesis that just like the bigger you are, the more free real estate there is, the more resources there are, so the better you are at housing these these feather mites. I, however, did not find significance with bill morphology, which was interesting because other studies looking at ectoparasites like ticks and lice found significance that if a bird had a bill overhang, they had fewer ectoparasites because they're able to preen it off better. But this could be due, in a similar study, Bonawada 2022, found that when we're talking about feather mites, the bill overhang does not have significance. And this could help us learn about the relationship between feather mites and birds. So it may be more of a commensal or mutualistic rather than a parasitic relationship, because birds are not creating adaptations to actively preen off these feather mites. I also found a significant difference in canopy level, which aligns with my hypothesis that the more you're in contact with them, the more you're going to get them. So the floor birds had significantly more ectoparasites than the endosteric birds. There's also significance in some families. So Furnaridae family includes like ovenbirds and wood creepers. And so these birds are like insect following, they're always in the leaf litter, they're playing with bugs. And so they were found to have significantly more feather mites than Trochilidae, which are hummingbirds, and Tyrannidae, which are flycatchers. And so this also aligns with my hypothesis that contact rate is going to affect ectoparasite abundance. There's no significance in foraging behavior. This is likely just due to the subjective nature of its categories. So a lot of insectivores are also omnivores. There was also no significance with migratory status and molt stage. Again, this could be due to sample size. There's not a lot of birds molting during this season, so future studies should look at seasons in like the dry season when birds are actively molting, so we can compare those loads. There was no significant difference in the composition of the feather mites between migrant and resident species. So I found the same family of feather mites in migrants as I did in residents. And this could be due to the fact that maybe feather mites are more generalists, but it could also be due to sample size or to the fact that I wasn't able to identify to like species level, so maybe we would see a difference in species, just not family of the feather mites. So in conclusion, morphological features such as body weight and wing chord are going to impact the ectoparasite load because of the ability for them to house these ectoparasites. And then life history traits are going to impact contact rates, which will impact the ectoparasite load as well. So there's a mixture of factors that relate to the ectoparasite load, and it's important to study what these factors are so that we can relate it to bird health, because as I said before, all avian taxa have a feather mite association, so just through this prevalence, we can assume that it impacts bird health. And future research should look more into this relationship of feather mites and birds to see if it is more of a mutualist or a parasitic relationship, and also to try and do the same study in the dry season to see if molt or migratory status has. I would like to thank all of these people, especially the people in the MOSI program, Lucy Moreno, Victorino Molina, Diego Rodriguez, and Molina, for waking up with me and for teaching me all there is to know about misnets and birds. I'd like to thank my advisors, Natalie and Federico, as well as Dr. Heather Proctor, who I reached out to. She works at the University of Alberta, and she kindly was able to help me identify the feather mites that looked pretty similar to me. So, also my homestay family for waking up and feeding me breakfast at 4 in the morning, and the Institute of Monteverde in New Zealand.
Now we have time for questions.
Yeah.
How is the bird aware of having these mites, and what does it affect? Why do they bother to preen them off?
Yeah, so this study actually showed that the birds aren't really that well at preening them off, and multiple studies have shown more recently that these feather mites may actually be more commensal. One even showed that it could be mutualistic because they can eat harmful fungi that have been found on bird feathers. But, yeah.
Why does the bird want to get rid of them?
Usually that is in the case of like ticks or lice. So the stuff that's like sucking their blood or eating their skin or yeah. That's what is interesting about this. This kind of shows that they can't really tell that they're there. But one study found that if they are in very great abundance, it is going to negatively affect them. Yeah.
What's your favorite bird that you saw in this study?
Oh, so many. Probably the white-breasted blue wren. Yeah.
Are you just looking at the primary feathers to check other parts of the body, like belly and chest?
Yeah, so I did just look at primary feathers. And when we handled the birds, we did a look for like larger ectoparasites. And in one bird, we only found like a Boscidae diptera fly. So I just decided not to look at those just due to lack of finding them. But when I emailed Dr. Heather Proctor, she said that it would be interesting to look at also feathers in the body, because like plumage feathers are going to have different mites than just the primaries. But yeah, I did just look at primary feathers.
Muchas gracias. Okay, next speaker, Eliana Chandalov from UC Santa Barbara is going to speak about the blood parasites abundance in bats, depending on anthropogenically places or natural places. And for a given clinical problem, the first slide just is suffering blood parasitosis, but it's coming now. Muy bien.
Hi guys, I'm Eliana. I'm studying blood parasite abundance in anthropogenically impacted populations versus natural population of bats. So first of all, the purpose of this study is, my interest in this study comes from the fact that humans, while they infringe on the natural environment more and more in recent years, are purposely or accidentally providing food sources to wild mammals. And this draws a larger accumulation of for bats specifically, it draws a larger accumulation of bats to one place, which would create grounds for easier disease spread, whether that's through hemoparasites, which are found in the blood, or ectoparasites, which are found externally, and that can be like mites and flies, as we just saw in Olivia's presentation. And this study is important to me because I believe that it's valuable to see the human impact on wild populations, and doing a side-by-side comparison can be really helpful for that. So bats are the perfect disease reservoirs, and this means that they can host infectious agents in their bloodstreams, and they can live and multiply within the bats, and then the bats can spread those agents to other bats or to other species. And some of the reasons that this is possible is because of their socialization habits, where they spend time in large groups called roosts. Also, they have very high species diversity, so parasites can really thrive in them. And because of their high species diversity, they can also harbor a wide range of hemoparasites. And they're also the most abundant animal order in Costa Rica, so there are just a lot of them. Also, due to their body form, they have hollow bones and are lightweight, so they can fly far, which contributes to how wide their distribution can be, and they can occupy a lot of different niches. Also, microbats, which I studied a lot because those are nectar bats, they cast out particles when they echolocate, called echolocation aerosols, and those are potential hosts for contaminants. There are a lot of risks associated with human impact. One of those is that because there's a provided food source, there's a lot of accumulation of those food sources. Often that's a place where bats can share the vectors, which are the ectoparasites, and the ectoparasites often contain hemoparasites, so that's a big way that they can spread. Also, during times of environmental stress, those constantly available food sources are places that the bats always know that they can go to get that food and to be under less stress, and so they're likely to stay around the same area for a longer period of time and to have higher exposure to each other. And meanwhile, bats that stay in continuous forest and rely on their own food sources are not subjected to the same cross-contamination. So then my question is, how does parasite abundance differ between bat populations in anthropogenically impacted areas and natural areas? So to compare our two populations, we have from the hummingbird gallery over here on the left, the anthropogenically impacted population. I caught 10 bats here, and I used a butterfly net, and I would wait, I had some help with this too, you'll see my acknowledgements. But we would wait by the dark hummingbird feeders, catch them with the butterfly nets, and then observe them. My natural population on the right here comes from San Gerardo on a field trip and Crandall Trail, Monteverde Institute. And I caught 8 bats by mist netting, which I'll get into in a second. And then an important note is that all the bats from the hummingbird gallery were nectar bats of the species Hylanectars underwoodi. And then from San Gerardo and Crandall, I had a lot of Carolia bats and Sternera bats, which are fruit eaters. So for my misting procedure, I would set up the nets at Crandall and open them just after dark. Check every 15 to 20 minutes to see if I had captured any bats. If I did, then I would wear thick gloves to protect my hands, because often the bats were thrashing about and trying to bite their way free while I was untangling them. But I had to be very gentle and be very meticulous in the untangling, and you can see that in the photo on the right here. After I untangled them, I would place the bats into fabric bags until I was ready to observe them. My main point of data collection came from my blood sampling. So how that would work is one of my team members, usually Katie, would be holding the bat for me. I would spray the foot with alcohol to sanitize it, and then puncture the vein on the foot with a fine needle. And I tended to get better at this as I did it more and more times. I would bring a sterilized slide to the foot to collect the drop, and then take another slide at a 45-degree angle and make a thin blood smear. While I was doing this, another one of my team members, usually Federico, was holding cotton to the wound, or to the puncture, to clot the blood. And then we would give a shaggy haircut to the bat that we caught to make sure that we didn't recapture the same one again to mark it. And then we took photos and videos of each bat. Then my lab work consisted of staining slides. So I begin with soaking it in 100% ethanol for 2 to 3 minutes, as you can see in this picture on the left. And then I created a staining solution. The reason for doing this was to make sure that the slides were more visible under the compound microscope. And so that was a 4-to-1 ratio of buffer to Jameson stain. And then after I let that sit in a petri dish with the Jameson stain for 11 to 12 minutes, and then wash with distilled water, and then I would observe them under the microscope and take photos. So for my results, this first graph over here is about the mean number of hemoparasites in anthropogenically impacted areas and natural areas. You can see the population labels on the x-axis with anthropogenically impacted being the blue bar, and its average was 6.2, and then the natural area being the red bar, and its average was 4.3. And then on the y-axis is that average number of parasites. My next graph, I was comparing the number of ectoparasites and number of chemoparasites on each bat itself. So on this scatter plot, each dot represents one bat, and you can see its number of chemoparasites on the x-axis and number of ectoparasites on the y-axis. I put a line of best fit on there, and as you can see, there's a pretty weak correlation, which was a little bit surprising. Its R-squared value is 0.079, so very, very low. These are some of the parasites I observed from my first bat. This is a Hylonicterus underwoodi in my anthropogenically impacted population caught at the Hummingbird Gallery, and this bat had a total of 13 parasites, and these were the largest parasites I observed. Bat 2 and Bat 3 were also Hylonicterus underwoodi bats from the Hummingbird Gallery, and bat number 2 had 9 hemoparasites, and bat number 3 had 15 parasites, and that 15 parasites was the largest parasite load that I observed in my study. And then these last ones, from bat 12 to bat 18, are from my natural population, and I observed 10 hemoparasites each. So overall, my data does differ from that of previous experiments that I've looked into that explore a similar question to me. And because it should be that there's higher hemocyte prevalence in anthropogenically impacted area bats than natural area bats. But in my statistical tests, I couldn't find too much of a difference between the two populations, probably due to limited sample size. And then there was also surprisingly a low correlation between ectoparasites and hemoparasites. And the reason that's surprising is because the ectoparasites typically harbor hemoparasites, so you would expect there to be a relatively direct correlation. Some possible explanations for this could be that the populations were relatively overlapping. All of these bats are coming from a continuous forest, and I wouldn't be able to tell if they were lingering among the same group of bats. Also, a possibility is that in addition to the nectar bats, depending on the nectar food source, which I knew at the Hummingbird Gallery, the frugivores that I caught in San Gerardo and Crandall also could have been using anthropogenically provided food sources like fruit trees, and I wouldn't have a way of controlling that or being aware of that. Also, the Hummingbird Gallery is an ecotourism site, so it's probable that those hummingbird feeders are clean, so they're not as much as a breeding ground for contagions as I thought. So overall, there is a higher average number of parasites in anthropogenically impacted populations than natural populations, despite the statistical tests I did. And it's clear that bats are evolutionary gifted either way. Their features allow them to harbor diseases, and then this can relate to zoonotic diseases, how there may be spillover as humans infringe more and more into the natural populations and use these food sources to bring bats more out of their natural habitats. Overall, I believe that human impact would have been clearer if more isolated populations have been observed, so that's what future studies could look into.
Muy bien, we have time for questions.
Did you see any similarities between the types of parasites you found in like the same species? That was difficult to analyze. I'm not a parasite expert, and this is all super new for me. So I did find a lot of like worm-looking ones, but I didn't get into the classification too much. because I felt like that was beyond the scope of my project.
Okay. Thank you again. Okay. So the penultimate presentation of this section is coming, and it's from William Rice from UC Santa Barbara. William Simpelo, but now Pompelo, is going to speak about behavior changes in wild and captive nectar bats.
Gracias, Fede.
Voy. Dame cambio. Sí. Típico.
Okay, my name is Will, and I study the behavioral changes in wild and captive nectar bats, the subfamily Clausophaginae, in response to predator cues. So first off, you're not hallucinating. Those are poison dart frogs, not nectar bats. And I show them to display a little bit about predator-prey dynamics. So the species on the right uses aposematic coloration to display its toxicity to potential predators to avoid predation, and the species on the left is an imitator, a mimic, which uses Bayesian mimicry. It produces less toxins but still avoids predators by showing in a very conspicuous manner similar coloration to the toxic species. So I want to show these guys also because in captivity they stop producing their toxins as a result of differentiation in diet. But it is an example of how captive animals, when they're taken out of areas of high predation pressure, may have a change in their anti-predatory mechanisms, even though they've been evolved for millions of years. So now we're going to watch how a bat avoids predators. They use crypsis. They're not very conspicuous at all. They come out at night, they fly quickly, they're very agile, and they fly silently, which I say in air quotes because while to the human ears we can't hear what they're doing, almost this entire time they're producing ultrasound in order to navigate, to locate food sources, and to detect and avoid potential predators. And stop me if you've heard this before, but they're understudied. Largely because it's hard to see them, and they're also under-eaten. They have some of the lowest mortality rates of any vertebrates in the world, and there's a very small amount of evidence that suggests specialization in eating these bats. For the most part, most predation is opportunistic, but for bats especially, there's pretty much the only evidence of predation comes from opportunistic predation in basically the remains or the fecal matter or evidence after they've already been eaten. And so for that reason, they're under investigation by me. So I wanted to investigate how they would respond to visual and olfactory cues and how they would change their foraging behavior. So I went to two locations, the Hummingbird Gallery, which Eliana introduced us to, and the Bat Jungle, which is a ecotourism site and bat rescue center, and very unique because there are not a lot of captive bat enclosures in the world. So my hypothesis here was because the wild bats experience constant predation pressure, constantly use their echolocation to navigate, to locate food, detect and avoid predators, and because they use olfactory senses in an environment full of different smells, that they would decrease their foraging visits for predator cues, but not for a neutral object that I placed in a similar position. In contrast, the captive bats, although they still possess innate and learned predator avoidance, and a bit of a limitation here is that I don't know how long each bat has been in the bat jungle. Some are born there, some have been there for a month, some have been there for years, but they do not experience predation, and they live in a very controlled, constant environment. So I predicted that they would not significantly decrease their foraging visits for any treatment. So my methods First, a control. This is just the hummingbird feeder or the nectar disc at the bat jungle to get the normal amount of visitation, followed by a random treatment, either a model snake, which was a visual cue. Unfortunately, it was a cobra, which are not native, but I found it under Frank's bench outside his house. Then a feces from a boa imperatora juvenile that we housed for a little bit of time, which is the olfactory cue. And then a black bag or a tarp, which is the neutral object. The reasoning for the neutral object here, is that if the visitation rate decreased for any object, I wouldn't know if it was because they were distinguishing between a predator or not. So if they decrease their visits for everything, I'm able to tell if it's a predator or just a neutral object. And then a rest period where I would let them kind of re-equilibrate to a predator-free environment. So the methods just record the number of visits during each treatment. Here's what the model snake looked like at the hummingbird gallery, and that's the snake odor at the bat jungle, and the neutral object looks just like the bag there. Record the number of visits, and then data analysis was an ANOVA with post-hoc Tukey's tests. And here's what a visit looks like. And in case you blinked, this is what a visit looks like slower. So what they're doing is they're hovering, and they're sometimes called humming bats because they do this kind of stalling maneuver, stick out their tongues, get some nectar. And they're extremely active because some species will drink double their body weight in nectar every single night, which also makes them extremely active and important pollinators. So for my results, in the hummingbird gallery, on the left here is the control, the no treatment. On the right is the neutral object treatment. And the two in the middle are the visual and the olfactory cue. So they did not significantly decrease their visits in response to the neutral object. They significantly decreased visits only in response to predator cues and had a similar response to both visual and the olfactory cue. These are the wild bats. And then the captive bats in the bat jungle decreased their visits in response to all cues, significantly decreased their visits in response to the olfactory cue when we compare it to the neutral object. And while they tended to avoid the model snake, they did not significantly reduce their visits when compared to the neutral object. So what does this mean? First off, in the wild bats, the foraging groups involved many species. The Eliana recovered 10 Hylonicterus underwoodi, which is probably the most abundant species there, but through video evidence, I found Anura jeffroyi, Glossophaga species. There's likely a lot of bats. Some may be habituated and have a colony nearby, and some of them may be visitors. There's some studies that show nectar bats traveling as far as 40 kilometers to hummingbird feeders, which are largely used as kind of stopgaps. These are not a full nutritional diet. It's really just sugar and water. And then also it shows that boids, such as the boa imperator, because they significantly avoided the snake odor, we can probably say that they are opportunistic predators of nectar bats, just as literature suggests as well. And that they're using a variety of senses. They're using their ultrasound, they're using even vision in low light, and they're using their olfactory cues to avoid predators and decreasing their foraging behavior, which also aligns with a study on acoustics in fruit bats. Unfortunately, because of the understudied nature, especially of nectar bats, I kind of have to do comparisons with different groups within the Philostomids, which is the New World leaf-nosed bats. However, they also decrease their foraging visits in response to predator cues. And this whole kind of thing is that they're weighing their predation risk and the food rewards with what they're seeing. Sometimes they are identifying a predator and turning around, and they're often doing what we call predator inspection, which has been observed in bats many times before, which is basically they're going to hover up to the predator, the visual cue, or the snake odor. They're going to look at it, and they're going to probably emit a bunch of ultrasound or smell it and figure out what it is. And then go away and come back and go away. And sometimes they keep feeding, and the other ones may see that they're feeding and, I don't know, become a little bit more courageous. So the captive bats, in contrast with my predictions, they did significantly avoid all objects, including the neutral object, which for me tells me that all these objects are disruptions in what's a very controlled, low-stress environment. They sit in there for basically 12 hours a day, and they know their food source is always there, even after I stop annoying them. So they'll go up and sort of roost and might wait, knowing that they still have food sources later on. And because of this lack of predation pressure, I believe that they would have this decreased response, but they were still able, very significantly, to identify the olfactory cue and avoid it, decreasing their foraging behavior. But their visual response, while it was slightly diminished, they did not significantly avoid the model snake in comparison to the neutral object, which corresponds with something that Bino, the manager of the bat jungle, told me, which is that these bats generally don't use their ultrasound much anymore. So they're basically mostly relying on their olfactory cues to avoid predators and to find their foods, their food sources. But in this environment, they don't need to use the extra energy, which ultrasound does use some kind of extra energy. They're able to just sort of relax. And so this is another one of the effects of having animals in captivity. They may change their anti-predatory mechanisms, even if these are, highly costly evolutionarily evolved factors. And I'd like to acknowledge my advisors, Natalie and Federico, of course Frank, my homestay family, the bat jungle, including Osito, this little guy that kept me company all day, the Hummingbird Gallery, family, a girlfriend for allowing me to be in Costa Rica for 3 months, everyone else, and the Montserrat night tours for interrupting me like every single night at the Hummingbird Gallery with flashlights and all, because they taught me perhaps a much needed lesson in patience. Thank you.
Muy bien. Time for questions.
I'm assuming that you visited the Hummingbird Garden and the bat jungle multiple times. Do you think that over your study period they might have gotten habituated to putting items like near them, which could have resulted in them kind of enjoying it more?
Mm-hmm. Yeah, there's various limitations with habituation, including the fact that the snake odor was probably losing its odor over time. But when I kind of looked at the data, the distributions were still sort of varied. And I was seeing these varied visitation rates based on day-to-day activity and sometimes like minute-to-minute activity. But I looked at it and I didn't see any significant decrease or increase in visits using the same model, the same place over time.
Do you know how long the bats have been in the bat jungle in captivity? Multiple generations, or they all rescues?
So the majority are rescues. There are a few that are in that were born there, but for the most part they're caught in the wild. And this would be sort of a further study. Would be great to be able to tag them for their age, for how long they've actually been at the bat jungle, and then to be able to track their kind of predator detection and anti-predatory behavior as a function of how long they've been in captivity. But I didn't have access to information.
You mentioned that Bino says that the bats in the bat jungle have stopped using echolocation. Did I understand that correctly?
I don't think they've stopped it, but he tells me they have anabats in there, and he says that they don't go off very much anymore. In fact, it used to be sort of a tourist attraction to listen to them. And not only are philostomans incredibly hard to grab on recording, but also it seems that, especially because if we look back here, this environment is it's like mimicking dusk, but it's a higher ambient light than the pitch black that the wild bats are in. So even though they're not blind, and even though they have reduced vision, and they certainly are still using ultrasound, I do believe, I do agree with him that they're probably reducing it and not using it as much or relying on it as much as the wild bats continue to.
Thank you. And yes, it is a very consuming energy thing to be echolocated. More questions? Gracias. Well. You have your computer. next presentation is the last of this section, so sorry to tell you, but this is the life. So, Katie Van Power from UC Santa Barbara with a very interesting study about the impact of light and bats.
Hi everyone, welcome to my presentation, Marcelo and me, where I'm going to discuss the impact of light pollution on bat species diversity in Monteverde, Costa Rica. So light pollution is a worldwide problem that has been growing recently with increases in urbanization. Locations that are experiencing new urbanization are ones having the most impact from light pollution. In fact, some studies are showing that light pollution is increasing by 10% annually. In the context of this study, I'm defining light pollution as the presence of any unnatural outdoor light from the hours from dusk until dawn. And ecologically, this has huge impacts on species. The classic example of this would be sea turtle hatchlings becoming confused by the presence of city lights and moving towards those instead of the ocean. Monteverde provides the perfect backdrop for this study, as Costa Rica has increased its urban footprint by 112% since the 1980s. And Monteverde itself has changed from being a small agriculturally based community into an ecotourism hub that is now seeing 200,000 visitors annually. So how is bat diversity related to the light pollution? Well, bats are obviously nocturnal species, so the presence of light impacts their day-to-day activities. And in Costa Rica, there are 112 species of bats. This gives them super high niche diversity, which means they fulfill multiple different aspects of the ecosystem. They have diets that range from everything from blood to insects to nectar, fruit, and more. This makes them the ideal bioindicator to study the impact of light pollution. So this brings me to my question: how does the presence of nighttime artificial outdoor light affect bat diversity in Monteverde? And I hypothesize that light pollution will have a negative impact on bat diversity. So going into my methods on how I looked into the bat diversity in Monteverde, I used two different collection methods. I used a recording device called an AudioMop, which is a supersonic recording device, and then I also used MistNet. For the AudioMops, I'm looking at capturing the diversity of insectivore bats, as they use an echolocation signal that comes out like a cone, so it's easily picked up by the recorder. And to do this, I used two recorders per location, and the recorders were set to record for 20 seconds every 5 minutes from 5:30 p.m. until 8:30 p.m. I also used two MistNets at each location. The MistNets were 12 meters long by 2 meters high, and they'd be opened at 5:30 p.m. until we reached the maximum capacity of bats we could manage for that night, or there was a weather issue because they can't be worked in the presence of lightning or rain. When we were removing bats from the MistNet, this is Eliana demonstrating, we would go ahead and start with the legs, move on to detangling wings, body, and then finally the head. Once we had the bat detangled, we would place it in a bat bag, bring it back into our home base area, and then we would identify their species. Just a little bit more about my methods. This is a map of the different study sites I used. Two were in the Monteverde area, and then one was in San Gerardo. Those three study locations each contained two sites, one dark site, which was my control group, and then a light site, my experimental group. The light site was lit by a 9-watt LED light bulb, and the dates for my recording occurred from November 13th until the 28th. However, due to some issues with the weather, I chose to only include data from missetting days that I received at least one bat, and AudioMop dates where I received more than one bat pass call. So what is a bat pass? Here we are looking at some sonograms, like we did with Quinn's project, and this is basically how I go through and identify what species of bat I'm listening to. Just to give you guys a little preface of how I would do this. So for my Myotis pilosotivulus, they have a much higher beginning of their frequency. So they start around 60 kilohertz, which is significantly higher than my Myotis riparius or my Myotis nigricans. And the difference between those two is the nigricans tend to have most of their call underneath 55 kilohertz, and the riparius are above 55 kilohertz. Moving on to my results, this is just a graph of all the different species I found between the audio mock recording and then the MISNet. These are just also some images to go along with the different species I saw. Just a couple cool things to point out. These are going to be my myotis. Those are all insectivores. And a Norajophy, which is a nectivore. And then these guys are all frugivores, my Corellia, which have a short tail. And then my Spirina, which do not have a tail. Moving on to my results that I received from my data collection. This is an average number of bat passes per hour. In the purple, you can see in the dark treatment, and the blue is going to be the light treatment. Between these, as you can see, there were more bat passes per hour in the dark treatment across all species. However, to determine if this was a statistically significant difference, I went ahead and did a chi-squared analysis between the two treatments for all three species. And I found that for the Myotis nigricans, it was a statistically insignificant difference. However, for my other two species, it was statistically significant. Moving on, I also performed a Shannon-Wiener index between these two treatments. And the Shannon-Wiener index is on a scale from 0 into the natural log of the number of species present. So with 3 species present, that's on a scale from 0 to 1.10, which with 1.10 as the highest diversity and 0 as the lowest. So as you can see, these are considered lower diversity levels. However, the dark treatment is significantly more diverse by about 10%. So what does this mean? Going back into my Myotis pilositibulus and my Myotis rariperius, these are statistically significant amounts, meaning they are likely impacted by the presence of that artificial nighttime light, whereas my Myotis nigricans were not impacted by this light presence. And this may be due to some diet differences. The nigricans may be more willing to go into light because there's a greater source of food there for them. However, overall, I would say that AudioMox supports my hypothesis, as the dark treatment was shown to be more diverse with that Shannon-Wiener index. And then moving on to my results for the misstanding data, these are all the species that I found. On the x-axis and the y-axis is the number of individuals per species. Do go ahead and note that there is no overlap between the species in the light and in the dark. And due to this, I chose to look at the total number of individuals present in the dark treatment versus the light treatment. And then I wanted to determine if this difference between 6 and 3 was statistically significant. So I did another chi-squared analysis where it was determined to be a statistically insignificant difference. And because of this, I believe that the weather likely impacted the results from my data collection. There was a tropical storm occurring during this time, which likely resulted in bats being moved from their normal habitats, or also bats being in starvation mode where they are more willing to risk exposure to predation to receive food. However, I do also want to emphasize, looking at previous research, it is likely that frugivore and nectivore bats are less impacted by light pollution, and this is probably due to their diet as they eat fruit and flowers and have to be more willing to move wherever food is available. So to kind of wrap everything up and conclude, light pollution is a growing issue worldwide, and bat diversity is a great way to go through and understand how this is impacting our ecosystems. To look at the impact of light pollution in Monteverde, I used a combination of mist-setting and audio-mock recording, and then with that, I performed a chi-squared analysis and Shannon-Wiener index to look into my results and their impacts. From these results, my hypothesis is partially supported as bat diversity was impacted by the presence of light pollution for insectivores. However, it was not impacted in nectivore and frugivore bats. However, again, this is likely due to the tropical storm present, and if I had more data collection, this could be a different result. Overall, I think there should be more research looking into the impact of light pollution, as light pollution is a growing problem, especially in neotropical regions, and it would be great to raise awareness for this and bring more funding to solutions. I'd like to acknowledge the lovely Federico Chichia and my partner in crime, Eliana, and all these other names here, my host mom, Martha Solis. Natalie, thank you for reading over my project. Adriana for explaining how to use the audio box. Frank and Katie Joyce for letting me use their property to do my research. The Estación Biológica and Crandall Memorial Reserve. And then finally CIE for letting me use their materials. Thank you.
questions?
Clara? Just curious, you mentioned how like there was a specific number of bats you could catch in this nets at a time. What was that number?
So what we would do is we basically need to make sure we'd be able to process the number of bats in like a timely manner. And in the course of like the data collection, generally the limiting factor would be the weather. So if it started misting or raining to a point where the mist nets would become wet, we would no longer be able to do that for the night because it increases the danger for the bats. It becomes a lot harder to detangle them. Then also they can be exposed like to the elements and get hypothermia and have issues like that.
So, like for my research, was it fruit and insect bats? Sometimes we'll risk leaving before sunset or before it gets like to night. In order to, and they might risk predation to do that. So did you put out mist nets or do audio moss before it was dark at all?
We did not.
Okay.
But that would definitely be an interesting area to keep looking into.
Yeah.
Me?
Who raised their
Okay,
hand?
what was your favorite part of it?
Oh, you, of course.
Okay. Gracias, Katie. So, you remember the project about gestures? There is an old world monkey as doing something like that I don't know what it is. It is like code. I guess we will have a picture. Okay. So
listen up. We're gonna go like in the grass, in the sun, and take a quick picture. And then there's still a ton of refrigerio, so we'll have some refrigerio, cafe, and then come back as quickly as possible.
I saw that. I've been looking at it.
¿Esto qué se queda?
Ya.
¿Esto se queda?
¿Sí?
Vamos a ir a tomarnos una foto, ya venimos.
presenter.
I'm really sorry for eating all the panelitos. So this is another box of panelitos.
Wait, is that?
This one? Yeah. They're trying to mess up one.
You actually just found this in your friend's. No, I was told it was there. But.
Thank you. You ever seen a cobra in Costa Rica? Same for the bats. Good study, don't worry about it.
But. Wait, is it not?
So right? No, Naric's next, right?
Garlic roll.
Who's yours? Oh, hell no. I'm doing on my computer. I don't have my computer. Do you want to put your computer?
I have it on my computer too.
Yeah, but do you want to put it on mine?
Shout out to Narc's uncle.
Narc's uncle.
Shout out Narc's uncle. We're so excited for Narc to go.
I can't wait to learn about microplastics, Narc.
Narc is a very good boy.
That's on YouTube forever.
Never mind. No. If you have it and it works. Okay, it's not. Go ahead.
Humans in Nature, and our first speaker is Narek Kirkjan, and he's going to talk about microplastic in fish in Kohidiki.
Okay, discovering plastic. Hello everybody, my name is Narek Kirkjan, and my independent research project was about the assessment of microplastic presence in the digestive tract of Lucianus, Peru from the Bajo Muñecos fishing site. So introduction, what are plastics? Plastics are synthetic materials derived from fossil fuels, and their invention in the early 1900s has revolutionized the way we live. They quickly gained popularity because of their affordability, versatility, and durability, and they come in an astonishing variety of colors, sizes, and shapes, serving countless purposes in modern life. However, this convenience has led to a staggering reality. Over 340 million tons of plastic are produced annually, and most of it goes into landfills and as waste. Now, what is plastic pollution? Plastic pollution is the accumulation of plastic waste in ecosystems around the world. Over time, these plastics break down from exposure to natural elements like sun, water, and wind, and the result, microplastics. These tiny plastic fragments are less than 5 millimeters in size, and they infiltrate ecosystems, food chains, and even the air we breathe. So, the research question I sought out to study was, what is the abundance of microplastics in the digestive tract of a commercially caught fish species, Lucianus, Peru, at Bajo Muñecos, a prominent fishing site? So, methods. So the first 2 to 3 days in Guayaquil, I spent talking to local fishermen, trying to figure out two main important facts. First, what is a popular commercial fish that they like to eat in the region? Because I don't like to eat fish, so the fisherman's opinion was pretty important to me. And two, where am I going to actually catch these fish? So after countless conversations, I decided that I would be in Bajo Muñecos, which is about 2.4 kilometers off the shore of mainland Costa Rica, the perfect distance where it's still affected from macro and microplastic pollution. And I chose Pargo Rojo, or the Pacific Red Snapper. After deciding those two, I went on countless fishing trips, and this is me after catching my first fish. Woo! And then after catching the fish, I would put them on ice and transport them back to the Lara house, where I would gut the fish with the help of the Lara brothers, and I would put the digestive tracts into these glass jars to prevent any additional plastic contamination from plastic bags or plastic jars. Once I got the tracts, I had to measure the relative displacement of each digestive tract, and I did that by measuring the displacement of water. And once I found that, I used that to calculate the amount of water I would use, and I used 10% of that to calculate the amount of potassium hydroxide I would use to dissolve the digestive tracts. I created a solution of 10% potassium hydroxide to the volume of water, as you can see here. I let the solution sit for 24 hours, and then I filtered them through coffee paper. And I apologize for my Guayaquil peers for having to smell this every day, every time they walk into the laboratory, because fish guts after 24 hours of digesting doesn't smell the best. So after that, I let the coffee filter air dry for a couple of minutes, and then I sectioned them off onto glass microscope slides, and I analyzed each digestive tract or each coffee filter for 30 minutes under a compound microscope, searching for microplastics. The results, I found microplastics in all 20 of the fish sampled, and there was a total of 94 microplastics found. The most prevalent type was microfibers. So as you can see here, there's like a yellow microfiber next to a blue microfiber, and this is all in one fish. And then over here, there's a microplastic fragment. It's a little blue fragment. And then over here, there's another microfiber, and then another one in red. More statistics. So the data revealed an average of 4.7 microplastics per fish, with a standard deviation of about 1.92, indicating some variability in microplastic presence. The median was 5, and the most frequent observed counts, or the modes, were 3 and 5. And then this is just a graph highlighting that, yeah, all 20 fish did have microplastics in them. And then this is another graph showing the number of microplastics in the number of fish. So again, 3 and 5. There were 5 fish with 3 and 5 microplastics, followed by 6 microplastics, 4, and so on. The lowest number I had was 1, and the highest number I had was 10. So discussion. The presence of microplastics in all fish underscores the widespread contamination of microplastics in marine ecosystems. The large proportion of microfibers highlights certain trends in plastic pollution. So one of the main pollutants of these microfibers is actually washing machines. So as we all know, there's synthetic fabrics and natural fabrics. And when you wash synthetic fabrics, they go from your washing machine into the ocean and then back to you on your dinner plate. And each like clothing article that's synthetic can release up to 1,900 microfibers per wash. So if you multiply that by the number of articles to the number of washes, there was like, I think there's about 30,000 per wash on average. So that's the first microfiber source. And the second source would just be degrading fishing gear. So Quainikul is a popular fishing town, and most of the fishing gear is plastic fishing line, ropes, and nets. And over time, they just get cut off, degraded, and they move, and they're beached, and they just, again, due to natural elements like sun, water, and wind, they break down. Further discussion on microplastics. The real issue is that microplastics can act as chemical sponges, and they can absorb pesticides, heavy metals, and hormone-disrupting toxins. And they can actually cross biological membranes, so they do leave the digestive tract and enter the circulatory system, and then they reach your internal vital organs. Over here we could see, this is a microplastic and cell interaction. So first it attaches to the cell membrane, and then it gets internalized by the cell, so it's able to enter the cell. And then over here is just another diagram illustrating a plastic entering the cell membrane. And these microplastics ultimately pass through the food chain, affecting humans. So right now I'm only talking about fish because my project was analyzing the digestive tracts of the Pacific red snapper, but don't think that these aren't coming back to us through the food we eat and through the marine food chain. So conclusion. So microplastics are pervasive pollutants that contaminate organisms and ecosystems. Again, all 20 sampled fish had microplastics in their digestive tract. Microplastics carry harmful chemicals and infiltrate biological systems, and we need urgent cooperation to mitigate plastic pollution at its source. As I explained earlier, these are just some examples of natural fibers. There's plastic and animal-based, there's cotton, hemp, linen, silk, and wool, and then there's the synthetic fabrics, which are popular amongst a lot of us as like outdoorsmen or outdoors people, and it's the quick-drying polyester towels that we all bought to bring here, or the nylon shirts that are supposed to also dry fast. And I don't blame you guys, because all my clothes have been wet for the entire time, but it's something to think about is how, even though you might be dry, the ocean is getting more plastics in it. And then, again, this is just a beach that's just been polluted by single-use plastics, so that's another thing we can take upon ourselves. It's to stop using single-use plastics, and to try to use reusable sources. And it's important to reduce growth in plastic production rather than just trying to individually stop using plastic, because as corporations and governments continue producing plastic, it doesn't matter at the end of the day what we as individuals do if the world doesn't come together on this. We need to eliminate avoidable plastic use and expand reuse and refill systems. Acknowledgements. Thank you, Emmy, Stephen, Minor, Annabelle, Lara, Leo, Frank, Joyce, and my incredible family. Thank you, Purvia.
Thank you for the reality check, Nadik. Were there any questions?
Yes, quick.
Did you notice any trends on like which fish had more plastic that you observed? Like were there more for certain species, or like if they have a certain diet?
So I only looked at one species. It was the Pacific red snapper, the Lucianus peru. And all fish were about the same size. Some were a little smaller, a little bigger, so there wasn't really a difference based on the size of the fish. And then also, they were all caught in the same area, so there wasn't really any like regional difference that I could attribute the plastic consumption to. You
I'm just interested in how you do with the rest of the fish, because you only test for the digestive.
like the rest of the fish after I gutted it. They were given to like local families, like my family ate some, and then I gave it to John's family and Jaden and Owen's family. Actually, I texted in the group me, and a lot of people left me on the red, so only whoever texted their families got to eat some fish. Yeah.
Did you tell the people who ate the fish that they were being tested for microplastics?
To the best of my Spanish ability, I tried. Katie.
Are red snapper known to like have higher amounts of microplastics, or?
They're just known to be really commercially popular. Like everyone in the region loves the meat of the fish.
What method of fishing were you like using a line?
I was using like a hand reel, and I would cast it with like a sardine bait, and then after just reel.
Okay, last question.
Not a super important one. Is this part of the reason you don't eat fish, or is this just really validating?
Yeah, this is part of the reason. No, I just don't like the taste of fish.
Okay.
Miguel.
Okay, go for it.
How many fish did you catch yourself?
Myself, I caught 2 fish out of the toilet. in the first week, I didn't actually catch it, but I reeled it up. So.
Thank you, guys. Okay, our next speaker is Sonia Jekyll from UC Santa Barbara. And she'll be talking to us about vitamin C content in oranges.
Is it ready?
No. Okay, hello everybody. My name is Sonia Jekyll, and this is my presentation on the analysis of vitamin C concentration in oranges cultivated under different agricultural practices. And now you may wonder, why is this even important? Well, conventional agriculture consumes large quantities of fresh water, pollutes water supplies with fertilizers and pesticides, contributes about 1/4 of the greenhouse gas emissions, and often requires extensive land use, which leads to deforestation. So overall, not too great for the environment. But we still need to feed our 8 billion people on Earth, so we need to find a sustainable way to keep feeding all these people, but also not contributing as much. And that's where sustainable agriculture comes in. And so a few different types of sustainable agriculture are polyculture and organic farming, which is what I chose to look more closely at. And if you can see on the picture, it is a polyculture farm in Costa Rica that we visited. It's Finca Florencia in San Luis. And polyculture is basically when there's a bunch of different types of plants grown on the same plot. And this improves the soil fertility, which reduces the amount of nutrient depletion. And then the other type of sustainable agriculture that I chose to look at is organic farming. Organic farming is when you don't use any chemicals like pesticides and fertilizers. And so some ways that these can help the environment is that it improves soil fertility. As I mentioned earlier, the nutrients aren't depleted so that you can use the soil for a lot longer, which reduces the amount of deforestation required for agriculture because you can use the same plot many, many more times. Fertile soil also can store carbon and help with carbon sequestration, which is super important with climate change because we want to reduce the amount of carbon in the atmosphere. And then finally, sustainable agriculture doesn't use all these chemicals, so there's less water pollution and air pollution because it's just not being released into the environment. However, a major problem with sustainable agriculture is that it is quite expensive and not super accessible to everybody. And so it often requires a personal incentive for someone to switch besides it just being better for the environment because they have to go out of their way to pay more and choose the sustainable option. And so a personal incentive could be like the nutritional quality of different fruits and vegetables. And so part of the nutritional quality that we get from our fruits and vegetables are vitamins. And vitamins, they play an essential role in our human health. They do pretty much everything, like everything you can think of, they're very important. And for my experiment, I decided to look more specifically at vitamin C.
So vitamin C is super important for immune function, metabolism, collagen synthesis, bone health, like anything, like you get a cut or a boo-boo, like it'll fix it. So it is super important that we get the vitamin C that we can, like, in our diets. So I wanted to test if sustainable agriculture had more vitamin C than non-sustainable options. So I looked at oranges more specifically, and I hypothesized that conventionally grown oranges had less vitamin C than sustainably grown oranges. And to do so, I had kind of two main steps. I first collected the oranges, and then I did a titration to chemically determine the concentration. And so for collecting the oranges, I collected 9 oranges per type. So I collected 9 oranges from the conventional place, which was Mega Super Campo or Mega Super, and then 9 oranges each from Trapiche Don Simon and Finca Florencia, which were polyculture farms that we visited in our class, and then 9 oranges from the Whole Foods Market, which were certified organic oranges. And then once I collected all the oranges, I decided to separate them by ripeness in case the ripeness had an effect on the concentration. So I separated it by green, and yellow. Green oranges were 75% green, green oranges were 50% yellow, 50% green, and then yellow oranges were 75% yellow. And doing this, I realized that oranges in Costa Rica are not actually orange. They are but anyway, then I titrated the oranges to determine the vitamin C concentration. I did 2 titrations per orange, so a lot of titrations. And I did an iodometric titration. So in the burette there was iodine, and it was slowly added into a diluted orange juice, and the iodine would react with the vitamin C, creating a colorless liquid. But once the vitamin C fully reacted, then the iodine had nowhere to go and nowhere to turn colorless, so it would turn a dark yellow, and that's when I knew to stop the titration. And from there, I would know how much iodine reacted, and therefore could mathematically determine how much vitamin C was reacted, and then finally determine the concentration of vitamin C in each of the oranges. From there, I did some statistical comparisons. I did a Crisco-Wallace test, and the first one that I did was between polyculture and conventional farms. And so this graph displays on the x-axis the farming practice, so the conventional Trapiche Don Simon and Finca Florencia, and then on the y-axis it's the vitamin C concentration. And I did that, and I combined all of the ripenesses just because there was no difference between them for these ones, so it didn't make sense to split them up. And when I combined them, I saw that there was actually no difference in the vitamin C concentration between them. So there was no significance between conventional and Trapiche Don Simon, and then conventional and Finca Florencia. So it was all about the same. Sustainable farming did not have more vitamin C in this case. And some reasons I thought for that was that the variety of oranges is a much more robust variety than other varieties. So the variety is Naranjas Criollas, I think, and these oranges are native to Latin America, or originate from there, and they're not selected for sweetness or juiciness, and so that may also mean that they are less susceptible to slight changes in the nutrient content between the different farming types. And so another reason is that the monoculture, to start, we didn't know where the oranges came from for conventional. They don't say it, Super Compro. So we just assumed that they were from a heavy monoculture, but in Costa Rica, they're pretty environmentally conscious, so maybe the monoculture looked more like a polyculture than like a monoculture in the United States, which is on the right, which is just lines and lines of orange trees. So that not extreme difference between the monoculture in Costa Rica and a polyculture might also cause less significance. The next test that I did was comparing the certified organic to the other farming practices, and for these I did find that the ripeness had a difference between the vitamin C concentration, so I split them into categories. My first category was with the green oranges, and the Whole Foods Organic had significantly less vitamin C than Finca Florencia, a polyculture. So That was very interesting. And then for the yellow, green, oranges, the conventional also had significantly more vitamin C than the whole foods, and the same goes with the Finca Florentia, had more vitamin C. So overall, there was less vitamin C in the certified organic, which is interesting. The yellow oranges I didn't mention here, but they had no difference, so I didn't think to add them. And some reasons I think for this are that the Whole Foods market had a different variety. They were a lot browner and larger than the other oranges, and that likely may have caused a difference. This contradicts other research. So Valencia oranges are highly bred for sweetness and juiciness, and they found that organic Valencia oranges had more vitamin C than conventional oranges. Yeah, so that was interesting. And then in conclusion, the polyculture farms did not have significantly more vitamin C than the conventionally grown ones. And then the certified organic oranges, they had significantly lower amounts of vitamin C due to being a different variety. However, you should still purchase organic foods and sustainably grown foods. It is a lot better for the environment, and that consequently does improve human health. It reduces the amount of natural disasters and like fires and all sorts of pollution and things like that. So it is still super important, and it will impact your health to, like, positively to consume and purchase sustainably grown foods. And then future research should explore ways of making sustainable agriculture more accessible to everyone, so that research like mine are not needed and anyone can already choose the sustainable alternatives. And I would like to acknowledge Naomi and Federico for being my advisors, my homestay families, Yoni and Oscar, see you back there, the Institute, and Finca Florencia and Trapiche Don Simon for letting me use their oranges, and then also the animals that kept me company.
Thank you, Sonia. Are there any questions?
Yes, Katie?
Did you taste test the oranges, and if so, did you notice a difference?
I did not taste test the oranges because I was a bit on a time crunch, and they were also in the lab when I was cutting them open. And when he was working on E. coli next to me, so I decided it's probably best to not do that. Yes.
Joyce, go for it.
Oh, Joyce.
Sorry, could you go back to the first graph you put up? Yeah, so the ones you have conventional and those one, I'm wondering if there could be a difference in the soil type, because Trapiche and Simon and Finca Florencia are nearby, but you know how in coffee it's really affected by the soil type, so I'm wondering if there could be some, I don't know what it is, mineral or something that
Yeah.
affects the soil type.
It could be. Yeah, I don't know where the conventional oranges are from. I think they're from a northwestern region in the conventional, and that may have a different soil type, or like it may not have that.
It's not a huge difference.
Yeah.
You can't see that the spots sort of.
Yeah, they may also just be to like error by my part because it was hard to end the titrations at the same time each time. But yeah, I think that yeah. Yes.
Do you think some of the possibility that the Whole Foods had less vitamin C was because it's grown in like one of those, like, even though it's a different type, it's in the monocrop kind of style, even if it's done organically?
Yes, that could very well be. It could be, another alternative. Yeah. Yes.
This is the iodine, so is that an acid you're adding?
It's not necessarily an acid base. I just pulled a picture up from the internet. It's called an iodimetric titration. So the iodine will react with the ascorbic acid and the vitamin C.
Okay, we have time for one more question.
Yes.
Do you think there are other ways that we can measure the quality, like other vitamins or minerals?
Yeah, initially I also wanted to test to see if there was more beta carotene in like carrots and stuff like that, which is what makes carrots orange. And using that, you could do beta spectrophotometry, which is like measuring the absorbance of the of the compound, and you can kind of measure the the concentration in short. Unfortunately, though, I did not have access to those resources.
Again, thank you, Sonia. next speaker is Clara Polsky, and she's going to be talking to us about differences in gas emissions in cow manure.
Okay. Hello, everybody. My name is Clara Polsky, and welcome to the stinkiest investigation of the day. However, I feel NARC may give me a run for my money there. Okay, well, today I'll be talking about the differences in gas emissions from cow manure based on diet and manure moisture content on dairy farms here in Monteverde. So, just for some background, dairy cow manure, burps, and farts are all significant, have a significant contribution to greenhouse gases, and animal agriculture contributes to 15 to 24% of greenhouse gases in the atmosphere. Methane, the primary gas from agriculture, is released from cow manure through a process of anaerobic decomposition. Studies have shown that grain feed increases methane emissions, and grain-heavy diets are digested differently from grass or hay diets. What happens is that different microbes produce different amounts of methane within the rumen, and within manure specifically, different diets cause different ratios of carbon and nitrogen, which in turn affect the amount of methane that is being emitted. It's been shown that grain-heavy diets produce 42% more methane than low-grain or high-forage diets. And as for rainfall, moisture content has an impact on gas emissions. Wet manure creates anaerobic conditions that favor methane-producing microbes, which lead to higher emissions. Which leads to my research question of what effects do cow diet and moisture have on gas emissions from cow manure? And my hypothesis is that farms with higher grain to grass ratio will produce more gas, and the higher the moisture content, the higher the gas emissions will be. So during my project, I visited 3 farms, all ranging in different levels of being regenerative. My first farm here is Finca Paraíso, which has been regenerated for 5 years, and they use some concentrate, which is kind of a mixture of corn, molasses, salt, minerals, stuff like that. Second farm is Finca Beo Amanecer, which has been regenerative for 3 years and uses kind of the same kind of concentrate, which is they just feed it while they're milking the cows. And Finca Pablo Herrera, which is my conventional farm, which uses concentrate but also uses a silo of corn. So, starting my methods, I collected 20 samples, 8 samples from Finca Paraíso, 3 samples from Finca Beo Amanecer, and 9 samples from Pablo Herrera. And basically I would watch the cows until one of them defecated, and then I would approach the manure and scoop it up in a plastic cup of that was 162.5 milliliters, and then use a popsicle stick to level the top. Then I funneled it into a container, and I did this for every single one of my samples. However, not all my samples were exactly the same. For each 3 farms, I sampled 3 different cows. And for one of each, I did do just solely only manure, but then for other 2 from each cow, I added a ratio of 1 to 0.25 milliliters manure to water, and then another that was more water, which was 1 to 0.75 milliliters. The 0.25 milliliters of the 0.25 ratio, what ended up being 40.62 milliliters of water added to my initial 162.5 milliliters of manure. And then with more water, it was 121.88 milliliters. And then I sealed the top with a balloon and secured it with rubber band. Then in the following days, I had a colleague hold hold the top of the balloon, and then I took a string and wrapped it around the largest part of the balloon to measure the circumference, and then brought it over to a ruler. So for my results, some of the balloons increased, but also some of the balloons decreased and kind of like pulled in on themselves. There's also seen here there was there was a mold that grew on some of them. Okay, so for the no water results, my data was a little bit variable. However, there were some trends. It ranged from 3.5 centimeters to 12.5 centimeters. And there are two things I'd like to point out. One is here, the orange lines from the Microbiota Menacer. They all either stayed relatively the same without much change, or they decreased, different from the other ones. This could possibly be because they were a different breed of cow than the other two farms. Also, the second thing I'd like to point out is here, the triangles on the red line indicate that it's from the conventional farm, Pablo Herrera's farm, and the triangle indicates that it was the first cow that I looked at. The other shapes also indicate, like the triangles, cow 1, squares, cow 2, and circles, cow 2. Then as for some water and more water samples, there was quite a split in the findings from both of them, and there was some split and separation. The range was from 3.25 centimeters to 10.25 centimeters from some water, and for more water it was ranged between 5 and 10 centimeters, which does not necessarily align with my hypothesis really at all, simply because I had hypothesized that the more water would mean greater circumference, but that did not happen. The no water had the greater circumference. Then, for my discussion, the variability could be explained by the fact that, like humans, all cows are unique and have their own unique digestive systems. And also, they're all pregnancy, their time along in their pregnancy was all different, so possibly that could have some effect. As for the switching, the lack of consistency, especially with the more water, how like the third cow from the conventional farm kind of swapped. Like it used to be, the circle one used to be at the top in both of them, in the no water and the some water, but then switched to the more water. This kind of inconsistency could possibly be attributed to there simply being too much water in my third sample, size section. Then also, as for the decreasing of the balloon circumference, that could possibly be attributed to aerobic decomposition, where simply the manure was using up oxygen that was already in the container. And similarly, gas absorbed, there could have been, the water could have been absorbing gas as well. And then the concentration levels could be one thing. Although there was not a lot of concrete conclusion from the diet, there at least was that one example of the cow from the conventional farm that was greater, that had a greater level of gas emission than the others. And as before, there was, the different breeds of cows could have had some change from the but just kind of a conclusion, it's just that though I got variable results, it encouraged me to investigate lots of different possible conclusions that could hopefully encourage future investigations. And I'd like to acknowledge all the people who made this possible, especially my advisors and the people who, and the owners of the farm, Lavenda Vargas, Gretel Cruz, and Pablo Herrera, who allowed me to come to their farm, and of course the cows.
Thank you, Clara. Are there any questions? Go for it, Cassie.
Did you ever accidentally get food all over you? I did. I got it on my beautiful bracelet that that Sonia made me, and I had to throw it away.
Other questions?
I have a question. On your graphs, farm B disappeared on your second two graphs, showing water content. Why?
I just didn't pull data from them. My main focus was for 3 different samples of 3 different locations or levels of regenerativeness for specifically diet rather than moisture. Moisture was a little bit of an add-on of just to see the two most extreme ends. Right?
How long did you normally have to wait for a cow to poop?
Honestly, not at all. They were all pooping.
So if cows produce more methane when they are fed concentrate, why not just stop feeding them concentrate?
While they're pregnant and they're needing to create create a calf within them, the concentrate really does honestly add to the health of the cow. Like it offers sugar and energy for them that things like just simply grass while they're in the pasture isn't sufficient enough. So it still is needed. It could be thought about how it's more about the issue of adding on grain and corn rather than trying to eliminate concentrate.
Any other questions? Okay, thank you, Clara. Okay, so our next speaker is Olivia Palanconi, and she's going to talk about economic trade-offs at these same farms, actually. So,
no, it's all there. It's fine. The thing is on.
you gonna go?
Yeah, it's gonna be plugged in. Do you want suspense? I'll be in there for a second.
That's for the clicker. So, can you just click out on your?
Okay. Good evening, everybody. My name is Olivia Palanconi, and today I'm going to be talking a little bit about my study of the economic trade-offs between profitability and environmental sustainability here in Monterrey. To give you guys a little more context, dairy farming has been an integral part of Costa Rica since Spanish colonists brought it here in the 1700s. And today it's evolved to be a key agricultural sector that contributes a lot to food security and the economy. However, recent shifts towards sustainability, like Costa Rica's goal of 0 carbon emissions by 2050, have prompted farmers to adopt more sustainable practices here on their farms. Which led me into my question of what are the economic implications of adopting regenerative versus conventional practices, and how do farms balance profitability with sustainability? And I pretty much am going to explore how different farming practices impact financial performance as well as ecological health. And then, sorry if you get a little deja vu, Claire and I might have visited the same farms. So I went to 3 different farms, Finca Periso, which is a very small regenerative farm, and they have about 19 cows, and they currently are milking 7 of them. And then we also went to Finca Beba Manicer, which is slightly bigger. And Finca Periso has also been regenerative for 5 years. And then Finca Beba Manicer has been regenerative for about 3 years. And they're slightly bigger. They have 27 cows and are currently milking 13. And then lastly, went to the larger conventional farm, Finca Pablo Herrera. And they have 70 cows and they are currently milking 32. yes, these are all the and I interviewed all the farmers here, which was part of my data collection. And so in my interviews with the owners, I asked them about 3 separate categories. The first being the cost. So I looked at what they were feeding the cows, if there was any like external labor, all the utilities they use, and like all the transportation necessary. And then I looked at the milk production per month for each of the farms. And then lastly, I looked at operational inputs such as energy and water use, and then water usage, and then also the sustainable inputs for the regenerative farms. And then with all that data, I conducted 2 analyses. The first, the break-even analysis, which identifies the production needed to cover costs. And I did this by compiling the inputs and outputs, and then dividing that by the milk production and also costs and revenue. And then I also looked at the profitability analysis, which has 2 parts. The first, the gross profit margin, which is pretty much how do we create efficiency in generating revenue relative to production costs. And then the net profit margin, which is just overall profitability with all the inputs and outputs from the farms. And then with these analyses, we're first going to look at my results for Finca Parrot. And this is the graph from the break-even analysis. And the pink line shows the total revenue, and the green is the total costs. And then this point right here is called the break-even point. And that pretty much is the point at which the farm starts to make profit after they've paid for all of their inputs. So yeah, and the break-even point for Finca Parrot, I calculated to be 328 kilos of milk. So that's the amount of milk necessary for the farm to make a profit each month. And then this graph looks pretty similar. This is the graph for Finca Parrot, and it's practically kind of the same thing. You can't really tell the difference too much just by looking at it. But the break-even point for this farm is 539 kilos per month. And then here is the break-even analysis graph for Finca Pablo Herrera. And as you can see, it's much larger. And yeah, so the break-even point is 26,000 kilos of milk per month, which is much higher than the other 2. And yeah, so I'm going to talk a little bit about, profitability analysis. Yeah, so I can't forget about that. And then, so here is the revenue, direct and indirect costs for all 3 of the farms. And just by looking at this, you would assume that Finca Pablo Herrera is making the most revenue just because the graph looks like that. But once you did the GPM and MPM analyses, you can see that Finca Pablo Herrera has a much lower, not only GPM, but also NPM. And NPM is the real indicator of total profitability between the farm, and especially in comparison with the most regenerative farm, Finca Parrot. So it is significantly lower with about 12.5% compared to Finca Parrot's 74%. Okay, now I'm going to talk about the discussion analyses for break-even analyses for Finca Parrot. Okay, so with the break-even point of 328 kilos of milk, the farm is currently actually producing 2,520 kilos of milk per month, which is a surplus of about 2,200. So obviously this farm is making some money each month. And this can be attributed to their full adoption of regenerative practices, specifically relying almost entirely on pasture feeding, which cuts costs for any feed needed, and natural fertilizers. They do not pay for any fertilizers on their farm, which also cuts costs. And they do this using their closed loop system, which is a key regenerative practice, and it eliminates the needs for chemical inputs. And yeah, so they're able to produce far beyond their break-even point, highlighting the economic resilience and the ability to stand market fluctuations and climate change. And then similarly for Finca Parrot, they use a lot of the same practices. They've just been doing it for less time, so that might be why they have a higher break-even point, but they're still making much more of a surplus, with their surplus of about 2,500 kilos of milk a month. And so it might be a little higher because they've had less time to fully adapt to with all the regenerative practices. And another limitation to the study is that they use Jersey cows, which is a smaller breed and produces less milk a month compared to the Holstein crosses that are present at the other farms. And then looking at Finca Pablo Herrera with a break-even point of 26,000 kilos of milk a month. When I conducted this study and I talked with Pablo Herrera, currently in that month, he only produced about 1,700 kilos of milk a month. And we can attribute that to the tropical storm that lowers milk supply production in the cows, and also he had to pay a lot more for labor on the farm because a lot of his fences needed to be mended, and he had a lot of destruction from the storm on his farm. So that's why his costs were higher that month. So hopefully he's not always having such a shortfall in production. But yeah, so this shortfall and this larger break-even point means that this farm is struggling to generate enough revenue to cover all of its costs, making it much more sensitive to fluctuations in feed prices and other market conditions, as well as impacts from the climate. And then to discuss the profitability analyses, Finca Parrot and Finca Beo Manizar had high GPM and NPM, which indicates that they have a lot more resilience and are just making a lot more profit in comparison to Finca Pablo Herrera, which had lower GPM and NPM. And this just shows that this regenerative practices do work and that they are economically and financially viable. So yeah, which leads me to my conclusion that regenerative farming practices do offer an advantage to Costa Rican dairy farms economically and sustainably, which is good for everybody at the end of the day, and that they are not mutually exclusive, but require careful cost management and strategic investment in all the farms. And hopefully that more farms are inspired by analyses just like these, and that more education can be offered to them, to the public and the farmers, so that we can help them convert to regenerative practices, and the world can be a better place economically and sustainably. Naomi, thank you so much for helping with everything. You were like the biggest blessing in this project, and also Federico for all your support. Thank you so much as my secondary advisor. And of course, all the farmers, thank you for sharing. Thanks to them for sharing such valuable information with me. It was super interesting. And yeah, and Clara for the pictures.
Thank you, Olivia. Are there any questions? No idea.
The farmers that you talked to that have switched to more regenerative practices, did they talk at all about kind of the effort that they have to put in, or how pleased they are with it? And then my second is just if you know anything about scalability, like building smaller and what it might look like.
So the two, Orlando Vargas and Gretel Cruz that I talked to, they were very pleased with their transitions to sustainability. Gretel specifically mentioned that she immediately noticed she was making more money per month, and that when she was practicing conventional farming practices, she was losing money each month, and she didn't even realize until she reevaluated her finances and realized that she was. So she's definitely very pleased. They also talked a lot about how they noticed a lot of improved health in their cows, and how that had cut back on vet costs and stuff like that. And then in terms of scalability, that's definitely a big problem, because I truly understand how it's much harder for larger farms, because they just in general have much more inputs and outputs. So yeah, scalability is definitely an issue. I think in future studies, it'd be very interesting to do a cost-benefit analysis where you determine the costs that would be required to switch to regenerative farming. I just unfortunately did not do that. So yeah.
By the way, that's my sister Noelia, so anyone who wishes. Frank.
Actually, Will has his hand up too.
Go for it, Will.
Did you or do you plan to show this to Pablo Herrera?
No, I hope he never finds out about this. I just don't want to embarrass him, but he was so sweet. But yeah, he should have some education. He should learn about regenerative practices.
it could benefit.
Go for it.
Two things. One, a comment related to that. The first question is, are other inputs like doing farm tours like you folks did and think about
Yeah,
was
so-
that included in income?
No, I did not include that. It was strictly just the dairy production to keep
Okay,
it.
and the second thing is, and you might want to get help from people who actually know about this. One is to possibly change names in your paper. That would be the first thing. And then the second thing is, people have the right to know what you
Yeah.
concluded. However, it probably is not well advised to promulgate it to the world, because the next time they go to the bank, they may not be able to get a loan because you've shown they're crashing. So let's talk about that.
That was just 1 month. It was just 1 month. I want to be clear. That was just 1 month for Pablo.
Yeah, but this is another important thing, and that is, honestly, there is a need for helping people figure out the viability of whatever they engage in. Having restaurants, having pensions, Airbnbs, so they can make informed decisions about what's reasonable, what's doable. So it's an awesome study, but you got to be careful with it.
Thank you, Olivia. Are there any Okay, our next speaker is Bjorn Kishinevsky, and he is going to talk to us about a color analysis in primary and secondary forests.
Color is more than beauty. It means life. From the vibrant toxicity of the blue poison dart frog, whose aposematic coloration blares warning signals to its predators, to the mesmerizing beauty of orchids, who attract pollinators so lustfully they don't even need to offer pollen in exchange, to the male violet saberwing, whose bright blue plumage attracts its female mates, to the inconspicuous camouflage of the to the inconspicuous camouflage of the wood frog, which blends into its environment. Color plays a vital role in nature. And before we dive into its potential implications to understand climate change, let's dive into how we as humans perceive color. We perceive it across 3 axes. Lightness, which is how bright the color is. Its chroma, which is how intense or saturated it is, how far it is from a neutral gray. And then we have its hue. Is it red, green, or blue? I invite you to take this moment to observe what colors you see in your visual field now. Isn't it interesting how foundational color is in our lives, but how rarely we notice it? This is similar to the phenomenon of wood in our daily lives, how much it surrounds us, like in this room, but how rarely we recognize it. But this wood has a hidden cost. And in fact, by the time this symposium is over, which is eagerly soon, there will be 1 million more trees cut down. But who cares about a few scrawny green leaves? The millions of species living in those forests. Now you might be thinking, Lord, why in the world are you asking what is the most common color in nature in a place called Monteverde. Well, Monteverde, as you know, underwent significant deforestation in the 1950s to 80s. And it created two types of forests that we see. Secondary forests, like here in the San Luis plot, which was deforested, recently purchased by the the Monteverde Conservation League. And then we have this primary forest in the back, which is untouched. And this leads us to the two questions of the study, which is what are the colors in secondary and primary forests, in tropical humid cloud forests, and what might these colors tell us about their ecological health and biodiversity? So my research explored two reserves in pre-montane and montane tropical humid cloud forests. The first was the Rachel and Dwight Crandall Reserve and the Reserva Uricancha, which both offer a mixed, sort of interdispersed primary and secondary forest composition. The first part of my study was understanding what is the differences. So secondary forests, as you can see, is primarily characterized by smaller, homogeneous, thin, short trees with a lot of visibility throughout, and then not an interconnected canopy. Whereas primary forests, those which have been left untouched, are typically characterized by a developed canopy, a triple layer tree story. We also see a diversity in tree widths and heights, and then we have an abundance of epiphytes. Now, my study employed a novel technique called color quantification, which analyzes images and then extracts their colors. So, for example, we can take this primary forest image here, run it through an image color summarizer, and it tells us the 10 most prominent colors and their proportions. So, so if we look across the 284 photographs, we obtained 8,520 unique color metrics. After analyzing them, averaging them, we've created two forest-specific color palettes. But before we dive into that, I want to ask you, what do you think is the most common color that was found in primary forests? Green. Well done. Yes, it is green indeed. More specifically, we have a light chroma and hue value as C. Now, what do you think is the most common color found in secondary forests? Is it lighter or darker? Lighter. Yes, well done. And it has these LCH values. Now, very interesting to note is that the chroma and hue values for both are practically identical. It is just in their lightness, where the secondary forest is about 170% lighter than primary forests. Now, let's dive into those palettes to get a deeper look into these two forest types. These are the 12 most prominent colors in the primary on the top, secondary on the bottom, ordered by their light, chroma, and hue values. Now, to dive in a bit deeper, these are the color palettes for the 30 most prominent colors, sorted by their proportionality. Now, diving into the primary prominent colors, we can see that primary forests offer a slightly deeper, darker color palette. And this could be explained by the more developed canopy, mature vegetation, as well as greater shade. Whereas the secondary forest's lighter palette, especially in the beginning, is probably due to those gaps of light. Now, future studies should definitely look into exploring more reserves across different life zones and across varying times of day. Now, it is with great gratitude that I have to thank you to Naomi for your consistent support throughout this whole process, to Natalie Sanchez for your positivity, for Federico for being you, to Frank Joyce for orchestrating this entire program, and to the owners and founders of the 2 reserves that I got to explore. Now, in the powerful words of Coco Chanel, the best color in the whole world is the one that looks good on you. After doing this study, I think that a dark, deep green looks really great on the tropical, humid cloud forest. And I say, let's keep it that way.
What's your favorite color?
Green!
Can you talk about the way that this can be used to assess the ecosystem health?
For sure. So I think the main point is to see that primary forest, at least in these life zones, is a much darker and deeper color. And so we can use that sort of as a baseline for what good ecological ecosystem health looks like and what we're trying to get to. Yes?
Did we use a website or app or something like to get the colors?
Yes.
Okay. Did it take into account how much light was hitting the leaves?
So, you might be able to see here, 3.72%, I'm assuming is coming from these light gaps. So.
Is there a way to translate them so that they're all the same lightness? To get the same color. I don't know. So one of your, did you just use, okay, how about this? Did you just use that picture for your sample?
For this?
Yeah.
For this, yes. These are
And that created your gradient?
Well, I used, so across 284.
Is there a way you can account for the exposure of the camera?
Yeah.
Ah, great question. Yeah. So what I did was, well, first my methodology was along the trail I would take a picture in all 4 directions and then also one on the soil. And then what I would do is I would look back and forth between the image, the actual environment in the camera, and I would adjust the exposure level to be as similar as possible. Yeah.
Do you think similar analysis can be used for different items, or is it only applicable to colors?
I think it would be really interesting to explore how other forests explore. What is the color palette of Isla Vista?
What is color?
Wow. Did you hear that question? What is color? If you want the color is basically energy hitting the photoreceptor cells on our eyes, which is basically photons, particles of energy that are vibrating, and then they collide. And when they collide, they either get absorbed or they get reflected. And the light that gets reflected, that energy, that's what our eyes can perceive.
That's not what I'm asking.
Well, that's your answer.
Any other questions? Okay, thank you,
I hope you're still listening because I hear they saved the best for last. Yes. Okay, so hello, my name is Allison Flax and I will be presenting on the perceived perception
or the perceived relationship between humans and hummingbirds in Monteverde. So in recent years, ecotourism has made immense impact within society. And ecotourism can be defined as responsible travel to natural areas that conserve the environment, sustains the well-being of the local people, and involves interpretation and education. And it usually involves nature-based activities that promote environmental well-being. So specifically, I wanted to focus on the Hummingbird Gallery in Monteverde, because Monteverde is a very popular destination for ecotourism because of its rich biodiversity. And the Hummingbird Gallery is a place in which you can find many people who are interested in observing the environment. However, it may have positive and negative effects on the environment and social structures. So my question was, what are the ecological and social costs and benefits of ecotourism at the Hummingbird Gallery? And I was interested in speaking with humans as well as observing hummingbirds in order to learn more about this topic. So I performed a stakeholder study, meaning identification, analysis, and understanding individual groups' interests on a specific topic. And I focused on 3 main groups: tourists, guides, and the hummingbirds themselves. And I spent 7 days at the Hummingbird Gallery, and I used my cell phone to record interviews with 25 tourists and 10 tour guides. 13 of the tourists I interviewed were men, and 12 of the tourists I interviewed were women. All 10 of my tour guides were men. And then when I asked background variables, yep. This is a photo taken by my lovely homestay father, who is also a tour guide. I started with background variables like age and education, and then I asked similar general questions to both groups about hummingbirds and the effects of tourism on conservation in Monteverde. I spent 180 minutes observing hummingbirds in 10-minute increments, and I would look at 1 feeder at a time, count the number of visitations, the number of aggressive behavior I saw, and the amount of plant visitations I could see within approximately 10 feet. And I divided my 3 hours of observations into when humans were present versus when humans were absent, and then I compared the data using a t-test. So what I learned from my background variables that tourists were highly educated on average, with 11 of the 25 tourists graduating with an undergraduate degree, and 8 out of the 25 tourists graduating with a postgraduate degree, and the other 6 had not exceeded high school. And then I also learned that a majority of tourists had previously seen hummingbirds, with only 7 of the 25 tourists I interviewed never seeing hummingbirds in their lives. And then finally, I learned that most tourists mentioned nature or wildlife as a reason for coming to Monteverde as their travel destination, with only 33% of responses mentioning fun or leisure. So here are some graphs that have to do with how hummingbirds are affected by tourists and how tourists are affected by hummingbirds. The dark green bars are tourist responses, and the light green bars are tour guide responses. These graphs are shown in percentage of responses rather than percentage of participants who responded, because each participant was allowed to give more than 1 answer for each question. So as you can see, the way that hummingbirds are affected by tourists, a lot of tourists thought it was either behavior or there was no effect. And tour guides mentioned mostly behavior, health, or no effect, where health was only mentioned by tour guides and wasn't ever acknowledged by tourists. On the contrast, the way tourists are affected by hummingbirds, both groups mainly focused on how it invokes a feeling of joy or an interest in nature in humans. Here are some direct quotes in relation to how tourists are affecting hummingbirds. One tourist said they're not afraid of people at all, and they kind of fly right up to them, while one tour guide said, I don't know what kind of disease they contract or what kind of health situation they have, but I think tourism is affecting them in a negative way. And then how tourists are affected by hummingbirds, how hummingbirds affect tourists. One tourist said, I just think they're so beautiful and interesting, and a tour guide said they learn about special kind of bird that flies in a different manner. And then here are some more graphs about a more broad perspective of conservation in Monteverde. So I asked, what are the benefits of tourism in Monteverde, where most of the tourists replied saying the economy or the environment was benefiting off of tourism. And with the guides, they mentioned the economy and education the most. All 10 of my guides mentioned the economy at least once in their answers. And within the issues behind tourism in Monteverde, most tourists said either degradation, disruption of ecosystems, which once again was never mentioned by tour guides, and then pollution and the economy. And then tour guides mentioned degradation, overcrowding, and the loss of culture, and this idea was also never mentioned by tourists. And so, again, some direct quotes. What are the benefits of tourism in Monteverde? A tourist said financial stability for the people, the environment, and the animals. And a tour guide said 70% of the population in Monteverde depends on ecotourism. And then what are the issues behind tourism in Monteverde? One tourist said pollution, noise pollution, light pollution, air pollution, garbage, environmental degradation. So I think that displays a thorough knowledge of ecocentric ideals and understanding their ecological footprint. And then a tour guide said tourists affect their culture and completely change our culture. We are losing our culture. And this was my one question that was different for the two groups, but still open-ended. So I asked the tourists, are you an eco-tourist? And I asked guides, are the tourists you meet in Monteverde eco-tourists? With a majority of the tourists saying yes they are, or they try their best to be, and only 28% of them replied that no they're not, or they don't know anything about that. With the guides, they said either yes, all of the tourists they meet are eco-tourists, or most of them were. So none of the guides thought that a majority of tourists who come here are not eco-friendly. And then for my hummingbird data, I identified 6 species, and I saw no changes in visits to feeders or aggressive behavior. However, I did see a difference showing that it was more likely for hummingbirds to visit plants when humans were not present. So I think this kind of shows me that a lot of the views of tourism in Monteverde are positive, and I think that I learned that tourism really benefits the economy in Monteverde, and I can also infer that a connection with nature and higher education leads to more care surrounding environmental issues, and this is something that has been studied and suggested before. And I also found that a majority of tourists who visit Monteverde, specifically the Hummingbird Gallery, have ecocentric views in mind, and Monteverde is a hotspot for environmental tourism because of its rich biodiversity. However, there are still adventurous and leisure activities that people like to do. And finally, I was able to find that tourism may have ecological impacts on hummingbirds, specifically relating to natural foraging habits. However, I think those social benefits that are gained from it outweigh those completely. So why does this matter? Tourism is a major economic stimulant in Monteverde, which has shifted it to depend on the replanting and conservation of the forest rather than cattle ranching and deforestation as a major financial resource. And I think this study helps to ensure the sustainability of tourism by inquiring about the perceptions of tourists. I was able to learn that a majority of tourists in Monteverde most likely care about environmental issues and are aware of their ecological footprint. And this leads to the question, what could be more sustainable with tourism in Monteverde? And I think maybe we could adjust feeder placements so as to encourage more natural plant visitations. And we can also continue educating tourists so they feel more connected to environmental issues. And the more awareness is spread, the more likely it is for tourists to be conscious of their actions. So in conclusion, my study underscores the interplay between humans and wildlife, showing that there are mostly positive social impacts of tourism, and it can be sustainable if managed and controlled properly. In Monteverde, parks should continue putting in place capacity limits to conserve natural landscapes, and this kind of control can positively benefit sustainable tourism. Finally, I would like to thank Naomi Solano so much for being the best primary advisor I could ever ask for and answering every question I ever needed, and Natalie Sanchez and my father, Mark Flax, for also being advisors to this project and helping me figure out how to do a sociological project in a biological program. And I'd also like to thank Frank Joyce and Federico Chinchilla, the participants of my study, my homestay parents, and the Monteverde Hummingbird Gallery. Thank you.
Thank you. Gally, are there questions? Go for it.
In your graph on how do hummingbirds affect tourists, one of the things you had on there, the how do hummingbirds affect tourists? Fear.
Yes.
How many tourists are scared of hummingbirds?
Yeah, so quite a few tourists I interviewed were genuinely scared of the birds flying past their ears, and it is a little jarring when you get in there and you're in between 3 feeders and a hummingbird goes right past your head and you've never had a hummingbird fly past your head before, or even if you had, maybe you're just sensitive. Claire.
What was the most funny or interesting or unique response you got from someone?
It was that maybe it's not so normally in Costa Rica to see 2 boys kissing each other. Maybe that's not so normally for us. Don't know how that came up. Welcome.
What do you think can be done in terms of the concerns about losing culture? Like, how do you think that tourists can be more responsible and what kind of work can be done there to make sure that Montevideo isn't losing its culture?
I think that has to do with education. So I think most of the tour guides who mentioned a loss of culture said something about how people will come here and want to do things like party after they've gone out and seen nature, and that's just not really what this town is for. Like, you're kind of coming here for nature and that's it. Well, they said Barmigos is rustic. Actually, Oscar Fennell right there, he said they should come here for Barmigos, it's rustic. They're not looking for a disco. So I think that, I don't know, it's hard to maintain a culture when there's so many people coming here, but I think through education and understanding things like that better, you can promote culture, keep it in place. Yeah, Leo?
If, now that you've ran a sociological study, if you were to do another one, is there something that you will keep in mind or that you will do different?
Yes, during my first few interviews, I was asking a lot of leading questions, which I totally omitted from the data, but I would say things like, I would ask, how do you think hummingbirds are affected by tourists? And they would say, I don't know. And then I'd say, okay, well, do you think their behavior changes? And they'd say, oh yeah, probably. So then that's me leading them into that, and so I stopped doing that towards the end. And I realized that was not good. And I omitted all that data. And if they said, I don't know at the beginning, I said that they didn't know, or they said no effect. That's what I did.
Did you interview the penguins?
The penguins?
Yep.
Oh yeah, sure does.
Let's beat your beer if you're on. So hold on to it. Listen, everybody. Woo!
Not getting my act. It's a continuation of the DDT, but nobody's listening. I'm going to do this in 2 minutes. Normally, it takes about 3 hours. So I'm just going to say a couple quick things so that you can get on the bus and get back to the station. 10 weeks ago, we were meeting for the first time in the Hotel Cox. And this is always the case, but the script for independent projects is not written. And today we found out what you managed to pull off, what you produced, and it was really, really impressive. Huge diversity of things. Leora, you left me speechless, without words. I'm still processing it. Other people did projects that took a ton of effort, and they're convoluted and complex, and I might understand them in a few years. Other people did simple, elegant projects, but it was all a great, great mix of awesome stuff. And I would just say simply that thank you, students, for putting your effort into this and sharing this with us today. I would also like to thank Irene for doing the live stream. I'd like to thank Joyce Letton for translating. I'd like to thank the Monteverde Institute and many other entities. We'll talk more again on Saturday night. I will briefly mention other individual people like instructors and services coordinators like Federico, Natalie, Emmy, Naomi, Francini, and Paola who make this happen. And but especially to you, the students. We've been sort of trying to pull things together. It's been a challenging program on any parameter, so I'm also coming out with a high or low, which is a p-value of yes, this was a challenging program. Yes, for you and for us. The weather looked like, you know, heck, we're not going to do projects, we're just going to do things on Google, whatever. But you pulled it off, the weather cooperated a little bit. And like I said, it was really, really great effort on your part. You guys are troopers. And a couple other logistically announcements. I'm going back on a personal resolve, but I will send up the projector just in the off chance that you want to watch something. But I'm holding you, Grace, personally responsible for the following. You have to use the fan so it doesn't blow out the bulb, and you have to put it back after it cools. And if it's not done, you're responsible.
Survivor at 7 p.m.
You won't have eaten dinner by 7 p.m. So the other thing is, listen up. Advisors will appear sometime tomorrow at the station, but don't lose your paper copies, okay? Keep track of them. Don't say, oh, the dog ate it or the pisote ate it. Please keep track of them, okay? And I got a text message that says, Bar Amigos has been closed because there's no water and there's no liquor at Bar Amigos. So it might open after you leave on Monday the 16th, but yeah, it's closed indefinitely, I think. That might be false information. But